收音好好啊，我哋應該唔使開聲。
係咯
Can we test the audio of this room so that we know that it's going to be？
你有一個。
佢哋 boardroom 個 account 好似未入嚟，
所以我而家幾 set up to make sure that 唔會撞咯。你等等。
boardroom 我都仲未入啊？佢應該有啲講。
係啊，佢哋有啲。有一個 message 我 reclaim host， 我唔使 reclaim host 係咪啊？
我唔使，而家我係個 host 啊嘛。
我 stay co-host 唔使係咪啊？
係啊。
所以我 off 咗佢㗎啦。
係。
你點樣俾嗰個？你點樣俾嗰個黐嗰個 host？ 你俾起我哋嘅 login details 係咪啊？
我唔使啊，我直接喺個 Zoom 入邊可以 assign 去做 host 嘅。
OK。OK， 等啊，佢哋 set up 緊一個 sound， 我哋 retest just bear with us。
我都要等佢 join， 即係個 boardroom 個 account join， 跟住我要 promote 佢個 account 做 attendees。
咁樣 OK 啊
？OK 啊，
冇聲，全部。
係暫時我用聲，因為佢哋未學，我哋埋暫時嘅。而家我。
I saw Matt before.
No, I think this is the first time.
哎呦唔得，所以我
有人。
OK， 而家聽唔聽到我哋？
有 echo 啊，你哋。
係啊。Everybody, do you have any microphone on other than the only them?
Muted.
Right
now, the only microphone. Somebody's.
You turn this.
Computer
而家好唔好啲？而家好唔好啲 ，Martin？
Computer voice 就。
Martin， 你有 echo。
唔係而家有冇啊？而家應該 OK 㗎啦。
而家好似咦都係有 echo 喎。
我哋就聽到冇喎。
你聽到你 OK？ 而家 try again testing。OK or not?
OK， 而家 OK。
Good. I
think it's because the door. I think people.
It's not you. 我哋每個 close 咗呢個。
Computer voice.
OK, good.
Computer speaker.
Good.
No。
你講啊 ，Martin。
因為我諗 delay 得太犀利啊，因為咧你哋係
delay， 所以有 lag time 係咪啊？係啊，
我諗 delay 咗30秒左右咯，所以我會聽到自己把聲喺你哋個 speaker 度。
There's a lag.
I'll use mine. Mine already locked.
Why is there a lag? He said there's a delay. Why is that?
冇30秒咁誇張啊。
Wi-Fi issue. So, his delay means it's slower than your phone.
That means that when we talk, he only hears it after 30 seconds.
I think it's 30 seconds。我哋問咗佢都答得到。
Okay, Martin，
再嚟講
Martin。
係喺度。
see OK 啦。
5 seconds, 3 to 5 seconds, not 30 seconds.
There's a delay?
A little bit of delay.
I see, okay.
So Martin, when I say Martin, can you say yes?
Yes.
Okay, Martin. Martin, say yes when I call your name, okay? Martin.
你連續講佢都唔知 yes to which one. Yeah, I think it's okay.
No response until now.
Martin 同我哋講啲嘢啊。
hello，而家仲有冇 delay 啊
Two seconds, only 2
哦
seconds.
，OK，
OK，
No problem.
嗰個 boardroom 係咪有啲暗啊？你覺得好似
係咪係啲光線？因為嗰個光喺度，你直接個鏡頭望住影緊佢咧，係會有前面嗰啲人黑嘅，因為你個光係最後邊光。OK， 知。Boardroom one, boardroom two, boardroom one 係喺即係
Part one is the camera. Is that one?
boardroom one 係嗰個啊？
Yes.
唔會啊，佢
No, boardroom one is coming from the front, so boardroom one should be this.
Oh.
Boardroom two
There's
is
only one
yeah, look at
member.
the screen. So Martin 啊，呢個黐名，你有咩黐？有啲
指明
指明。
指明仲要做乜嘢啊？
我諗之後佢可能如果有啲譬如 audience 問問題或者 whatever 佢可能 control 下，同埋我諗最緊要佢部電腦唔好熄啦。如果唔係嘅話，因為我之後會 assign 個 host 俾佢啊嘛，咁我會走佬噶啦嘛。
You must have your power cord on. Because
I will sit
you would-
over there and
Do you have a power
Okay.
cord?
yes,
Yeah,
I
but
have one.
Can you connect?
Martin said your computer must be
Must
on
be
all
on.
the time, the whole day.
So, is it really a battery consuming task?
Very. So where is your?
Because it's the whole day.
Yeah, maybe I can use lunchtime to charge it.
But we cannot take the risk. So you have
Yes,
to have.
yes.
So
No
do you have a
problem.
cord?
Yeah,
這
I
個
have one.
可以啊。
I have one. And
You keep
I will sit here. Maybe
Because,
later I can.
Yeah, when it's
log-
all.
Yes, but when everybody is all set, I will.
Oh,
Maybe yours, yeah.
Is there another slot here?
冇喐啊，冇喐。
There is one more slot here. You can use a slot here. There's one more.
唔使啊，they have here.
Oh yeah.
It's everywhere. Okay,
Yeah,
it's like
but
everywhere.
I will sit.
Yeah, then you use one of those.
Yes.
Because 佢哋嗰啲係正常，你個插頭嚟㗎嘛，唔使用個 extension cord。佢 sit anywhere，嗰度又有插頭，度度都有插頭。
Okay, but could you make sure that 佢唔可以熄？因為而家佢係 post。
Martin，你咁樣聽唔聽到我同 Winnie 傾偈㗎？
哦，聽到噶。
哇，好清楚，同埋冇 delay 啊，1 second 都冇。
係啊，頭先係咪嗰個
可能佢未醒。
嗰個
係 further down，I think so。
Oh, there's one over there as well. One here.
同埋呢度都，其實你都可以用呢個啊。係你會坐邊度？Sorry，why am I speaking Mandarin？普通話。
Here.
And yours is also bigger。唔係啊，佢嗰個都好大。
係啊。
Use the one on the wall。this one maybe。
但係佢唔會做嗰度。Yeah, there's one more here. You can use this one.
但係佢要坐呢度啊嘛
Too
，too far。
What happened?
Maybe use this one.
Once you get.
Hey, you can sit over there.
There is
So if that's
There's a
over there.
yeah, there are 2 here.
The coordinator. Oh, the camera can catch the time alert.
Catch the time alert. Where are you sitting?
So.
Wait, so the time alert, good point.
Depends on where.
The
So
time
I'm
alert.
adjusting the camera right now. So one is shooting that way, the other is shooting that way. But I'm gonna adjust that one to connect to this podium.
Okay.
So I don't. Where are you gonna sit?
Maybe I do not need to be in the screen. You are the coordinator during the pitch session.
Yeah,
Yeah,
but
Jimmy
so-
doesn't need to be in the.
yeah,
You are going to sit over there.
yeah, I need to be able to see you.
Yes.
I think you can sit over there,
Yes.
but you have to be connected because you are now the host.
Yes. And I will just sit here and.
Martin 啊，嗰個 timer 咧，
係
可唔可以有一個 timer 㗎？
You don't need to.
佢 timer for
因為10 minutes for presentation, 10 minutes for
佢應該 technical support 佢哋之前 set 過嘅喎，佢哋應該識 set timer 嘅喎。你問下阿劉毅啦，即係咯。
漏藝唔知喺邊嚟㗎？漏藝
佢話
誒
we are in，
Acid time
或者你同阿子明講話佢即係佢啲10 minute 嗰啲，佢應該識 set timer 嗰啲。
You know how to set the 10-minute timer and
Yeah,
put it on the
I've
screen？
already
佢要擺喺個 screen 嗰度㗎嘛，啱唔啱？即係我都睇到，佢都睇到㗎嘛。
last time when we start a timer it should appear on the sharing.
要叫佢俾出嚟睇。你同字面講點樣擺啦？
Is it good for the podium?
Yeah, very good.
Okay.
Very good. Who will control like
Camera?
cameras? Yeah.
I'm controlling right now, and then both camera right now are feeding, so both has. So if you look at the,
Hello.
Hi there, how are you?
Good. You are.
Jeff Leiter from Luminary Therapeutics.
Oh, nice to meet you.
Very nice to meet you.
Yes. You are from Hong Kong. You're one of the organizers, and very excited about this morning.
Yeah,
Yeah, we expect about 20,000 people actually
wow,
watching online.
gotcha.
wow,
That's quite a bit.
Yes.
And so it wasn't clear to me whether I get to present or not. Do you know if I do? I got invited and said, come to this pitch. So-
okay. Let me check with. Do you want to check? Do you
So
want
unless
to?
you are invited to present, then you're just a visitor. Because
Okay,
you already got the list of
okay, then I'm probably just a visitor.
yeah. These are all the companies that have taken part previously.
Okay,
Only when you have been with us since like the preliminary rounds.
okay.
Yeah.
Yeah,
But-
so I wasn't part of that, but
Yeah, but
I
yeah,
guess it would be a good group to start to get to know.
yeah, and there'll be investors around as well.
Okay,
To-
try and network, I will.
Sure.
For
Yeah.
sure.
Um.
So just sit towards the back, is that okay?
Yeah, sure. Or here, yeah, anywhere you feel comfortable.
Okay.
Okay, time Adi. Where are you based in Boston?
We're based in Minneapolis.
And you came all the way here?
Yeah, well, I was kind of here anyway, so I
Right,
decided to stay.
Right.
So I was here on Thursday and Friday.
Oh,
And I thought, well, heck, this might be a good group to get to know.
yeah.
So,
Yes, I think next time let us know because we have quite a good exposure of
yeah.
participants come.
And you are?
I'm Wingley. I'm from Hong Kong Science Park.
Okay.
You could call me Wingley.
Okay. Got it. Got it.
Do you have a business card?
I do. There you go.
Oh, thank you.
Thank you.
So we have. I'll just switch to. So I have two screen line streaming.
Right.
Okay.
The CPC application, there's this meeting that what is it called? The APM. That's right. How soon they want this? Because after we get to our trial, timeline, get it. Probably late June or late July. You can organize the APM meeting. Yeah, we'll let you know because I'm rushing my team to make sure the material, our application material, is ASAP, so that once submitted to CTC, then you can help organizing. And we will need your guidance. You will be previewing the application, right? And then you will let us know. Okay. External guys are okay. Industry from like a pharma company. Oh. Nice. Yeah. Okay. Thank you very much. You know, we were looking at the time and I'm like, oh, this needs to be done quickly so that we can make sure that full amount of the trial can be used to take advantage.
How's the timer?
I'm checking. It used to be automatically appear.
Can Martin help you?
The timer is there, I saw it.
Can you see my timer?
The camera I saw is like the boardroom showing the stage.
The timer.
No,
The
can you
timer.
see the 10-minute timer, Martin?
Yeah, right now I can see it's the top right corner.
Yeah, but it's not on the share screen.
But I want a screen that shows a 10-minute timer so that I can see it, everybody can see it. That's what you do for the screen. Can you do it?
It's not. I think it's like quite difficult unless like we do it in those like CPC APM. Someone use their own camera to load a video of a 10-minute timer.
Okay,
Otherwise it will be
Jimmy
very
do that?
difficult to do that.
Can you show Qiming how to do that? Qiming, hi.
Can you share another?
Can you do it on your camera? Martin.
Let me see how I can pitch in.
Martin can help you.
Can you see the timer on your own?
I can't see it.
So,
Usually you have to put it as your own camera. Right, you have to put it as your own camera. See? Like that.
Dr. Chang.
But the problem is the mode we are set right now is like only show the camera that who speaks.
Grace and only.
Yes, I think that will be enough.
I think at the time you
So
would
cool when you
have
control
20
the timer.
20 minutes.
Do you
Yes.
know how to control the timer?
Okay, so Martin, can you show me how to create this video?
But I mean that it's like there's some problematic thing about like using it this way because right now the viewing view option right now is like only show the speakers. So who is speaking, then it will show the camera screen of the speaker. So even someone set a timer there, the attendees won't see it. But anyway, if it's just for the panelists to see it, definitely like someone can do this job.
Yeah, but I think, but does someone has to on the timeline of it?
Yeah, it will be like constantly on off the camera to can.
Okay, can you do that?
Yeah, maybe I'm not into just.
Let me try to send you find the video out and send it to you. Basically, just set a virtual background, but the virtual background is a video.
Okay, I see. I will try to find it.
I can send you the video for a 10-minute countdown. Let me find it out.
Yeah, exactly.
So we have. Oh, it's only on camera, so okay, I see. Yeah. The other people's are not. They didn't turn on the camera, so it's not showing up. Okay. Yeah, okay. Sounds good. Wow, this is huge. Oh, okay.
And just end my screen sharing.
You can mingle outside if you like. There are people
Okay.
arriving. Oh, sure.
Hey, how are you? Hi, good. Welcome. Thank you. Jeff Leiter from Luminary Therapeutics.
新鲜，我分裂啊。
Okay.
这样放呢，还是说不要收那个 screen？
Okay.
哪一个？
是这样呢，还是这样？
Yeah, one of the pictures, right?
好的，这样。
这样更好是吧？
No, actually, I'm not. I wish I was.
Oh, no?
So I got a late minute invitation, and I thought, well, I'm in town,
okay.
so I'll
Okay,
stick
next
around.
year. That's all
Yeah.
next year. They
Yeah.
have been many years.
Yeah. So we're a cell therapy company.
Oh, we have a cell therapy spin-off.
Is it okay?
Yeah.
Called the cell.
Basically, I'm waiting.
What kind of cell therapy?
So we use gamma delta cells.
Has been
Okay.
Allogeneic.
Try to set up a timer.
Okay. Like, what's...
Susan, are your slides loaded on the computer?
Not yet.
Yeah.
Better to load it on the same computer.
Then there's one not
Yeah, so that mine already is.
This one?
Yes, this is the computer
It's already a couple
presented.
For the
times.
presentation.
I made it.
So I just need
So they
to.
come out there.
Yeah, do you
This
have-
is my camera and
Our
No.
ours also have this, so maybe send.
Yeah, that's.
Why?
So you can actually like find
Which, yeah, but you do always.
Yeah.
Also don't know.
But I think I do not.
Roche's presentation,
Jen-
they load on the same.
Yes, of course. So where?
So send to
Invaluable.
Send to you,
Send to
Can you
you.
send to me?
which,
Send to me. I'll give you my card. You can send it to me.
Email? Yeah. Okay. Yeah, I can email you.
you can send by email.
Yeah.
Would it be too big?
Absolutely.
Our students, they send in anyway,
Not too
big.
other than
I
WeChat.
feel like they are gamma delta. They
Okay.
should know they were-
Yeah.
Sure.
Right there.
Is there Wi-Fi available?
Yes, it's right on the table.
Because Johnson & Johnson's associated with Legend,
So,
Thank you.
Legend, yeah.
but they're not gamma delta.
Yeah, see? But it's not gamma delta T cell. Gamma delta antibody.
Probably.
Oh,
That's not.
Okay.
it's like 30 megabytes. We count is
No,
not.
it's an emergent field.
Yeah,
佢哋佢咗去 washroom。
So
Yeah.
More about sincere.
We want company to come to our office and
Oh, I see.
incubator.
Okay.
Yeah, I can give you a quick tour.
Yeah. Still have 15 minutes.
We've got multiple offices here too, right? That's
Yeah.
gonna be connected.
Yeah, so I'm gonna set up the, yeah,
I've
set it
up
right.
this way before.
Yeah.
So you're still in Cambridge, but
How many
not heard of
groups are coming today?
most expensive place.
You presenting?
Present
You want to take a
14.
look at
Yeah,
Cambridge?
let's do it.
Companies will be presenting.
Two, okay.
Yinchen, you want to have a quick tour? I can show you.
Maybe it's better for this door not to be open and closed.
Yeah.
So maybe the flow people can just enter from the other.
Oh, yeah.
So maybe we can
Sure.
get an usher to make sure that.
Yeah,
Only the presenter.
Yeah.
Otherwise, the people will be
Yeah.
waiting.
I'm waiting for money in the meeting.
Okay. You should be ready for that.
我是管这个 BD。
Martin 啊，佢等緊你 send 嗰個 video 啊，你係咪 send 緊啊？
咗啊，我 send 咗，不過佢可能
太大啦
video 有啲大，佢可能都要幾分鐘先收到。
OK，你同佢講，因為佢唔知發生緊咩事喎而家。
我聽到嗰個佢好似
我 WeChat 同佢講咗啦。
OK 得嘅，唔該晒。佢就揾緊方法點樣
Already received.
OK，good。Hello。
应该是没有我这边的，我就坐后面吧。
这里是给参赛队伍的，
不好意思，
这是给参赛队伍的。
参赛队伍可以啊。
OK
Maybe sit with any seat.
OK.
Are you one of the team?
Yeah, we're going to do the social role. We are here.
你好，你好，你好，你好，你好，你好。
阿 Rachel 嚟呢度冇問題啊。
冇。
Okay.
哦，
琴日睇過啦。哦，OK，
喺好
因為佢係 Arlington，
嗯，
阿姨喺度住啊。
係咩？
係啊，咁喺呢個 complex 㗎。
喺隔離先做。
哦，OK，呢度新㗎喎呢度。
佢話呢個 office 呢棟 building 5年。
哦，即係前邊住宅嚟。
係咩？
好好嘅，係咁樣樣。
跟大家說，
室嚟嘅。
一陣間咧，
咁你哋
我哋差唔多，我哋一陣間開始就都係可能有啲因為同步喺有啲人開喺中國又有 streaming， 因為佢呢啲我哋有啲 media partners 佢 live streaming 就啲 live streaming 呢啲，所以有1萬人 call in， 咁咧就
需要有個人至少站在這裡。
上面咧基本上我哋俾咗呢幾個係嗰啲喺亞洲，因為佢完咗11點半夜，咁然後就亞洲嗰啲就係按世嘅，總共有14類 present。咁呢啲就係而家嘅事，我係8個細嘅係好咁就開始嘅時候咧就佢哋介紹翻 BCIC 啦。講個 speech 關於香港 science 啦，跟住咧就呢個係讀住 Roche Accelerator， 咁佢我哋都會講，跟住就10點鐘開始演唱。我哋電話呢個已經喺度，我哋有個 preliminary 個 semi final， 係 online， 咁就盡量 in person 啦，但係有啲嘢
他那個
semi final 係
桌子裡
No, I just checked it. Shouldn't be in the spam.
呢啲係做 judge。
How big is the file? Very small.
我
I should be found. Sure.
有
Do you have a USB B drive?
The USB 咧，
可以參加，可以。
Oh, okay. Got locked out. Okay. Or
所以佢我哋坐 courtroom 咧，其實有好多 online office 係 meeting room，係佢哋 overflow，佢有電視機 stream。而家 lunch time， 佢哋會有個存在嘅。
That's right, right?
20 好似20萬片。
You said like Gina.
係帶啲人有個 quick tour。
Yeah, you should be
Full
like
day for you
quick,
here。
quick.
係啊，full day 啊，係啊。你同我食飯嗰時，我哋應該5點鐘上去，跟住我哋就會 make sure 我哋啲 judges 唔好打交，呢個贏嘅。咁然後咧就
So
班
just in case, that's my first name. Last name IYL at gmail.com. Just in case it's not for some reason coming through.
今朝我 focus 係啊，阿 Polly 做咗前年嘅 part。係啊，啟明創投呢個OK。咁跟住咧就佢做咗其中一間我哋園區公司嘅 board of director。問我下個禮拜得唔得？我未翻到去，所以我話咗我俾 send 俾佢。我話你中意你就 call in 啦。
Yeah,
係啊。我同
That shows up.
臨退休前嘅。但係佢個位邊度做
Yeah, okay.
我同你我問 Peter 應該都要退休，叫佢唔好做嘢 ，enjoy 佢先去仔嚟做嘢。
Did you get the 5 from the account? It's not necessary to have. Can you transfer again?
Wait, I gotta.
Should we reach out?
係啊。Boston
Oh, wait, okay.
好中意 Boston。
Okay, it's open now, so yeah, okay, okay.
That's because like I don't use the Wi-Fi.
Ah, I see. Okay.
It's still transferring.
Okay, so right now I'm trying to figure out. I have right now
我老公就
4.
988。
Okay.
哦，我係我老公19。
一個九。
You-
Yeah, this is for sure.
唔係啊，我已經捱佢啦，我撈㗎啦，我可能係啊，佢碌嘅啫，唔係 real 嘅。
Put it in presentation mode. For the PDF.
Sure.
So you
Yeah.
listed
I'm gonna
I
put the role.
Min is the president of the Boston Chinese Investment.
Oh,
This is Miss Alice Chen. She was Hong Kong STP.
I just came through.
Yeah, it takes long.
Remember?
Yeah, it takes
Several
more
years
time.
back. So she was my
Yeah,
boss.
Perfect.
Now
There's some village waters and
Yeah.
I did. I just need some coffee.
Very important.
Back to
How's jet lag?
to Hong Kong.
Okay lah.
Okay lah. So no more jet lag.
But I start with
Still a bit
Mercury.
like, you know, you get
In Boston.
Fox time hours when you wake up all the way round.
Yeah.
All the way make a loop.
Yeah.
We have busy schedule during
From
the
the
day.
extreme
Then
cold to the extreme warm.
Yeah, it's really beautiful. The sun makes a difference, makes you feel energetic. Right?
yeah. Typically, I think I went to morning jogging.
Okay.
On the river.
Yeah,
The past 9 years, I think the climate has been
The weather is much better than
Exactly. It's not as cold.
UK
yeah,
is
that's true.
Oh,
grey and
Like
drabby.
10 to 15 years ago.
Um,
Really much colder.
Really cold.
Yeah.
I already told
That's what-
Alice I found this very cold.
Were you here in 2015?
Yeah.
Oh,
There's a
you were hit
big
by
storm
that.
already.
Yeah,
that's true.
2015.
Oh.
Yeah.
And all the roofs got to crash.
But
That's
since
true.
then, you know, it's kind of
That looks good.
soft.
Everything is good.
Right.
Oh, that looks perfect.
Yeah, it's much brighter. That's
You did that. That's perfect.
our trick that we
I know.
This is a good
No,
trick.
very nice.
Yeah. So everybody can see.
Yeah.
there's no
So
question.
who,
Yeah.
who found this trick?
Martin.
Martin?
Because we do this all the time in science park.
But how come
all
we
our
don't have-
funded programs are all like this: 12 minutes presentation, 10 minutes Q&A start.
Oh,
Okay.
So as if it's real, like life together.
Yeah,
Yeah.
Cannot.
Nice.
But
It's
in
very
the semi-final,
seamless.
we don't have such feature.
We use
Oh,
yours
there's
like
another
a little clock up there.
Yeah.
But you couldn't hear the music.
That's true.
So I asked Martin to make sure that
Okay.
Jimin can know.
So
Like, this
Jimmy, you know how to change that or adjust or?
yeah. So make sure every time each team starts, you put it. Don't worry, she's the fast one. You'll be fine. Count down.
So we're gonna be like 10, 9, 10 minutes presentation, 9 minutes QA.
Yeah.
1 minute maybe set up for the next game. Yeah.
Okay. Sure. Okay.
Let me take a sip.
Yeah. You sit next to Wendy. Where is the host? I want to have a chat.
The host, okay.
Say what?
Your slides are okay.
Yeah, all good. It's already in the laptop.
Chusong, this is Alice. Alice used to be
Chusong.
STP
Hi,
board
how are
member.
you?
Yeah, 4 years
Hey, nice to
STP
meet you.
board member.
So you're now-
Several years back, she lives in Boston now. She used to be my boss in MSD. I have to say that one.
Yeah.
Chusong is our head of Roche Accelerator
Yeah.
in
Oh,
Zhangjiang.
yes,
Yes.
yes,
So
right.
you're now based in Boston?
I now lived here.
Or live here?
Yes.
Was still
I-
working or kind of half-work?
Not much working.
Not much working.
Really lazy.
But that's
No,
a spirit.
real life.
lazy.
She is doing the real life.
That's a spirit.
Super lazy. Yeah.
The spirit of living in Boston, right? I mean,
Yeah.
and here,
What about you?
based in Shanghai?
You're based in Shanghai.
Yes.
Yeah. Okay.
And
Do you come here?
We work with.
We have a
Team a lot.
collaboration, a
Right
collaboration
at
partner from
in the
STP.
horn. Yes. Good. Yeah. So wonderful. So were you here like the whole entire 2 weeks or?
Not 2 weeks. Flying back 2 days after Greece.
Okay.
Yeah.
Uh-huh.
And then, of course, I think, we'll be busy May and June anyway. I think on June.
May there is a big event in Hong Kong as well. There's
Okay.
Asia Summit of Global Health.
Oh, okay.
The week of 13th,
All right.
16th and
Yeah.
17th and 18th or 16th.
17.
Yeah,
There'll
something
be
like
quite
that.
a lot of events.
Yeah, yeah.
And now June, and everybody will be back here in West Coast
Oh,
for the bio, right?
yeah,
And then
yeah.
bio, the first week of June, bio in San Diego.
Is it okay? Right. I was at the Global Women's Health Summit right here in Boston
Okay.
in the middle of April, 3 weeks ago. But then we had a big storm here. Yeah.
Oh, there was a storm.
Yeah, Mrs. Biden was here too.
Okay.
The First Lady.
Okay.
Yeah, really want to push women's health, all the biodevelopment, and they want to make it's the Biden's administration. They call it moonshot.
Yep.
The moonshot health. They put in like another large billions of dollars into women's health. So because most of the trial indicator is all based on man data, so it's not
Right.
enough
Right.
in the women's as the
I would agree.
database.
That's a big initiative. I mean, it's a lot of money.
Yeah, it is a lot of me. So I was involved quite a
Okay.
bit on more on the policy front now. Yeah, that's on
Initially, one of them.
the R.
So you're now working more like an advisor, consultant,
Yeah.
rather than working for a particular company.
Yeah, right, yeah. More on the healthcare policy and,
For the government or
for the NGO,
for the NGOs?
yeah.
Okay. Yeah, that makes sense because I see here.
Pharmaceutical reimbursement, quite a lot.
Yeah, there's a big. It's one of the largest industries here
Yeah,
in the entire Boston area.
right.
That's
yeah.
why everybody's here.
Yeah, all the major players.
Yeah, absolutely. Most of the multinationals,
She can do that.
biotechs, venture capitals, they're all here.
Yeah. So either Boston or in San Francisco. Yeah, Bay Area. But I think Boston is more conducive for healthcare.
Yeah. I
Biomedical.
have to agree. Yes,
It's right now.
it's more translational and industrial
So these are the companies.
focused.
Yeah,
Where
and because we're so many hospitals.
exactly, and lots of clinical and academic
Yeah,
resources. The Bay Area sometimes things can go a bit crazy. So, but other than that, I think, yeah, they sometimes come up with crazy breakthroughs. Most of the time, if you compare with Boston, Boston is more still the powerhouse.
yeah. I think it's the tradition too. We have the legacy.
Yeah.
Yeah.
Very
I used to work in the hospital right here at this room when I was much younger. When I right off of college here.
And then you were with.
Then I went back to Hong Kong and I worked for Merck.
Oh, that's the.
I worked for Merck in Boston and
Okay.
then transferred to Hong Kong.
Okay.
And then I was in Beijing for 2 years.
Okay. Yeah, Merck is it's doing great in China, at least. It's the largest pharmaceutical company in China in terms of revenue.
Now?
Last year,
Last
2023.
I thought it's AstraZeneca.
Merck is the largest. Merck, AstraZeneca.
I read something and the AstraZeneca people told me they're number one.
Merck is the largest. For sure.
But unfortunately, I don't have any more stock options. Me neither. We just checked yesterday. It's 100 and something. 120. When I sold everything, it was 60 something.
So when it comes to the roadshow only, around 4 o'clock. So the hostess can take over. Because we are going to be in
Alice， 如果我哋喺度 pitch 嗰時，你需要離開你去 washroom 啊，隔離networking 啊，你 feel free 到啊 ，yes， 可以出去。Very nice place.
I'm not sure.
Actually, very interesting because I'm quite involved.
I've been there.
I'm more on the policy fund,
I'm not sure.
healthcare.
Oh,
Like you said,
Yeah.
how tiny, but
DC as well. I like to do.
And then they run.
Not like me that much.
Some's been
Reimbursement.
always 5 to 10 minutes.
Yeah, very important.
We should probably get seated.
Almost.
no.
okay, so
Economics.
Yeah, BCIC.
Maybe I can if I meet now. Wendy doesn't speak, right? Presentation, the opening. You don't. No, just you. Yeah, just me, and then
Jeff Letter.
and the choke.
Yes.
Okay.
Where are you?
And Song.
We're based in.
Oh, 松山少林。
We're a self.
松山的松树的
I'm yes a visitor. I
上
got
面一个山是这个高。
啊，我的 meaning very tall.
check it out. Yeah.
山高山啊
Yeah, high mountain.
，high mountain. High mountain in the autumn in fall.
Literally. I don't know why it was given that name. Great. I think it's opening their
Share your screen. Yeah, I will do it right now.
So I think they're now coordinating with Virginia to find a slot and make sure the agenda is aligned. So I think they want to make sure they want to try to see if you will be there in the afternoon.
也有人好像。
2 p.m.
不是吧 ？It's a public holiday. 15th, May 15th was a public holiday. Nobody works on that stuff.
Oh,
Really?
Yes.
yes. I don't
It's,
follow the baseball that much. I only think, oh my God, what traffic.
活蛋，
对，我跑不去。
15th May cannot.
Okay, it's not a public holiday in England.
It's a I marked it in my calendar. Yes. And then 16th and 17th are ASGH. So that week is very, very busy because already 5 days, one day less.
Yeah.
So all the meetings, all the visiting delegations from various countries, they're all visiting on the breakout week.
Okay.
But 15 no good because nobody will be working. No guests will. No guests will come on that day.
True? Then we have to find another day.
Yeah, if that's the week. Better the day before. I think 16th and 17th. We have some people wanting to have events on those 2 days. We had probably very.
Yeah, probably most of people will be
at the convention because it's ESG and medical and healthcare fair
Yes.
at the same time.
Yes.
Yeah, sure.
And I can make sure 14 we have another event.
Come to our office.
The 22nd. Yeah, so 20th. Actually, you should.
So you know Grace
Yeah, we work together for the vote.
Really?
I'm here from
Okay, because Grace knows American and STP and then Very good. I'm actually based in Hong Kong. I live in Hong Kong, and then our office was used to be in Northport. And then we moved to STP in December last year. So we now have a very nice office setup, thanks to Grace's support. And we have this we call it the Hong Kong Collaborative Innovation open up to university taking advantage of I think very good policies that are in place. Encourage yeah, I've been in Hong Kong for well, we don't want to do something that's in Hong Kong. Yeah.
Mic is dropped. Okay.
Yeah. Hello, how's it going? We met online, I think.
Yes.
Yeah, nice to meet you.
I'm Grace.
Hi, Grace. I think we met online as well last time, right?
Sure, yeah.
Yeah.
Afterwards, I might invite you to introduce us to the judges. So,
Yeah, sure, of course, of
I'll
course.
give you a prompt and choose one as well. I will indicate that you can introduce yourself as judges afterwards.
I see.
Yeah. Oh, okay.
And now we have
Afterwards, before we start the
okay, now there are
conversation,
plus
introduce
topics,
yourself.
right? And
And then
then
I will go around to ask the judges.
and I think one of the reasons we be better connected to the biotech industry in general, right? Because we used to just have an office in North Point, but primarily because we're sort of public companies in Hong Kong, so we created an office. But we don't have the necessary people and resources to build around some serious sort of pharma capabilities, right? So like, you know, definitely can be used a bit.
So, do you find good access? Recording and
Access. I think translation is the main challenge.
Should we get started?
Yeah, it's pretty exciting.
So yeah, why don't we get started? Yeah. Hey guys, good morning, ladies and gentlemen, and good evening to our audience in Asia, and good afternoon to our audience in Europe. Welcome to the third Biomedical Pitch Competition final stage, co-organized by Hong Kong Science and Technology Park Corporation and Boston Chinese Investment at Sincere Innovation Center in Boston. I'm Han Zhang. I'm really honored to be the host for today's event. I just remember when I was doing my summer internship a couple of years at the Cambridge, my mentor told me that if you want to know what's going to happen in the pharmaceutical industry, make sure to come to Boston each summer. And here I come. And I'm pretty sure I'm not here for the lobster. But look around. So Boston has hundreds of biotechs and venture capitals in Boston area. So which make the Boston is the best place in this world for innovation and investment in the healthcare and biotechnology. So today we are in a time when the need for such innovation cannot be greater. And after several rounds of pre-selection in the past 2 months, we have selected 40 teams out of 100 to present their groundbreaking innovations and disruptive solutions that have the possibility to revolutionize the current healthcare system and biotechnology. And to our teams for today, I wish you all the best luck. And to our audience, whether or not you are in person or online, thank you for joining us on this exciting journey. Let's make today's event a celebration of creativity and possibility. Okay, thank you all for joining us. First off, I'd like to introduce Mr. Ming Yi, the current president of Boston Chinese Investment Club, to introduce what the club is doing and what we have achieved for our scientists and investors. Mr. Yi. Thank you, Han.
So, welcome everybody. And so this is the third year we have been hosting the Biomedical Pitch Competition. And with that, in the Hong Kong Science Technology Park, and is co-organizing this event with us. And so my job is next page. So first, I'm going to give a simple introduction to what is BCIC and how we're doing, and also about the history of this biomedical pitch competition. especially the past historic achievements. And finally, I want to just thank those contributors for this event. So next page. So BCIC, simply speaking, is just platform trying to connect the resources from like United States and also the Asia area as well. So in terms of resources, we are focusing exclusively on investment, so the capital, the talented people, and also investment opportunities. Next page. So these are simple facts about BCIC. There's my echoes.
Mute the Q genetics.
Okay, Q genetics. Right. So we get almost like a 30 years history. And before we were called PCFA Boston. And so currently has around 10,000 members globally. And so core member teams, annualized, we have like around 100 people. So what do? And so we are more than just healthcare. And so we have finance and blockchain, high technology as well. But it seems like the success of the healthcare is overshadowed the other sectors. And so in terms of format, it looks like we have just like a roadshow, pitch competition, conference, but in reality, let's change to the next page. We are doing more than that. So through the pitch competition, we help the early stage, middle stage companies basically trying to engage with the potential investors with these funds. And we also help the middle late stage companies for in the sales and the license BD as well. We have strong BD teams, and especially we hope focusing on the companies from the Asia area who want to penetrate, maybe expand the business in the United States. So in this, we are doing far more than just healthcare. And so we have done one case in the new energy companies and also e-commerce companies as well. And finally, for the late stage companies, we help them to navigate in the IPO market. So far we have done 2 cases, right? And so that is what we focus on. Next page. So back to the healthcare team. So what are the team will do? So we have around team size of 50 people for the healthcare, and all professionals. Like we have venture capitalists. They're from branded names and MSV and Eva and Qingyuan, and also proximity as well. So we have serious entrepreneurs, 5 people, and the companies of some like entrepreneurs already was like more than $100 million. And quite a few like senior advisors. They're focusing on investment and also the technical scientific fields. Finally, in the BD, so we have strong BD, solid BD teams as well. And they all have their own companies, consulting companies as well, have done achieved a lot of really sound data sheets and done a lot of cases. So finally, execution teams. That is the core of our team. So for this event, because of thank you to them, we have around 20 people, and 70% of them are just professional, very strong students. And so this volunteer, they're serving as volunteers, the team serving as volunteers for us, and they either standing in the room or maybe serving outside. That's healthcare team. All right, next page. So pitch competition. And the purpose, we're trying to source the high quality startups and help to engage with the fundraising efforts. And so for this year, we have been 3 steps, how solid our screening process people have asked. And in the preliminary, so far we collected close to 100 startups. And in the preliminary, Hong Kong Science Technology Park and us, and invited 60 teams for the preliminary and pitch competition. And so as a result, 5 teams got golden ticket to the final stage. And also 20 teams, we invited 20 teams for the semifinal. In the semifinal, there are 4 judges. And in finals, there are 14 teams. They are going to present to us. And also, in addition, there are 2 teams we think they are pretty good, And we invited them for the roadshow only, not for the competition. So today, 6 teams did the presentation, and the 5 judges, Hong Kong Science Technology Park and Roche Accelerator, and also the KCB, Kendall Capital, and the Peptide Tree and the LB Venture. So Peptide Tree is going to be sitting online as a judge. Next page. Right. So look back at history. This is 30 years. In 2022, and 48 teams signed up, and 6 for the final competition. So as a result, you can see these names right here: first, second, and third places. And in addition, by state, as a result, so far, close to $25 million has raised the company has raised $25 million. And there are quite a few companies landed in the Asia area, and Bosch Eagle, and in Asia, and also the Hong Kong Science Technology, like $5 million in reward as well. In the second, 2023, 85 startup companies signed up for that. 12 invited into final competition, and 6 additional for the roadshow only. And first, second, third places. The Miro, by the end of the year, raised $24 million. $24 million. That's a big numbers for last year, especially in challenging environment. Also, the few other teams are pretty good enough, like some legs, and as a result, one is still on the due diligence in the due diligence phase, and 2 got term sheet already. And we believe in the next 2 months window, more companies will officially announce fundraising efforts. So that's for this last year. This year, close to 9 companies signed up, and we invited 14 for final competition, and 2 additional teams for roadshow on. And comparing to the last 2 years, we have more diversity in terms of for example, in the past, we focusing more on the most of the teams are just therapeutics. But this year, we have some medical device team, which is really solid, and also in terms of phases, from early stage to immediate stage, and also become more international. And this morning, for example, this morning, 6 teams, they're all from the Asia area. 1 is from Singapore. And so at the end of the day, where should the results? And so next page. All right. Keynote speaker for today. And Grace Winmy from Hong Kong Science Technology Hub, and also the Dr. Tom from Roche Accelerator. Right. So our host is going to read the bios next. So we can move to the next page. In the co-organizer Hong Kong Science Technology Hub, and this year was the first year we have been organizing with the Hong Kong Science Technology Hub. It is a great journey, and we've been chatting each other on a weekly basis for the past half year, and we really enjoy this partnership. Also, thank you, Roche, as a strategic partner and also sponsorship, and they're serving as judge as well. Dr. Chang have given us a lot of advices. Also, thank you, Cynthia, and using the venue as the semi-final as well, and also they're going to sponsor the VIP dinner for the night. Also, the 2 attorney firms, and also the 3 VC judges. One is the KCB Kendall Capital, and the Peptide Tree and the LB Venture. And finally, the 2 more sponsors for this event, and they are Goodwin Turning, and also the Baker McKenzie as well. Right. Thank you, Dave. One more page, I think. Right. And also, we want to thank you to the media who has been doing the live streaming for us today. And they are eDrug and Deep Blue and Cyber and the PD. The Annie is here. And Gene Online, the eDrug. And also, we want to thank the rest of the partners who has been helping us promoting this event. Right. So, final, last page. One more page. Yeah. So you can just reach us and through this I mentioned the code and also the website as well. All right, thank you, and I want to give that to all the hosts here.
Right. Thank you, President Lee, for your great introduction to the Boston Science Club. So I'd like to introduce our first keynote speaker, Dr. Grace Law. So all of our keynote speakers have very long achievements, so I have to prepare this name card to read their bios. So in her role as the head of Institute for Translational Research in Hong Kong HKSTP, her team are responsible to drive the transformation in healthcare by speeding up the translation of innovative biomedical technologies to benefit all the patients and the Hong Kong society. And she has over than 20 years experience and expertise spanning across different areas, including the clinical trials, research, regulatory affairs, sales, and marketing in different in different market environments and geographic locations. Prior to joining the pharmaceutical industry, Dr. Lau was an academic staff of the Chinese University of Hong Kong and a guest lecturer for a number of higher education research institutes. Welcome, Mr. Lau.
Thank you very much for the kind introduction, and thank you, Min, for the opportunity to work closely together in the last 6 months. And we definitely got to know each other very well. So I would like to use the next 15 minutes. We're running a little bit late, so I'll try to be short. I want to give you a picture of the biomedical technology ecosystem in Hong Kong and what we're doing in the Hong Kong Science and Technology Parks to drive this forward. Now, next page, please. For the last 10 years, Hong Kong has been putting in a lot of effort to develop Hong Kong into a center of innovation and technology, an international hub. And for my part, I drive the biomedical technology. We see this as the future for Hong Kong, driving technology and innovation. Next slide, please. So this is a picture of Hong Kong Science and Technology Park. In Science Park, my team works in Science Park, so this is the base for research and development. But Hong Kong Science and Technology Parks also manage other locations, including what we now call the Inno Parks. So they're dotted around the other parts of Hong Kong. So when our technology, they go down the translational journey, they are ready for commercialization, because Science Park is dedicated for research and development. So they're welcome to use our Inno Parks, which are like industrial estate, to develop or set up their manufacturing plants. So these are dotted around the other parts of China. We also have center site in the center of Hong Kong. And also we also just started branching into Shenzhen, into the Greater Bay Area to facilitate companies in China wanting to explore and go global. And also for global companies to either stay in Hong Kong Science Park or go into the Shenzhen Greater Bay Area to explore the China market. Next slide, please. So this is a snapshot of what we are like now in Science and Technology Parks. We are 22 years old just as of this year. I have a guest here, Ms. Alice Chin, who was a former board member. So in her days, like more than 10 years ago, Science Park was a building site. But with all our effort, we've now developed it into a park full of big buildings. And we have 20-something buildings, and we have over 400,000 square meters of laboratory and lab space now in Science Park. We're fairly full, 92% occupancy, but we continue to find space, like reclaiming around the river, the ocean, and continue to find space to build more buildings to house all these incoming companies. We're proud to say we are now over 1,700 companies from 27 different regions and countries around the world, so fairly international. And one of the key requirements for companies to enter Science Park is that they must be engaging in R&D, and over 50% of the staff must be in R&D. So it's a very vibrant ecosystem. Over 20,000 people work in Science Park, and you can see over 14,000 are engaging in R&D activities. Now, I won't go into a lot of details, but we have been able to attract a lot of startups, over 1,000 incubators, and we have also nurtured them to grow into small, medium size, and we have attracted some big companies into Science Park as well. We're proud to say we are a generator of unicorns, so we have I'll be introducing, we have other functions other than providing space for your research. We have our own CBF Capital and Venture Fund. In the past several years, this not too big fund and yet very successful. We've been able to find the right investment opportunity, and $1 that we ever invest into our companies, we generate 19 times of private money from outside Science Park. So we are a generator of fundings for our park companies as well. Next slide, please. So this is a glance of what we do. We are not just an incubator. So throughout the whole journey of translation, from when the university teams only have a great idea, so they can enter Science Park into our ideation program, just a year with little funding, but it's time for them to get mentorship, how to craft out their proposal or their project. Then they can apply to our incubation program. There's a general incubation program of 3 years, but for the biomedical technology area, it's a even more structured program of 4 years. And then when they grow a little bit bigger, when they graduate, they can apply to our acceleration program, elite program, and ready for global expansion. So throughout this journey, we have our funding and capital. We offer them different kinds of office and lab space and shared facilities, including physical labs. I'll talk a little bit about what we have built in the last few years. And technical support, promotional, help them to promote in LinkedIn. Whenever they have a key achievement, we can help them to set up key events. And this is actually, we'll also talk a little bit. Next slide, please. Okay, so throughout this journey, we also have different kinds of funding, like matching them with the right partnership. For the non-biomedical, we have all kinds of partnership. And for biomedical, we match them up with specific biotech VCs as well as multinational companies who can become their co-incubators. And acceleration, we have different levels of funding, and the STP venture fund, which I have alluded to. Next slide, please. So one of the, apart from funding, one of the key attraction in Hong Kong for the biomedical technology area is we have a very good university. The basic research is excellent. In just the city-state of Hong Kong, we have 5 universities which are top 5 of the QS 100. And especially for the universities with medical school, the 2 University of Hong Kong, the Chinese University, they have the clinical research done in those teaching hospitals. And 2 other hospitals are world-renowned, and the data generated, accredited by FDA. So doing clinical research in Hong Kong is also another attraction. We go to the next one. I want to introduce the other program that the Hong Kong government has been putting in effort from maybe 4 or 5 years ago, a program called HK. So this program started 5 years ago, and the government put in 10 billion Hong Kong dollars and selected 28 different teams from our universities. Now, one of the key requirements is that they can be just University of a Hong Kong university team. They must be partnering with 1 or more of a globally renowned university, like MIT, Stanford, Harvard, etc. So each of these 28 teams, they're all housed in Science Park, and they have very big teams working on various projects with many PIs. So this has been better nurturing research and translating into further down the road. So the reasons for them to spin out from university is they're encouraged to do translation, spin out companies, and not doing exactly the same as they are. Next slide, please. So a quick glance. These are all on websites. So these 28 centers, half of them are held at HK, and including the different kinds of therapeutic areas, oncology, diagnostic, medical devices, etc. And the other half belongs to AI and robotics. So these are all on websites, and you're welcome to take a look. Next. So we're proud to say we have homegrown unicorns. I think Professor Dennis Low is well renowned for the non-invasive prenatal testing, which changed the world in terms of prenatal testing. Professor heads the lab, and they will be launching the surgical robots very soon. So these are all unicorns, homegrown, as well as attracting international unicorns to land in Science Park. Next slide, please. So I would like to zoom in a little bit, provide you with a little bit more information about the Institute for Translational Research, which is the institute coined for the biomedical technology and build up the ecosystem for the development of biotech. Next slide, please. So our one main focus is to develop Hong Kong into a preeminent hub for translational research in the biotech area. We don't just take care of the technical part. One of the key things is we want to instill researchers with entrepreneurial mindset, help them to face the challenges of being a CEO, leading their company, building a network, helping them to match with investors. Next slide, please. So we see ourselves as a super connector or a platform. So we call our companies partner companies. We are legally the tenant. They are in the park. But we work with all these companies as our partners. We are strategically positioned to have direct contacts with the universities. They have spin-off teams into Science Park, and we also help them with their pitching, their hackathons, etc. And all the other research institutes, they are fully government funded by the Hong Kong government, and they're also in Science Park. So we act as a central platform to connect them, all these stakeholders. And I mentioned we connect them with our own venture capital or private equity, and also help them to apply for different government fundings, including the Innovation and Technology Fund provided by the Hong Kong government. And connect them with strategic partners like our co-incubators and enterprises who would be adopting their technology. And of course, many of the companies in ITR, in the Institute, are developing regulated products. So we are in very close relationship with our regulatory authority. Next slide, please. So latest snapshot, we update this every month because our number keep going up. As of the latest, we have over 250 biotech companies under the Institute for Translational Research, coming from 12 different countries or region. And just for these 250 companies, some of them are small companies. We have over 1,600 researchers working in these companies. They belong to 3 different teams. The biggest team is therapeutics, which is headed by Dr. Wing-Li Yun here in person. So her team looks after the companies in small molecules, large molecules, biologics, vaccines, cell and gene therapy, some traditional Chinese medicine, in various stages as well. Some are already in phase 2, phase 3, some are very early stage in the platform company. The other 2 teams, I have 2 team heads looking after medical devices like the surgical robots, ophthalmic equipments, etc. We also have some big companies as well. And my other team head leads the medical diagnostic. So we have a focus on precision medicine, genomic diagnostic, etc., and some AI assisted technology. And we have some partners who are like CROs have representation within Science Park. So what I put here at the end is, as I mentioned, the half of the HK centers, they are focused in healthcare. So my team also look after these HK centers in healthcare, and they are very big centers. From the centers focused on healthcare, there are over 1,200 workers. So you can see the ecosystem of biotech in Science Park is very sizable. Next slide, please. So a quick introduction. In the last few years, with the government putting in a lot of effort, we built a lot of shared facilities, core facilities, including basic chemical co-working labs ready for companies to move in. When you're small, you don't want to spend too much time building your own lab. So these are all fully furnished labs. You just bring your team, your special equipment, and you can go and work in the chemical co-working lab. And then a whole suite of other shared facilities, including proteomic, metabolomic, genomic core labs. All these are offered for use by our partner companies. And for the incubators, they have their own bio co-working lab and office as well. Next slide, please. So what we realize is from bench to bedside, these companies are companies, they need different types of support, core facilities to help them go through this translational journey. So we built our own biobank, accredited, ISO accredited, So our companies can use to invest into their own cryo tanks and freezers, so they can bank in our biobank. And we just started a platform for sharing very precious biological resources. So we're fostering biobanks and the university to share material as well as data. We also just achieved a GLP compliant preclinical testing lab and animal lab. We have small animals right now, rodents. This is phase 1. We hope to be able to move on to phase 2 as well. So companies, they can get our resources, get our support to design the GLP compliant preclinical studies in PK, toxicology, etc. So we work as like a small CRO to support our startups. Now moving on, because we also have medical devices, so we built our own medical device testing lab for mechanical testing right now. But hopefully we would also expand. Now for cell and gene therapy, we work with the 2 universities, University of Hong Kong and Chinese University, to build 2 ATP GMP facility to facilitate the manufacturing of clinical material for clinical trials for cell therapy. Now, one of them already got a GMP license, and very soon Hong Kong U would also get the license. So in this way, we want to help the companies doing cell and gene therapy to have the local site to manufacture their clinical material. Next slide. Okay, just pictures. I hope one day I'll be able to show you around, let you know, visit in person. We have lots of visitors from different countries and different universities wanting to understand the ecosystem in Hong Kong. So I want to spend just the last few minutes talking about the several funded programs. So IncuBio is our flagship program. It's been going for 4 or 5 years now. So we started to get graduates. So this is a all-round program, not just for funding. Within this 4 years, each incubatee can get up to 6 million Hong Kong dollars of financial support. But I think more importantly is we give them all-round support, like in the regulatory area. They can use our shared facilities and equipment, of course. But we also match them up with the right mentors. They can help them do fundraising, help them to pitch, help them with IP issues, financial issues, etc. So throughout these 4 years, they are followed very closely by my team, supporting them on each milestone. But at the end of the day, we want them to be able to financially sustain and be able to commercialize their innovation to the next step. Slide, please. So to go with the IncuBio program, in the last 2 to 3 years, we have brought on site co-incubators. So these are big multinational companies. They were signed on in different stages. So their role is firstly play as a mentor. They can select, we match them up with the right method, like incubatees, which meet the company's strategic direction, whether it's oncology or digital health, etc. And so they get to know each other, and they may become their mentor, become their investor, or may become, in the long run, I hope they would acquire or invest into these companies. So the next stage, please. So we have a collection of different multinational pharma companies, diagnostic companies, and the last one on the right-hand side, and Shanghai Pharma. And the last year, we collaborated, and they actually invested to build one whole floor of co-incubation space within Science Park. So this is the first time we have investment directly into Science Park to build a co-incubation space for with a international company. And hopefully in future, we would see Hong Kong become a center of co-incubation with many more other multinational companies. Next slide, please. So the other 2 funded programs I want to introduce. The first is Clinical Translational Catalyst. 5 years ago, we have single digit, maybe 4 or 5 clinical stage companies. As of today, we have over 35 clinical stage companies. So one of the things we do is we want to see them move on, filing the IND and then move on to phase 1, phase 2 programs. And for this, we have designed this Clinical Translational Catalyst program, also led by Dr. Yun here. So for either therapeutics or medical device companies, they can apply to this CTC program up to 1 million 8 million Hong Kong dollars or just over 1 million US dollars for each program, for each project or clinical trial. And apart from the funding, again, we have put in place other support measures like matching them with the right CROs, CDMOs, etc., and also help them with the regulatory side as well. So this is one of the programs that has proven to be very successful and welcomed by companies who are already moving into the IND and clinical stage. The other program, next slide please, is MedTech Co-Create. We wanted to encourage collaboration among our companies. So any two companies, if they talk to each other, like in Kendall Square here every day it happens, two companies wanted to collaborate on something. They can put up a project plan and apply for this MedTech Co-Create funding. And for each project we have 2 million Hong Kong dollars funding. So we do all other things around this program as well. For example, matching them with institutions like hospitals, other institutions who would want to adopt their technology. And we have bring in a lot of awareness program for the medical profession, for example, to understand how they can use technology to improve their daily practice. So I think that's probably more or less what I would like to share with you. We have a vision. Although we have 1,700 companies in Science Park now, we have a vision in the next 5 to 6 years. We want to develop into a global international innovation and technology hub with 6,000 companies. So for this, we would be able to help many more companies to fundraise and commercialize and increase their valuation. So in terms of talents and investment, we put in a lot of effort to help support Hong Kong and all our startups within the ecosystem. I think I will stop here. There's some backup slides, but I would be very happy to talk to any one of you today or in future. And please connect if you're interested to expand globally and into other markets outside the US. So thank you very much, and I hope that you all enjoyed today's pitch.
All right. Thank you, Dr. Lau, for your great introduction to the Hong Kong ISTP and the Institute for Translational Research. Our next keynote speaker, Dr. Wenli Yong, the current Associate Director of the Institute for Translational Research of Hong Kong ISTP, and she has more than 20 years of international pharmaceutical.
In terms of, in view of time, we'll pass to. Yeah.
Okay. So, I will introduce her more later. So we will move on to our third keynote speaker, Dr. Tang. Dr. Tang is currently the Roche Accelerator, and this is the first in-house accelerator in this role to expand the Roche capability in early research development in China across pharmaceutical companies and diagnostic by partnering with scientists and venture capitalists. So prior to joining the Roche, Dr. Tang was a strategy consultant at IQVIA Consulting Services Singapore office and the Roland Berger Munich and Shanghai office with equity research experience from Merrill Lynch, Singapore. Dr. Tang holds a PhD in neuroscience from Humboldt University of Berlin, Germany, and a master in neuroscience from University College London, UK, with a lot of peer-reviewed publications in Science, Neurons, Nature Protocol, Cell Reports, etc. Welcome, Dr. Tang.
I would like to thank the two co-organizers to invite me to Austin. And of course, it's always a pleasure to be here at this time of the year to see the cherry blossom is really beautiful here. So I would love to use this time to not just to show the beautiful pictures of Boston, but also to talk about what we do here at the Roche Accelerator. And our motto is really to accelerate breakthrough innovations into next generation therapeutics. So next slide, please. So brief introduction of Roche. I think everybody may already heard about the name. To give a bit of more flavor into the entire group, Roche is a family business founded more than 125 years ago. And this is still primarily family owned and the business, even though we're a public company. Roche has a whole long history of innovation. We are not only home to 3 Nobel Prize winners, we're also one of the largest R&D spending companies in the entire field, maybe not just in healthcare, but also across the entire industry. we spend around $15 billion every year in R&D. And by doing that, we are proud to be able to develop multiple innovative therapeutics and diagnostics in serving our patients worldwide. And of course, we have not just therapeutics, we also have our diagnostic business, which is strategically synergistic because we believe we can serve the entire value chain for the patients from the initial diagnosis to the treatment and the entire holistic solutions to the patients. Next slide, please. We are primarily focused on a couple of therapeutic areas. Most multinational companies are focused on oncology. We do that as well. Roche has been with the reputation of being a very oncology-focused company, but also we are now very strong in neuroscience, immunology, as well as ophthalmology and infectious diseases. And in the recent years, we also pivoted into cardiovascular and metabolic diseases, as well as obesity. Empowered by our innovative approaches in research technologies and personalized healthcare and digital healthcare solutions. So this will be the primary therapeutic area focus. And I see that most of the companies today in Roche are also working on those areas as well. So I very much look forward to discussing some of the exciting science with you guys. Next slide, please. So a bit about the unique innovation model of Roche. So very different from some of the other multinational companies, which may have one single centralized R&D center, either in Boston, Cambridge area, or in Europe or in West Coast. Roche actually has 4 autonomous R&D units around the world, including our US-based Genentech in San Francisco, our headquarters in Basel, our colleagues in Japan, Chugai, as well as the China Innovation Center of Roche, where Roche Accelerator is home to. So these 4 R&D units are each autonomous, have our own budget, decision-making, headcount, and therapeutic area focus. And we also look at external partnership and licensing deals individually. So of course, after the therapeutics or solutions are passing from a handover from R&D to pivotal trial or to commercialization, this will be handover or transition to global execution, where you have the clinical development team, you have the strategy, the manufacturing and commercialization team across the world in multiple countries and affiliates. In addition to our in-house research, we also acquired several companies in the past, including, well, Genentech being one of the most promising deals in the past decade, but also some of the smaller therapeutic companies, including Spark in Philadelphia, Comart in Berkeley, and Foundation Medicine and Flatiron actually here in Boston. In addition to these acquired companies, we also have external innovation partnerships or licensing deals, collaborations with more than 250 partners around the world. So next slide, I will give you a flavor about some of the recent deals that we have done, but also before that, I would love to give a small introduction about the China Innovation Center of Roche. This is one of the 4 autonomous R&D units for Roche, as I mentioned. This is essentially the first and now probably the only present fully owned multinational R&D center in China. And we can do this from early discovery all the way to clinical development. We are focusing on oncology, immunology, metabolism, and novel modalities and technologies among some of the broad interests of the Roche Group. Of course, here one of the features of the China Innovation Center Roche, or CCOR, is that we fully leverage and get access to the ecosystem by including my team leading the Roche Accelerator, but also by working with academic institutions, as well as licensing innovative assets from companies around the world. So I can give you a next slide, please. So, for example, in the recent years, we have done quite a number of partnering deals and acquisitions across different therapeutic areas, including immunology, cardiovascular, oncology, neurology, as well as some of the novel technology platforms and diagnostic solutions. So external innovation is really at the core of Roche's innovation strategy. In fact, I think more than around half of our assets or pipeline products actually coming from external partners. Next slide, please. The accelerator as one of, or as the first and only in-house accelerator around the world, around the Roche Group around the world, is we are really trying to bridge the gap from translational science to translational medicine, where we see there are two main gaps in this process, right? Because we can see that clearly from a scientific hypothesis from publication to a proof of principle, proof of concept in the preclinical setting, all the way to late-stage clinical development, translating your animal data to human. We believe there are lots of gaps, expertise, funding required that most of the startups may not be able to equip in, all at once. So there, we believe Roche has a unique angle that we could support these startups with our expertise, our drug discovery experience, as well as our footprint. And we know that Roche is just a pharmaceutical company. We cannot do everything on our own. So we also partnered with multiple partners, including venture capitals, local operators, as well as overseas partners. As Grace already alluded to, Hong Kong Science Park, we have worked together with Grace's team since 2023, and we are very good. We are very grateful that we could do this together in order to help companies to cross this valley of death and make sure that some of the good ideas are not lost in translation. Next slide. I think Grace already very kindly introduced the incubator program and the partnership with Roche, and I think here I can mention a bit more. So this is our first co-incubation program with industrial partner. We are really trying to use this opportunity to share companies, either they want to come from China to Hong Kong or vice versa, and we use this as a way to foster collaboration and fast track their setting up their operation in 2 different places, and of course by working with Roche by providing different opportunities in collaborating with us, but also being able to have access to the funding opportunities there in Hong Kong. Next slide, please. So why we believe that this process is really important, because we see that in order for biotechs to clear the path towards breakthrough innovation, there are lots of things you really need to tackle. All the way from the bottom, basically what you have observed in animal and laboratory studies, all the way to setting the guideline of the clinical practice. There are lots of steps and experiments and studies you need to do, all the way from in vitro and in vivo in animal studies, all the way to small-scale human cohorts, as well as multi-sites and randomized large pivotal trials. So we believe this is the way to go, and why we believe this is good for the entire industry, not just for Roche or for one single particular pharmaceutical company, because next slide, we believe the R&D excellence is the key to the entire ecosystem of the society, because if we could be able to do this to boost our R&D productivity by bringing more projects and assets into the clinic, we can increase the success rates, not just for Roche, but also for all the biotech companies. And we increase the value of each individual assets, whereby we could reduce the cycle time, reduce the cost of conducting preclinical and clinical trials by working with pharmaceutical companies. This will add value to our R&D and become more efficient. And this is a much better way of funding allocation, especially in this time, in the capital winter for the biotech market these days. Next slide, please. There are multiple and flexible ways that Roche can work with these companies. So some companies, they don't want to have strings attached. That's all right. Or they also Sometimes they want to have non-confidential guide and advice, but also we could also provide collaboration opportunity with Roche at the early stage, where we could co-develop or co-validate certain ideas or programs. But of course, we can do investments and obtain rights or exclusivity through capital support. But of course, this is in line with our involvement and your confidential disclosure. It's always proportional. So that is something we hold very strongly in our principle. Next slide. So at the moment, as you can see, we already have 17 portfolio companies as of end of 2023. And these companies span across 3 key elements, including novel biologies, novel technologies, as well as AI and digital. So these companies, I think in total, they already have a valuation of more than $4 billion. That's actually a great testimony of how this company have got a chance to become more successful since joining the accelerator. And Boston is so close to my heart because surprisingly, 5 of these companies, around 1/3 of these companies, they already have a Boston or Cambridge site. And I visited many of these companies these days. And I think Boston is really one of the leading powerhouses around the world for biotech innovation. So we have, Roche already has 11 early stage collaboration with these companies. We could do more. So that shows how we are selecting and working with these portfolio companies in general. So next slide. A very little touch on the facility. So this is a 5,000 or 50,000 square meter or 50,000 square foot facility just opened last year in Shanghai. This is a Roche standard flagship facility providing lab and office spaces for our portfolio companies, including biology, chemistry, as well as also level 2 labs. So if you got a chance to come to Shanghai, I can see some of these companies today, they do have a site in Shanghai. Feel free to pay a visit to our site. This is very centrally located. Moving to the next slide, I believe this is the last slide. If you're interested in learning more about the accelerator or what we have been doing, what's the latest progress we are doing, feel free to visit our English or Chinese website on roche.com or scan the QR code for the corporate Roche Accelerator corporate WeChat account to understand more about what we do. So with that, thank you very much.
All right, thanks, Dr. Chang, for your introduction to the Roche Accelerator. So we are just on time. So right now, I will leave my stage to Dr. Mali Liu. She will be our moderator for the next pitch competition. Dr. Liu.
Yeah, thank you. So good morning, everyone. And very excited to have our 14 finalists presenting to us today. And we've been through a long journey, as Min was saying, back in the January, when we were doing our preliminary pitch, we had 100 over applicants. And now it's down to the last 14. So very interesting indeed. I think just a bit of a reminder for housekeeping. So each presenter will pitch for 10 minutes, and then there'll be a 9-minute Q&A, and then a 1 minute for changeover. And there'll actually be a reminder screen to show the timing for 10 minutes. That we will try our best to keep each presentation at the same time. And before we actually kickstart, perhaps we'll give each judge the opportunity to actually say a few words. So to start, I'd like to invite Dr. Grace Lau to say a few words.
Hello, everyone. Again, I'm the head of the Institute for Translational Research from the Hong Kong Science and Technology Parks. So I've seen and I've been looking at all the presentations from all the teams. So I look forward to your pitching today and keep calm. Thank you.
Thank you, Grace. And Dr. Chiu Song Tang from Roche, if you could say a few words, please.
Yeah, so thanks, Wenli. Chiu Song speaking here. I'm a neuroscientist by training, but over the course of the past few years, working as the head of Roche Accelerator, we have screened and evaluated hundreds of companies working across oncology, immunology, neuroscience, and other therapeutic areas. So I really hope that we can discuss the science and translational potential in depth. And if there are potential opportunities that we could further engage and discuss collaborations with Roche, I will be more than happy to reach out and to discuss further. Thank you.
Thank you, Chu Song. And next, perhaps Dr. David Chu from Kendall Capital Partners.
Hi, my name is David Zhu. I am a founding partner at Kendall Capital Partners. We're here based in the Boston area, focusing on investing early stage companies spun out of different universities, supported by different faculty and research labs. So we are very actively looking at different companies across therapeutics and some in medical devices. We currently manage 3 different funds, one in Boston, one in Silicon Valley, and one in Asia. I look forward to being the contestants.
Thank you, David. And Dr. Michael Zhao of LB Ventures.
Hi, I'm Michael from LB Ventures. I'm one of the co-founders, and I'm very happy that I'm not the only trained neurologist in here. So yeah, really nice to see this organization is putting everything together, and I'm really looking forward to the pitches. Thank you.
Right, and our final is actually online. So Mr. Charles Lee of Pagoda Tree Partners, would you like to share your thoughts, please?
Hello, everybody. Good morning. I'm Charles Lee, Managing Director of Pagoda Tree Partners. Pagoda Tree Partners is an elite PE and VC fund managers with offices in Beijing, Shanghai, Boston, and Seattle. And we focus on breakthrough technologies and pipelines. And actually, one of our portfolio is Simonova, a spin-off from Sincere, where the competition is hosting right now. It's the second year we sponsor this pitch competition. It's my honor to be on the judge panel and wish the competition and all presenting companies a great success. Looking forward to work together in the future. Thank you.
Thank you, Charles. Thank you, Charles.
Yeah.
So now we're ready to start. May we invite our first company, AI-powered painless blood test device. So I believe it's Dr. Stephen Mo will be presenting. So can you hear us?
Can you hear me?
Please start when you're ready, and we'll have the timer to be on the screen as well.
Great. Thank you. Let me try to share my screen first. Can you see my screen?
Yes, we can.
Yeah, I'm going to zoom out. Is it good on your end? Full screen?
Very good. Yes, it's full screen now.
Okay,
and you can see the
great.
timer. Yeah, please start when you're ready.
Yes, great. Thank you. So our team is AI-powered painless blood test device. We're actually based in Shenzhen, China, in Boston, but because this week we are doing lots of clinical trials in China. That's why the whole team are in China. Unfortunately, we love to be in Boston. So thank you very much for giving us this chance to pitch for the final, and thank you for all the sponsor. As we know, there are lots of challenges revolutionized, such as blood testing. We know that traditionally people are very scared of needles. It's expensive, slow, complex. When I was a researcher at Harvard Medical School, I already collaborated with some of the teammates to try to think of or some of the way to solve the problems. Of course, there are lots of competitors in the market. But what we really think that we find a great solution, which is what we call the future nurses, which is a small economic device that can place on your arm. It's fast, economics, elegant, and intelligent. And overall, the traditional way of taking a blood test and send it to the lab and send it back take around 7 days. Our device can gather reading within 7 minutes, send it up, upload to the cloud, and then send it back very, very fast. So now I'm going to demonstrate or pitch some of the solutions that we have been working on, which is mainly to target small biomarker molecule, initially glucose, cholesterol, insulin, but over the next 2 years, we try to start testing some of the biomarker for infectious disease, and that's a work in progress. So first, let's talk about the device itself. We spend lots of time designing lots of models. The latest model, as you can see here, is a small, tiny device. It's only around 2 to 4 centimeters. 2 is the height, 4 centimeters is the diameter of the circular shape. The device is it's very, very painless because we use a very high level of microneedle. So how it works is when we approach a potential patient and client, we put the microneedle device on the arms, and within a few seconds, there are around 24 needles will go in and suck the so-called micro vessel, and then each volume of kit can be around 10 to 125 microliter. It's a very, very small, tiny reading, but this 125 microliter then can go to different chambers within the microfluid. So it's a very, very fluid detection, and each chamber can then detect to do the different chemical reaction with different disease. The overall design, it's not just the device itself. It's after we take the blood, how can we correctly measure the different so-called biomarker level at the same time, right? Besides insulin, triglycerol, glucose, and all those things. What we want to do is simultaneously then do the chemical reaction, then we transfer the signal from this chip device all the way to our AI system, which is cloud-based. And this cloud-based AI system is a software that we actually publish in Nature Review. We see lots of citation and get a wide publication. Because of this nature, we also get lots of interest collaboration from biotech, from hospital across the globe. So that's something we are very fortunate and thankful. So as you can see here, you can see my mouse. This micro chamber can take up to, depending on design, sometimes 24, sometimes 36 chamber. And each of the chamber can do a chemical reaction to detect the different level of blood device. It's very quick and we will say almost painless. When we do clinical trials on patients, they don't really feel lots of things. So how we then link the data, it's after we do the chemical reaction, we gather the information, we then link it to what we call our AI cloud. This is a proposal. Many of the components you see on the screen are not in implementation, but it's part of our greater scheme, which is how do we use a self-monitoring device and then link data sets with payer, with provider, with biopharma, do an offline and online combination to do a real-time, we call it synchronization, to healthcare professional in real time for medication. And that is the total solution they're working on. And we are working with lots of those different partners in Boston and in China. So you guys probably heard the story. It's a big news around 3 years ago. Elizabeth Holmes, she was worth 4.5 billion, and 4.5 billion wrong. And Wall Street Journal investigation, and it's all go down the hill. Actually, we are not anywhere near Theranos. First, they have no pattern. Second, Theranos has been around for like 13 years, not a single publication. We already published in Nature. Third, Theranos had lots of so-called secret patterns, secrecy, the secret here, secret there. None of the lab mates can talk to each other. None of the employee can talk to investor, right? Elizabeth Holmes did all this crazy kinds of fraud, right? Fraud. We are totally open-minded. If you talk to any of our investor or any of our partners, basically, there is no secret. We believe our technology is there, it's working. Already done clinical trials on around 150 patients. It worked pretty well. Of course, there is always arrow of uncertainty, but we are keep improving our advice. And the fact that we already $2.5 million US, I mean, the initial investor does believe in the value that we are adding. And the global opportunity for so-called POCT, point of care testing, is around 4.5 billion. If you look at the entire global market, the biggest one in US, of course, but immediately followed by EU and then China. EU, of course, is made up of close to 30-something countries. So individually, they still have their own regulation law. It's hard to do it. But in China, we probably want still main focus because our teammates are across Boston and China. We do lots of collaboration to hopefully, sorry, yes, 8, HKSTP IPRT mute. This is a great background noise. Thank you. So what we're doing is we're initially doing lots of device and testing, doing that in China to do the initial startup. But then we do lots of so-called clinical trials in both US and China. Our goal is to extend our pattern in our so-called clinical validation in both China, US, and EU. We're already winning lots of awards in China so far from the starting point. So a little bit background about our founding team. First, Dr. Sam is also online. You can see he's our CEO and the founder of this great idea. On myself, I did my undergraduate at MIT, PhD as a Rhodes Scholar at Oxford. And then before I started this company, I was executive at 3 Fortune 500 companies, including I would say the logo is switched here, including Optum, BCG, United Health Group, and Ping An Health in China. So kind of know the executive management very, very well. Our co-founder, Dr. Wayne, Dr. Kelly, and Joe, and we are all PhD background. We extended knowledge in lots of industry across collaboration. So we started this company not just to make a dream come true. It's we really believe we are kind of like the older founder. You know, most of the co-founders here are between age 35 to 40. We're not like 22 years old. Lots of ambition. But we do know that with industry experience and then come back to do the startup, we believe we have an advantage edge. And every single logo you see here are partner that we are already in contact and doing some sort of collaboration. Not just like an email and then I basically shake your hand. No. Every single logo you see here has some sort of clinical or financial transaction that's already happening because our technology is pretty innovative. So for example, from Harvard Medical School to BGI to Mayo Clinic, we did a lot of extended study, and the study is not only to prove the value of the testing can work, but also to give us confidence that we constantly need validation from the clinical to make sure it's accurate and friendly. And hopefully in one day we can go into mass production and help the public. And of course, we already win lots of awards, especially we have over 40 patents and such award recognition just from China itself, published in Nature Review. And what we're asking is the next steps, $7 million USD. We already received 2.5 million. For the next phase, we're thinking of $7 million USD to take 10% share of the current company. We haven't set up a Cayman top level entity, but we plan to set up Cayman that control both entities.
Stephen, it's time to wrap up, please. Yeah.
Thank you.
Yeah. Right. Thank you, Stephen. And now we'll start our 9-minute Q&A. So any questions from the judges?
So besides glucose...
Sorry, it's for the judges.
Sorry. Oh, I see.
Yeah, from the judges first, please.
Hi, Stephen, David. Nice to meet you. Where is your where are your labs currently based? Your labs?
Yeah. So our lab actually had 2 locations. Majority of our so-called clinical staff and the clinical trials are in Shenzhen, China. In Harvard Medical School, we have a small team, like 3 people, but that is more on the business-like collaboration. But most of teammates are in Shenzhen. Boston, of course, we're extending, but there still need we're a small team, around 15 people, so still working hard.
It's a question from Jusong.
Stephen, you mentioned this is AI-powered painless test. So maybe you can elucidate a bit more about the AI element here.
Okay, sure, no problem. So again, the slides I present here are more on the device itself. Where the AI comes in, probably this slide is too simple, but I'll try my best. Where the AI comes in is after the device make the chemical reaction, we'll have a reading. The reading will send a signal to the cloud. And what the cloud did is we already studied similar reading or similar biomarker reading from I would say almost close to millions of clinical studies. So what the AI did here, and that is exactly what we published in Nature. It's not the device itself. What we publish in Nature is this whole algorithm of how we can use the AI to do the study of the past clinical trials on so-called biomarker reading and self-learn the noise that we create from our APP and the device itself, and to give a signal we believe it's deleting the background noise, but also at the same time create with so-called a certainty level of the reading. So that's part one. AI to provide a signal reading. Part two is after we create this data, we then try to link this so-called AI machine learning with the different, because we're also working with some potential insurance company and hospital. So they will have claims, they will have different kind of provider data that they are in open source sharing with us. So what we did is we then take this, compare with real-time data to see if there's any abnormality. If the thing is abnormal, then of course the AI will tell us in real time that there might be some background noise, or maybe this reading is false, it's not as accurate. So again, I like to give the paraphrase, I like to bring up the story of Theranos because Theranos is over the news. We are not Theranos, or we are not anything near Theranos, is that Theranos, everything is keeping secret. Everything that we created in our AI or so-called open source system, we share with our investor, we share with our partners. So whenever we do a clinical trial or study, the data is stored online. As long as you are our partner or investor, anybody with our password, you can go there anytime, you can access the data. And we believe under our contract agreement, you are now selling that, right? Because we are all open source within our group. So the AI part really comes from the learning of the past data that different shareholder, different partner give us, and then use those learning to then predict the future to adjust. The future AI that we try to do is hopefully we can start working with real nurse and real physician to give them a real-time feedback, and they can provide some natural language processing to tell us, hey, if they think our real-time reading is anywhere close to what they diagnose in the real, what we call the medical setting. Thank you.
Does that answer your question, Chuseok? Yep. Any other questions from the judges? Stephen, Grace here.
Okay, go ahead.
Charles, you go ahead.
Oh, okay. Thank you. And yeah, and have you compared the accuracy of the result with more traditional and testing method currently available?
There are lots of traditional methods of testing, but the beauty of our device, I will stress, is we take very, very small, 10 to 125 microliter. Of course, this is much more than what Theranos claim, right, in the fraud. But if you look at the traditional testing, when you do a single blood draw from your vein, usually that requires at least 50 to 100 ml. That's ml. But our device, because it's at the micro environment, what we believe it requires different standardization. It requires different method of accuracy and different method of detection. So yes, we did compare with a machine, a direct-to-consumer machine. We bought it, we measured, it's pretty accurate. But that to us is not good enough because direct-to-consumer machine, it's for everyday user. So when we did a testing, almost all of our data are directly compared to Harvard Medical School and some of the big hospitals in China, what those big machine will do, what those laboratory will do. And that's how we do our benchmarking. And I don't know if that answered your question. Thank you.
Okay.
Thank you, Steve. Grace, you had a question? Yes, thank you, Stephen. You showed you have a lot of collaborators already. Can you talk briefly about your commercialization model? How would you generate revenue in the coming time?
Yes, that's a very good question. Thank you very much. So again, I'll come back to this page. before I go to this page, I know you guys noticed on my last page, there's a cute pig here. You guys see that? So the first step of our commercialization is how do I mass produce and have a market demand of my product, but at the same time, not run into trouble of legalization of what we call the investor come here and say you are cheating us. And we find out the great opportunity is actually the animal testing market here in China. As you know, U.S. produce lots of beef, China produce lots of pigs and lots of animals. So we are actually in collaboration, we're actually in talk with lots of the animal farms, those big companies here in China, because animal require lots of testing before they can be sold to the market, any kind of meat. So our initial trials will be tested on pig. Pig, the lots of general disease, we can do it very measured. And the cost can go down to around 1 RMB per pig, which is very, very cheap in the industry center. And also the second part is not only for we call the 畜牧业, right? We also target vet. Think about how many vets, animal doctor in China. In terms of vets, they just need to do a quick reading on if this animal are sick or not. And then, boom, they can give a treatment to the animal. So why do we start with actual animal, not human? Because the barrier is much lower. Whether regulation or law, it's not that high up compared to human. And we believe just the animal market itself. So the 45 billion that I show you, that's for human. The animal market in China itself, when we did a study, that is also at least 13 billion USD here in China itself. And nobody's doing that. I'll be honest. What people will do is they will take a few animals from the animal farm and then do the testing. But what we can guarantee is once we do the collaboration, those big, what we call the animal meat industry, we can do every single animal test. And then we are in top of not just the animal husbandry, but also with the vets. We have lots of what we call the vet corporation in China. That'll be the step one in the next.
Yeah, I think it's time to wrap up. But thank you for your sharing, and you're assured that your company is not Theranos, right? So, right, thank you.
Thank you.
So now we'll move along with the next participant. So the next finalist will be Affinity Biopharma, and they're also joining online. So please share your screen, and when you're ready, Affinity Biopharma.
Thank you very much for the opportunity to make a pitch to this esteemed group. Let me share. Can you hear me?
We can hear you, but we can't see you. So would you please turn on your camera?
I'm trying. I think I would block on your side because I was on.
Right. You can share it. Yeah, you can share it now.
Okay, thank you very much.
Let's do now.
Can you see my
your screen now? Yes.
But you still cannot see me. Well, I'm ugly anyway, so maybe you don't want to see me.
Actually, that's not the case, but
we want to make sure it's not a robot.
Yes,
I'm a real person, and Affinity is located.
We can see you now.
Oh, yes.
We can see you now.
You're very good looking.
Thank you very much. Affinity was founded 10 years ago. We're located in Shanghai, very close to the Roche Accelerator. Actually, we're situated in the building next to it. Affinity is funded, try to solve the problem of on-target off-tumor toxicity. So we try to design anti-cancer drugs that can only be activated in the tumor microenvironment. The technology is based on understanding of tumor microenvironment biology and also proteases that highly overexpressed in tumors, such as metalloproteinases, FAP, legumain. My laboratory at the Scripps Research Institute made the initial discovery that legumain, the only asparaginoidopeptidase, was highly expressed by tumor cells in the tumor microenvironment, as well as tumor infiltrating M2 macrophages. So we use some tools to design linkers that can only be specifically cleaved by these proteases, and our platform utilizes these linkers to design drugs. As you can see, we are working on small molecule drugs. Sorry. Small molecule conjugate or albumin drug conjugate. And next group of molecules are antibody and cytokines that are blocked in circulation, activity blocked in circulation, and can only be activated in the tumor microenvironment upon cleavage by legumain or other proteases. And as well as biospecific antibodies, especially CD3 biospecific antibodies, T-cell engagers. Last, not least, but we also utilize these linkers to design new second generation ADCs. These ADCs will have better stability in circulation, as well as more efficient activation in the tumor microenvironment, outside and inside the tumor cells. We have a team of 120 people with myself as the founder and the CEO, and also co-CMO role currently with Chris Wong as another co-CMO responsible for global development. He is based in Australia. And Yuan Liu is the CTO, and he's responsible for early stage development. And Dong Su is our manufacturing head. And we have, as I said, we have 120 people in Zhangjiang, Shanghai, covering from early discovery, lead optimization, clinical pharmacology, all the way to clinical development, business outreach, manufacturing, and we are building a small sales team in anticipating of first NDA application later this year. Using this platform technology, we have a very rich platform profile. And the first group of compound we're working on chemotherapeutic agents and also TKIs with higher toxicity. So the most advanced compound using doxorubicin as a payload, we're finishing a phase 3 trial in soft tissue sarcoma. The second compound using paclitaxel as a payload, and we're in a phase 2 in Shanghai. And we also have platin and DXD, etc. The second group of molecule are cytokines. We're focusing on IL-2, which is inert in circulation, but will be efficiently activated in tumor microenvironment. We're initiating a multi-site phase 1/2 in Australia and in Shanghai. And the third group of molecule we're working on tumor microenvironment activated antibodies, being a CTL4 antibody and CD47 antibody to solve their toxicity issues. And the fourth group are, as I said, a new generation ADCs. And we already have clinical proof of principle that legumin linker is highly stable in circulation and can be efficiently activated in the tumor microenvironment in human tumors. And compared to other technology that are using fusion protein, we have more efficient design process, and we basically using steric hindrance as a blocking mechanism. And most importantly, I think, is the high activation efficiency in tumor and the stability in circulation, and put us really with an advantage over competition. Let me quickly go through these platform molecules. The first molecule is albumin conjugated drug conjugate as the legumin as a linker, and the drug is given as a small molecule, but will conjugate to albumin in circulation. Exists in circulation as an albumin drug conjugate. And in a phase 3 trial, we finished enrollment of 306 patients, and we extended PFS and already saw a very clear OS advantage. And we saw CR patients in the treatment, and the patient gave most cycles already underwent over 20 cycles treatment. And to our surprise, we also found this molecule is highly active in last line lung cancer. And so we're in a phase 2 trial to enroll last line lung cancer using this compound. And the second group, we're having a tumor microenvironment activated IL-2, and it shows very good safety profile and animal activity. And we're, as I said, we're doing a phase 1/2 in Australia and Shanghai. We also applied this technology in the T-cell engager area, which completely blocked the CD3 toxicity in circulation. And I think we have a panel of antibody. We can also work with a group with their own favored antibody to construct this biospecific antibodies. And lastly, but not least, we apply this linker to ADC. And legumin activated linker are more stable in circulation. They have tumor preferential activation versus linkers like GGFG, exist everywhere. So we can expect higher efficacy, maybe a better safety profile. This is some data. And we also have some manufacturing capacity. We can make our own GMP clinical materials. And we are working on NDA submission in Shanghai for the soft tissue sarcoma indication. And now we're pursuing a two-pronged strategy for development. And we are internally developing selected candidates for single agent treatment as well as in combination treatment. And we are also actively pursuing collaboration with others. We've already established collaboration with multiple companies. We're very flexible in how we collaborate, and that's where we at. We're just finished we finished a B1 financing last year. We're closing up a part 1 of a B2 round here in Shanghai. And I think in 2 months, probably we're closing the second part of a B2.
Thank you, Dr. Ying. Yeah, I think the time is running out, so we'll proceed.
Thank you very much.
Q&A.
I will stop here.
Sure. So now we'll have the Q&A. Now we open the floor to the judges for questions.
Hi, Dr. Ying, my name is David. May I ask, do you have any process ongoing with the US FDA And for your lead indication, could you also talk about how how those compare to the standard of care based on data you have seen?
Okay, we're in process to initiating a trial of lagobacin in US, and we already started phase 1/2 for the cytokine in Australia and Shanghai. In case of our trial in soft tissue sarcoma, which is a first-line double-blind trial against doxorubicin, which is the standard treatment for STS. We saw a great extension of PFS, which is a primary endpoint, and we also have a second primary endpoint is cardiac toxicity. Lagobacin, because it's not activated in heart, so it demonstrates no cardiac toxicity at all. And we also already saw some early OS trend, so we think we can beat doxorubicin hands down.
Thank you. Thank you.
Thank you. Yes.
Dr. Liu, I have a question on your tumor-specific protease linker technology. So can I clarify, is that protease a cathepsin BNL or this is something different? If it is cathepsin, can you share a bit more about the cleavage sites and how many cleavage sites are there?
Okay, we prefer a single cleavage site, and the activating protease is called the lagoman, and it's also called asparaginoindopeptidase, which is the only protease that recognize a single asparagine as a cleavage site. When I was a professor at the Scripps Research Institute, my laboratory made the initial discovery that lagoman was highly expressed in tumor and also in TAM. It has some similarities to caspase, but with a different specificity and different function.
Hi, this is Michael. A follow-up question to the previous one. So in terms of the specificity, have you tried to do any profiling to see any off-target in terms of the activation? Thank you.
Okay. The off-target activation in terms by other protease is probably I would say no, because lagoman is the only protease that can cleave after asparagine. There's no other protease has this type of cleavage specificity. Off-site activation in terms off-site expression of lagoman, yes, there are some normal tissues express low level. Some like kidney, proximal tube of the kidney actually express pretty high level of lagoman, but lagoman exists intracellularly, and our drug either as a albumin conjugate would not reach the part, or it cannot easily permeate into cell to coming in contact with activated proteins. So we have excellent safety profile that support there's very little off-site activation, and that's the key for this technology.
Thank you.
Any further questions from the judges? Charles, are you online? Any questions from you?
Yes, and for your technology will be a pan cancer or a specific cancer type only in the future.
Can you repeat? I didn't catch your sentence.
Hello. Yes, and hello. Can you hear me?
I think one person in our next to me heard what you say, and I think the lagoman, the pan solid tumor marker, And this technology is of a very broad spectrum.
Okay.
Can be applied to almost all solid tumors, including lymphoma.
Mm-hmm. All right.
Yeah. Do you need further clarification, Charles, or does that answer
No,
your question?
no, that answered my question.
Okay, great. So if there are no questions from the judges, we can open it to the audience. Any questions from the presenters, from the audience?
So how different is your platform compared to Cytomax, which is in clinical trial in US using CD3 activated caspase, activated similar platform? How different it is? Have you done head-to-head comparison?
Right. Well, we have similar ideals. Now, of course, the small molecule conjugate is not on the pipeline in Cytomax, but the antibody and the cytokine, especially antibody, we have same ideals. The key technical difference is they are using a fusion protein approach, and we are using a chemical conjugation approach. I think I had a slide to compare the advantages and disadvantages. When you use a chemical conjugate, actually you preserve the substrate site much better compared to if you design the cleavage site in a stretch of peptide. And also you expand the chemical space. For instance, it can be a peptide bond, it can be amide bond, or even ester bond. Protease is a hydrolase, so it's not really only a protease. And so it has much better cleavage efficiency. And it can be more stable also. So you can have both ways, actually.
Thank you.
I have a question. So, in addition to Australia and China clinical trial, do you have plan for clinical trial in the US?
Of course, global development is definitely in our plan, and my approach this is try to get a clear POC in Chinese trials, and then we expand to US because the cost issues and etc. But I think Affinity is at that stage, and we need to expand to US and the European market.
Thank you. We have a question from an online audience. I'll just try and allow them to talk. Shidong, please go ahead, if you can hear me.
Yes, can you hear me?
Yes, please go ahead.
Yeah, I have a question. Like when you use the doxorubicin to compare, do you use the doxorubicin Lipson or you just use the regular solution? Another thing is the package.
Yes. Yeah. Liposomal dox does not have soft tissue sarcoma indication, so we have to use doxorubicin as a comparison because liposomal in STS is off-label use.
Okay, it's off-label use. Okay, so yeah. Another question is when you compare with
sorry, in the interest of time, I think you have to move on to the next presenter. Yeah, but feel free to reach out.
Thank you for the
offline. Okay,
Bye-bye.
thank you for being here, Farmer. I hope your clinical trials go well. Look forward to the results.
Thank you.
Sure. And for the next presenter, we have Invira Group. So please turn on your camera and share your screen when you're ready.
Hi, can the panel hear me?
Yes, we can.
Great, thank you. I'm going to share the screen. All right. Can everybody see our PowerPoint?
Still
Coming
loading. Still loading.
Okay.
Okay. You'll see a timer starting, and then you can start then.
Okay.
Let's restart.
Yeah, just give us a sec.
Okay, while we wait, I want to take the opportunity to thank BCIC for this opportunity. And also for the time and a lot of effort from the panel of judges. We are very honored to be at this stage of this. So happy to.
Timer has started, so please start your presentation. Yeah. Thank you.
Okay, great. All right, great. Thank you. My name is Carl. I'm the CFO of Invera Group. I'm responsible for basic general corporate financing functions, but mostly all of our corporate development and strategic communication, including what we do here. So Invera Group, we are slightly different from quite a lot of companies you see. Here's a problem we want to try to solve. So on the left of the PowerPoint, you see that it's great that in the past few decades, human science have evolved and grown in a way that a lot of treatment paradigms have come about, from small molecules to proteins, polypeptides, antibodies, nucleic acids, etc. But there has never been a stop of a chase for better efficacy, better safety, better targeting for any drug developer at any time. So we want to come with a solution. How do we help that? We're not here to try to say, oh, we're going to beat this drug or whatever. That's not our goal. Our goal is to make everybody's opportunity bigger. So we thought a way to do this is by using a payload vector technology. So at Invera, we have two big pipelines, two buckets of pipelines. One is the HSV1 vector. HSV1 is a very well-known virus. Also, on the other side is exosome. So what does it do? It basically acts as a truck to deliver basically currently known payloads from small molecules to antibodies, etc., whatever you have, and deliver that in a safer way, a more efficient way, in a more targeted way. So that's what we want to try to do. To just dive in a little bit, what we have in those two big buckets on the left here, you see oncolytic virus. We've selected and used HSV1 because most of the scientists here have been involved in the HSV1 virus for, gosh knows, over 60 years. And the payloads that we put on this vector are currently well-proven things like PD-1 antibody as well as IL-12. And we're using this vector to target the oncology space. We believe because of its replication capability and to deliver these antibodies and IL-12 in a specific oncology use, there's an opportunity to make these compounds more successful. So we are focusing quite a lot of effort on oncology there. Most of our trials now, we have 7 live registration trials, 4 in the US, 3 in China. They're all being done in phase 1 and phase 2. We are very different from quite a lot of oncolytic virus companies per se. We're more than just that. We're a vector company, but on the oncolytic virus space, we're the only one globally that has a intratumoral program, which is traditionally for everyone, intravenous program as well as an intraperitoneal program. This is basically due to the way we design our vector to be replication much more competent than all of our peers. And then on the exosome side, because we have a very heavy oncology portfolio with 7 registration pipelines there, we want to target a shorter registration path and target more general health aesthetics. So we're able to do a lot of surface arming, even lumen arming, even polypeptide editing on the gene side to make that exosome do exactly what we wanted to do. So right now we have pipeline that is being commercialized, being sold in China as products, as well as we just received a term sheet last night, and that will be sold in the US market as well in a aesthetics cosmetics area. So this is all done under one development platform. I won't have time to go through all the key technologies behind this platform, but I'll try to tell you the results. I'll skip this investment highlight page, but the point I want to drive here is we're quite different. We're not just a pre-IND kind of single pipeline, kind of go as far as you can in terms of IND and registration trial stage company. We have a commercializing pipeline, which is basically our buckets of exosome products, as well as a more traditional oncology registration trial. So what we have here is, our products can really, because of the way payload works and vector works, it can target basically all kinds of health problems, including lung, CNS, and I'll talk about pipeline there later. The difference we're trying to make is, as just for example, in the antibody space, I mean, most of modalities try to alter the immune environment, okay? But with the payload technology, what we've done is vector technology. With endocytosis is binding and fusion into your target cells, whether it's a tumor or a cell you want to improve the skin condition of, you're changing the tumor environment, which makes it a two-pronged approach to a cancer scenario. Or in a healthcare scenario, you make the product truly effective versus just surface the superficial applications. I won't be able to go through all the details of the technology in here. We've edited the genes out of a 150K pair of DNA of the HSV-1 virus. But what's quite special is we're probably the most, we know this virus probably the best amongst anyone in the world. And I'll show you the results because of that. And on the exosome side, we're able to play with even polypeptide. So it makes our exosome able to do targeting versus whatever everybody else has done. So here is, I guess, some highlights. We have 7 registration trials again. We've done over hundreds of patients, human patient trials. Here's some of the data to sort of drive home the point. If you look at the bottom right, you can see here that for our head and neck cancer, our current best result of 15% ORR is almost twice as good as standard of care treatment right now. But mind you, all of our patients are failed chemo, PD-1, and PD-L1. So these are last line patients. We're happy to also report, these are all published, on the late stage melanoma, also failed all IO treatments. We have patients that are in, we now have a almost 13-month median PFS. This is, I mean, incredible versus say PD-1 of currently 3 to 4 month median PFS. That's a significant enhancement. But we don't have aspirations to commercialize melanoma because of the nature of that market itself. But what's really amazing is the current data on BCG intolerant, unresponsive bladder cancer. Our current data you see here is 3-month complete response rate of 67%. This is basically on par with quite a well-known company now. It's a CG Oncology. They're about $2.5 billion market cap. That's consistent with the data. We haven't published our most recent data yet. That just came in about last week. Our current target RP2D complete response rate is over 75%. I can't show that on any PowerPoint yet because we haven't published yet, but that's currently our current most recent data. And that is without cis positive. So we're looking at 5 times market share opportunity than say CG. So this is quite an amazing backbone that we are looking at and designed. Too much detail on the exosome here. Here's a more traditional view of our pipeline. Again, 7 registration trials for the oncology side. Basically, we have 2A in China and US for intratumor. And then we have an IV, intravenous, first in the world for HSV. Both completed phase 1 in US and in China. Happy to report extremely good safety profile, low immunogenicity. We haven't seen more than sort of grade 2 AE, consistent with what the vector should do. And we have an ongoing trial with Duke University in phase 1 in the US. In head and neck, we've been granted FDA fast track designation. In GBM, brain cancer, we have been granted orphan drug designation for that phase 1 trial with Duke. We're very excited about bladder because of the very promising complete response rate. It's something that we think we're going to take it very, very far down the road in US and in China. Now, on the exosome side, because these are more cosmetic ingredients, these are generic sort of aesthetic use products, and we use a FDA-proven cell line. These are already being commercialized. It's being sold in China, and we are going to be entering the US market by the end of this year in cosmetic products. And that would basically highlight this company being a very well-balanced, company where we have a funding engine to drive that longer game of traditional registration trials. So without
Yep. Yeah, I think your time is just up. So yeah, definitely whet our appetite for more data from your blended trial. So now we'll proceed with the Q&A session for the judges. So any questions?
Yeah, this is Michael. I have a question for because you are using the HSV1 vector, right? So what target you're trying to trying to look for, and also is there any immune response for the HSV1? Thank you.
Yeah, thank you. Yes, we are using a HSV1 virus. The virus is a good virus because it's been with the human for I think over 3,000-5,000 years, and it's a dual strand DNA structure. So it's very difficult to so-called suddenly evolve. Let me see, show you here. Yes. So we've selected virus and all recent historical approved programs such as TVAC or DelayTech out of Japan are based on the HSV1 virus. And our team knows that virus the best. So in our human trials, even in the IV application where we inject the virus intravenously, we have seen good tumor response without sort of AE. So that's a sign that the virus is doing its job without triggering unrelated adverse events. So yeah, to answer your question, we've done that human trial in the IV scenario, and the drug response is good. We've seen viral activity increase over time in the tumor, but it has low immunogenicity.
Any question, David?
Yeah, since some of your trial already in phase 2, I wonder if you had some strategic discussions with a potential partnership going forward. Or 2 is can you talk about your and can you talk about your investors? Where are most of your investors, and where are you headquartered?
Thank you. So we already, for our Into Tomorrow program, is already licensed out in China to Shanghai Pharma. But we're also in strategic communication with other pharmas for outside market for other assets, including intravenous and intraperitoneal. Now, to get back to the answer of investors, we have a page here. There we go. So we have raised 6 rounds of funding. We just recently closed our C+ round with companies valued about $500 million US dollars. The list of investors is, we think it's pretty strong. We've got Hill House, Octagon, various regional sort of healthcare specific funds that's been supporting the company for a very long time. We are headquartered out in China. We have offices in Shenzhen, Suzhou, Beijing, Shanghai, as well. We have a small office in the US, but purely for business development purpose.
The last question on the on your, I think your HSV1 program has partnership with our combo study with Contenders. So just wondering how's that result coming out, and what's the decision of either progressing that program or not?
Yeah, so currently our main focus is the 3 monotherapies, in fact, because the 3 monotherapies have shown incredible results. Just the bladder cancer one is very strong, 75% plus complete response rate. Head and neck, designated with fast track. And we, in fact, I think brain cancer, GBM also has a huge opportunity too. So right now, we still want to go back to more mainstream sort of monotherapy approach first. But at the same time, we also have aspiration to focus more on collaboration or joint development with PD-1, in fact. Although, yes, you see that our vector carries the PD-1 payload, that PD-1 comes out from inside the tumor. So in a systemic environment where you have additional PD-1, we have seen very favorable results and outcomes with that. In fact, there's one very interesting observation we've seen. So all of our patients that come into our program in our trial have been basically IO failed, PD-1 failed. But after a oncolytic virus treatment, some have, for different reasons, clinical trial, that is, have exited the program, but they go back to the PD-1 and it became responsive. We believe that because of the nature of a late-stage tumor, the outer tumor surface has sort of no longer expresses all the required antibodies. But once the oncolytic virus, it works differently, right? It's endocytes, it breaks, and then PD-1 works again. So we think PD-1 really has opportunity because it already has a proven market for it.
Any other questions from the judges? Charles online, do you have any questions?
Yeah. Actually, my question is about capital markets. Right now, the pipeline already in the late stage. So what's your plan for capital market?
Yeah, for capital market, our aspiration is to potentially, get the company public. And I think, I can't disclose too much of that. So we're already at that sort of fringe valuation, $500 million or about. So it's obvious that the next step is to go public somewhere. Yeah.
Have you decided you will be public in Asia or in US?
We don't have any decisions on that yet, but preferably most likely in the US because most of our pipelines are being registered trialed in the US, and the exosome product is now being sold in the US by the end of this year.
All right. Thank you.
Thank you.
There is a question from an anonymous attendee, and the question is, how about the immunogenicity of the open vector?
It's low. It's low. The wonderful thing is, again, HSV1 virus has most adults have this as a carrier, and it remains latent in your body, and it's difficult to combat. But when we all been through a culture, and that experience, and it basically happens at tumor level too, when that gets injected in your body as a treatment, although it carries that PD-1 payload. So it can escape the initial immune response, but on the secondary immune response, it would wipe that out.
We have about a minute and a bit more, maybe a question from the audience. And if you could, say your name and your company as well.
Yeah, so based on what I know, big pharmaceutical company, when they looking at oncolytic virus, they interested more systematic delivery. Your drug delivers injection. Are you be able to do systematic?
Yeah, that's a great question. Thank you. Yes, and we agree. Most farmers are looking for, beyond into tomorrow, right? That's a limited opportunity. We have done phase 1 trial, completed phase 1 trial in both US and China for intravenous. Because of the nature of the design of a vector, it works. So you have seen here, the clinical trial has been a success in phase 1, and we look forward to carrying that towards phase 2. And the bladder program we just talked about is no longer intratumoral injection. It's an interperitoneal application as well. And we're able to achieve that very, very high complete response rate. Yeah, we're very excited. This changes the paradigm for oncolytic virus.
Yes, 30 more seconds. A last question from the audience online. So, Mr. Wu, Mr. Yuan Xu, please go ahead. Can you hear us? Right, I think we're running out of time, so we'll probably move on to the next presenter. So thank you, Elvira. Very interesting data.
Thank you.
And all the best.
Great. Thank you very much. Have a good one. Thank you.
Thank you. And the next company will be Exensis from Singapore. So if you're here, please share your presentation. I think it's quite a strong backlight. We can't see you very clearly.
Yes, let me adjust. Is better?
No, you're still very bright on your screen.
It's like UV is scanning you right now.
Yes, we can see now very clearly.
Trying
Good.
to use
Okay.
virtual screen seems didn't work. Anyway, thanks for the organizers and the panel of the judges to give me another opportunity to tell you a little bit more about Exensys, a young, creative, and fast-growing biotech in Singapore. So Exensys is very dedicated on antibody drug conjugate or ADCs. So I think we all kind of witnessed the success of ADC in the last kind of decades, represented by Daiichi and Seagen. And currently, most of the ADCs are kind of innovative on linker modifications, on the site-specific conjugation technologies. And we noticed that they're very limited or very handful payloads can be used. So that's why a lot of companies doing the innovations on existing toxin modifications like DXD structure modifications so that you can develop your own IP payload. So what Exensys is trying to offering is we hope that going to be the next generation of payloads. We call Exin dot. Because of its different mechanism of action, we hope that the platform has the potential to overcome the drug resistance to either primary or acquired resistance. So this is the core technology that we are kind of offering. And the way how we do that is basically we start from a chemo, which is approved by FDA. Before that, we actually do very systematic investigations on all the existing kind of chemical methods out there. We're looking for new chemical methods with different mechanisms of action. So that's why we narrow down onto this chemo we call TSS005. But in the meantime, we also believe the chemo itself is not suitable for ADC directly. So that's why we initiate the medical plan, a campaign when we start the company and to optimize the potency, number one, to get to the potency level to very similar to MMAE. And more importantly, to shorten the half-life from originally like in red 26 hours to less than 1 hour. So these two features with kind of more potent, similar potency to MMA and short half-life, we believe is probably better for ADC as payloads. And also after structural modifications, it kind of inherits the mechanism of action of the parent compound. So that's why we can show the potential to overcome the resistance. One example is on the right-hand side of the figure, well, DSA201 basically insensitive in this particular cell line. We keep the molecule, rest of the part the same, but just change the payload from DXD to our own payload, and then the ADC became super sensitive. So this is just one example. We have similar examples to show that cells insensitive to DM1, insensitive to MMA, insensitive to cisplatin, so on and so forth. Our payload is equally potent. So with that key technology, together with the other part, like we also have our own linker modification technology. We also have very site-specific conjugation will allow us to manufacture GLP batch at 300 gram scale, for example, to achieve very highly homogeneous product up to 95% as a single component. With this robust technology platform, we basically develop 3 categories of products. One category on the left-hand side will represent it as AT03 and AT01, which basically use our own Exin dot technology plus kind of validated target, well, has a clinical evidence with the other competitors. And the other category which we focus on first-in-class potential, where we use our site-specific conjugation with validated payload, so kind of mitigate or balance the risk and the benefits on both categories. And the other category we are kind of developing this year is to using a bispecific of ADC formats to target certain indications. So with that kind of portfolio in terms of where we are, our leading asset, which is AT03, target ovarian cancer. we started GLP talks and target for IND enabling IND submission by end of the year in US and China, followed by phase 1 trial. And the 8004 is slightly behind that. Will be submit IND filing by first or by latest second quarter of next year, and also initiate the phase 1 trial. As we're speaking, 8001, we don't have resources to develop internally, so that's why we take a BD opportunity to license this project out to a European biotech company, and we'll finalize the final legal agreement as we are speaking. So the 8006 is this year will be the lead optimization. We target IND submission in 2026. And from there, you can tell that basically from this year onwards, we'll have 1 IND per year, followed by 1 phase 1A trials per year. So this just give you some flavor about the portfolio or the project that the data we generated. For example, in 8003, we're able to develop a very selective antibody targeted the antigen, because in this antigen there's a very close family member. A lot of competitors wouldn't able to get a very selective antibody, but in our case, we can achieve more than 800-fold selectivity between these two very close family members selectivity. And we also demonstrate very robust in vivo efficacy in various PDX model. Just to give you 2 examples to show that our compounds, as shown at the purple curve on both PDX models, we can almost achieve tumor complete regression at a very low dose. And in one example, we put one benchmark, which is our key competitors in phase 1 trial in this project. That's apparently our compound is much better in this PDX model. And of course, we also generate some pre-tox data in monkey as well, which also give us a lot of confidence, because this is a new class of payloads, and the tox findings in the pre-tox in monkey give us a lot of confidence as well to support further development of these compounds, like I mentioned, target for IND submission by end of the year. So in terms of the markets, I will skip that. Is where, as we all know, kind of ADC is potentially can cover pretty broad indications as long as the cancer has the antigen target expression. So we can go through that later on. And of course, the team is important. No one can do a drug discovery alone. So my background is basically chemistry by training. And also we have a team responsible for biology, antibody discovery, and also conjugation technology, CMC as well. And recently, we also have a medical director on board to potentially support IND and phase 1 trials as well. And Dana is our CFO, part-time CFO, who is also on site actually. So free to approach to her if you want to know more of us about the company. So in terms of financial plan, so we secured seed funding of $50 million USD like 2 years ago. And this year we start Series A fundraising target for $40 million USD. The usage of this funding is mainly to support the 2 front-runner projects in IND enabling study, followed by the phase 1A trial, and also to support early stage of the project discovery. Like I mentioned, we have a bispecific ADC, and also we have a couple of other early stage programs as well. So in terms of investment highlights, I think we'll keep it short. So basically, our key differentiation is next generation of payloads, and we have very highly focused development strategy that in the next 18 months, we want to achieve 2 phase 1A initiation, and the target, the portfolio have a balanced approach of both validated target and first-in-class target. And of course, we have a team to support preclinical development as well as the clinical development in due course. So with that saying, I thank you again for your attention. Like I mentioned, Dana is on site, and Yingwen is also a scientist on site as well. Free to talk to them, and happy to take any questions. Thank you.
Right, thank you. Well, your timing is excellent. So now we open the floor to the judges, and we have Dana actually on site here right now.
Hi,
Any
everyone.
questions?
Pleasure meeting you.
Any questions?
Hi, this is Michael. I want to ask you a question for you mentioned you're targeting on a first-in-class target, right? Can you illustrate more on the target side, and also have you done any profiling for the whole profile on healthy either preclinical style preclinical experiment? Thank you.
Thanks for that question. I think we for this target, since I'm also adjunct at the National University of Singapore, I do have a colleague to do some kind of target validation work where they get the samples from clinical patients and do bioinformatic analysis. Turns out to be this target very selective on certain indications. So that's why we start this project pretty early on. And as we are speaking this project, we finished the lead optimization demonstrate pretty nicely efficacy similar to the leading assets. Currently we are doing the antibody CMC. So hopefully that we can also submit the IND by first quarter of next year.
Yeah, I'm guessing it's probably under NDA, right? The target.
Yeah, happy to
thank you.
happy to tell you a little bit more after we sign NDA.
Thank you.
I think Charles, Charles has a question online. Charles, please go ahead.
Yes, and I noticed that your DAR is 4. Can you explain more why, how it's determined is 4? Why not more?
I'm sorry, I probably didn't get your question. You are talking about AT04.
The DAR, your DAR is 4. DAR 4, right?
DAR 4.
DAR4, oh yeah, talking about DAR4, yes. So during the optimization, our site-specific conjugation, first of all, allow us to do a kind of pretty flexible DAR value in terms of DAR2. We can do DAR2, DAR4, DAR6, and DAR8. So during the optimization, we do compare DAR8 and DAR4. So we didn't, I mean, for some reason, it didn't show any advantage in terms of DAR4, probably due to the PK properties. So that's why we end up with DAR4 in the molecules.
Okay, thank you.
Yeah, I have a question on the payload. You mentioned this is similar to MMAE, so just wondering what's the pattern space of your chemically modified payload, and and you compare with DXD. Just wondering if you also compare with other toxins such as topoisomerase, and I'm not sure in some of the figures you showed comparison with DXD, but I don't know if you compared with some of the other benchmark ADCs or other standard of care. So 2 questions.
Sure. So I think it's my bad. I didn't make myself clear. This is not MMAE derivative. This is a new, totally new chemical matter. I mentioned MMAE is because of the potency range is similar to MMAE. I hope that clarify first. And number 2 questions that for the leading assets, our competitors does use MMAE as the payloads. And in that case, like I mentioned, that our efficacy in the PDX model is much better compared to MMAE derived ADC, despite that the DAR value is the same as DAR4, but the efficacy is much better.
Okay.
Do I answer your question? I hope. Yeah. Thanks.
Can you talk a little bit about your organization? Are you all located in Singapore right now, or any other labs located outside?
Yeah, thanks for that. We are headquartered in Singapore, where we are also in Science Park. It's a different Science Park. We're in Science Park 2 of Singapore, and we have a lab and a team about half of the organization in Singapore. And also we have a wholly subsidiary company in Shanghai, where I'm sitting now, actually. So we have about 15 people in Shanghai, about 10 people in Singapore. And as we are expanding to the clinical trial, we do have plan to have a small team in US to manage the local clinical CROs to do the phase 1 trials.
Any further questions? If not, we'll open to the audience. Any questions from the audience? There's none online either. So I think we'll close for today for you, Excellencies. Thank you for your presentation.
Thank you
very much.
Okay. And now we proceed to the next company, which is a medical device company, Agilus Robots. And I can see.
Hi.
Right. So can you hear us?
Yes, I can hear you. Let me share my screen.
Please.
Okay.
Yeah, if you could just wait for the timer to come on and then please start. Can we read? Yeah. Right. When you're ready.
Okay, great. Yeah. Hi, everyone. My name is Justin. I'm one of the co-founders at Agilus Robotics. I'm also joined by King. He's our senior system manager. So globally, more than 4 million people suffer from gastrointestinal and bladder cancers per year. Treatment options range from invasive to minimally invasive approaches. The most invasive approach, of course, being open surgery, which is often seen as a last resort. But taking a step down in invasiveness is something called laparoscopic surgery, also known as keyhole surgery, because only a small key incisions are made in the body to perform the surgery. But taking an even step further down, there's a lesser known and less invasive approach designed specifically for targeting early stage cancers. And this is known as endoluminal surgery. Another name is incisionless surgery because it doesn't require any external incisions whatsoever. All the access is made through natural openings in the body like the mouth and the urethra. Because of this, the recovery can be exceptionally fast with reduced pain for the patient. And the procedures can be outpatient procedures, meaning they come and go on the same day of the procedure. Ultimately, this means that there's lower healthcare costs, fewer personnel required, faster procedures, and generally quick preparation. Now, with those benefits in mind, I'd like to raise a hypothetical question to you. If a patient was told that endoluminal surgery was the best course of action, but their surgeon asked them to pick between 2 surgical techniques, there's option 1 or option 2. My guess is that the patient would reply emphatically with an option 2, please, and I'm sure the surgeon would agree as well. But the unfortunate reality is that right now, neither the patients nor the surgeons really have a choice in the matter when it comes to endoluminal surgery. Option 2 is just really not available. Now, despite that analogy to the surgeon strapping a knife to their head, endoluminal surgery can be an exceptionally effective technique in the right hands. So to perform the procedure, the clinicians are given something like a colonoscope, which has a steerable tip equipped to the camera tip. To perform the surgery, they have a electrosurgical knife, which is mounted to the tip. And to steer and perform the dissection, they have to move the whole camera with the endoscope itself. And the result is what you see on the right-hand side, a highly operator-dependent and manually intensive procedure that has not only disorientating camera views, but often paradoxical controls. And these challenges come down to the reliance on highly unintuitive and cumbersome surgical instruments that results in very steep learning curves, often resulting in hundreds of hours of training just to become competent. Ultimately, this means that this potentially very beneficial procedure is limited to select experts around the world. So our solution is to open up that access. We provide 2 miniaturized, dexterous, flexible instruments that are highly intuitive and simplified to control, using those 2 control pens in the bottom left. And the system as a whole has 3 key value propositions. The first is enhancing the surgical capabilities of expert surgeons while minimizing the learning curve for those trainees. Uniquely, our system is directly compatible with standard clinical endoscopes that hospitals already have. This includes both rigid and flexible, depending on the indication. This resultingly means that we're applicable to multiple indications, including bladder cancers and gastrointestinal tumors. Now, on the left-hand side is a real case of performing endoluminal surgery in the stomach by a surgeon in the conventional manner. So the surgeon inserts the instrument through their right-hand side. You see the knife peeking through in the endoscope view. And throughout the whole time, the surgeon holds the endoscope control handle with their left hand in a standing position. The view is very disorientating. There's poor visualization of the cutting plane, and it's generally a mess. However, on the right-hand side is what we envision for all endoluminal surgeries in the future. We have two highly articulating robotic instruments that can move independently from the camera tip. This reduces reliance on constantly steering that endoscope, allowing the surgeon to really focus on what matters the most, which is dissecting the tumor. You'll notice there's a tissue grasper on the left-hand side, creating very nice tension for the electrical knife on the right-hand side to do its work. Here's an example of our system being tested in the live animal study in Hong Kong. In the middle is one of our co-founders, Dr. Jason Chan. He's performing the procedure in the middle, holding the two control pens. In these live animal trials, we're able to test the safety as well as the feasibility of our system while incorporating realistic physiological factors like bleeding, muscle contractions, and respiration that would be seen in humans. Taking a look at the overview of our system. So we have upfront capital equipment alongside disposables following this razor blade model. The capital equipment include the positioning cart, which I'll highlight here, positioning cart, the endoscope holder, and the surgeon control chair, which is where the surgeon sits and uses the control pens. And as I mentioned before, our system is uniquely compatible with standard endoscope systems, in this case, an Olympus imaging console. And with the razor and blade model, the upfront capital equipment cost, of course, follows the positioning cart, endoscope holder, and the chair. And we have recurring per procedure disposables that are single use. And that includes the instruments and the draping to maintain sterility. There's also an optional leasing approach that we are considering based on the need of the hospitals. And taking a quick look at the competitive landscape in robotics, we can roughly delineate that between flexible and rigid instruments, as well as the instrument thickness. So on the bottom right-hand side is the classical surgical robotic system that targets laparoscopic surgery. You'll notice big names like Intuitive, Medtronic, CMI, as well as Edge, Shuri, for example. Their instruments are fully rigid because of the nature of the surgery, 5 to 8 millimeters in diameter, depending on the instruments. But taking a further step down is the single port approaches, slightly smaller, about 5 millimeters. And then we enter the small scale, where we go through natural orifices. About 4 millimeters, this company is working in the transvaginal space, and then further down is the transurethral at Virtuoso Surgical, about 3 millimeters. Now, more interestingly, at least to me, is on the flexible side, where we can start to gain access through those tortuous lumens in the body. Is EndoQuest. They're about 5 millimeters in diameter, and out of Singapore, in EndoMaster, they have about 4.2 millimeters in diameter. But the size limitation that they have results in their use of their proprietary endoscope systems, be it their own robotic or manual approach. So of course, we're occupying that top left corner. There's an untapped area for thin and flexible instruments that are directly compatible with those existing endoscopes. And having a look at the market opportunity, we're targeting primarily upper and lower GI, so that includes the esophagus, the stomach, colon, and the rectum, and the bladder indication, the urinary tract, we are very keenly interested in as well, due to the exceptionally high recurrence rate. The gold standard procedure they perform today is something called TURBT, and more than 80% of patients will undergo this procedure. But the interesting point is that more than 50% of these procedures will recur. And we've heard horror stories from surgeons saying that they've had to perform TURBTs somewhere between 6 and 7 times on one patient before it's finally gone. We're also interested in more niche areas like throat tumors, like the hypopharynx, and any other anatomy that can be accessed by natural orifices. Taking an example of the Chinese GI market, based on actual numbers of endoluminal procedures performed right now, we believe we can tackle more than 1.44 million sorry, cases per year just in China alone. And taking a glimpse at our traction. So the company was founded in Hong Kong in 2021. Since then, we've been working hard to refine the system and perform live animal trials to validate the safety and feasibility. And those trials were performed in the Hong Kong U Shenzhen Hospital, as well as the Chinese University of Hong Kong. This year, our key milestones is the start of our human trials. So first in human trials will be starting later this year, as well as subsequent FDA submission. And later on, we're targeting the NMPA/CFDA as well. And we have key study locations identified already, including the Zhongshan Hospital of Fudan University and the first affiliated hospital of Guangzhou Medical University. And so far, we have raised $17 million in pursuit of our endeavors by our venture capital partners. With our team's passion and diverse expertise, combined with what I believe to be a very strong value proposition, we believe we're very well positioned to capture this untapped market for robotics and really drive the much needed change in endoluminal surgery. Thanks a lot.
Thank you, Justin. Again, finishing in perfect timing. Now we'll open the floor to the judges. Any questions? Charles, any questions from you online?
Yeah. So what's your plan for sales and in US and China?
Right. I think for the Chinese side, it really relies on our connections with those main primary hospitals that we've identified previously. And we'll be working with them for the clinical trials as well. For the US side, we also have contact points in, at the moment, I can't say the name, but we're working with a very high, one of the premier clinics in the US as well. And hopefully we'll have some news about that soon. But King, did you have anything to supplement about that?
Yes, and our FDA, we will go for 510(k) approach for FDA submission. So actually our FDA approval, if everything is smooth, it will be very fast. We hope so. We hope we will get the approval next year. Yeah, and also after the FDA approval, we will also go ahead to for local registration for Hong Kong and also some Southeast Asia countries. And also we will go for the GBA approach, GBA policy to go into the Guangdong for some pilot establishment in mainland China. Yes.
What is your plan for the pivotal trial? Whether it be in the US or China, how many patients do you need for that trial? Your pivotal trial.
Okay, let me answer this one. For actually, we will mainly go for our human trial in Hong Kong. It will be around 10 case to 20 case within this range. And also, for the NMPA clinical trial, we are targeting for 150 case in mainland.
Would that satisfy US FDA? Or would that satisfy US FDA?
Oh yes. Actually, we have consulted to few FDA consultants, and they all actually they think we are easy to go for the 510(k) approach for PDK device approach, so we don't really need pre-clinical pre-market clinical study at the moment, but we will still have this study in Hong Kong first.
Okay, thank you. Thank you. Justin, I have two questions. One is on the technology barrier. So because I'm not a medical device expert, so just want to understand how easy it is to be replicated or be followed by other companies or major existing providers to capture that empty space that you mentioned. The second is on your business model. You mentioned that the key business model is to sell the products to hospitals or to the existing
device manufacturers such as Olympus. So want to understand, would that so who is actually more willing to purchase? Yeah.
Right. Maybe I'll answer question 1 first. Sorry, but I just had a mental blank. What was the first question again, very briefly?
Oh,
Continuing on the technology.
Yeah.
Yeah. Sorry. Sorry. Yeah. So I think you probably noticed that in the medical device market, at least in robotics, there's a really big trend. I don't want to say copycat, but it does really seem that way. Where, for example, Intuitive, there's a lot of companies following afterwards. So I totally get that concern. For the endoluminal space, so far what people have been going for is still a rigid link, or how do I explain it? It's like rigid components at the tip of the instrument, whereas the body is flexible. So the flexible part gets into the body, but the tip is still rigid. But for us, we're taking a completely different approach that we've already patented, which is a totally flexible. So even the end effector is flexible, and I can even share the material. It's a nitinol material because it's a common material out there already used in the medical industry. But the way we treated it and the way we calibrate the instruments is very much a know-how thing that is kept internally. And for the outward design, the appearance, yeah, we have patents for that already, and one of them has already been granted.
Yes, and for second question, actually we for mainland and for Hong Kong at least, which is this market we are familiar with, we are targeting to directly sell to not directly, but sell to the hospital by a distributor. Or in Hong Kong, we will definitely go for the hospital directly. But also for other countries, we are not really familiar for. We are open to have cooperation with other medical device manufacturer. We can license, or we can have some cooperation, or we ban, which is quite open actually. So different model in different countries, definitely. Yeah.
A follow-up question on that. So because you mentioned your precision is like 2.5 to 3.5 millimeter, that is really tiny. It would be nice if you can integrate your system into the current Olympus or Canon system in the hospitals, because they already have a lot of systems in the hospitals right now. Is there any possibility that you can just integrate your system in their current system? Thank you.
Yeah, actually, that's exactly what we're trying to do with our system. That's a unique selling point that we've been trying to push, which is the instruments, because they're so small, they're directly compatible with those endoscopes that hospitals and surgeons already own and are used to using. So that really ties into the whole accessibility point of view that we're trying to make, not only in terms of the price, but for the nurses and the hospital staff already, because they know exactly how those are used. They're used to the quality. And yeah, we believe that that's a key selling point for our system right now.
Actually, there's a follow-up question from an anonymous attendee, also related to Olympus. So since the device relies on endoscopy, do you have an agreement with endoscopy provider Olympus?
Yeah, so luckily we don't need something like that, because we are just an accessory system to that. So there's many companies out there that already have accessories to the existing Olympus systems, maybe a bit more basic, manual approach. So luckily we don't need to seek their approval for that. But if they want to come and collaborate with us, we're definitely open to it.
Very good. Right. Any other questions from the audience?
This is Chang Hao from Kobe Venture. Just one question. Do you think your technology platform can be expanded to the lung cancer, like the intuitive Scion?
I would say technically we can do it. Technically we can do it, but we don't exactly want to be bumping heads with the giants in the industry. So that might be something further down on the pipeline, I would say.
So your product is just upgrading the GI endoscopy, am I correct?
Yes, the GI endoscope as well as for bladder transurethral resection.
Got it. Thank you.
Thank you.
40 seconds for maybe one more question. Right. So I think we are no more questions for you, Actually, so thank you very much. Very interesting videos that you shared.
Okay, thanks everyone. Bye-bye.
Thank you.
Thank you, Justin. Thank you.
Bye.
Right. Our next company is actually an in-person presentation. So now we're moving from online to in-person. Pure genetics.
Great. Thank you, everyone. So Pure Genetic is a clinical stage, a biotech company. We're focusing on develop first-in-class CAR-NKT cell therapy for solid tumors. Our early research and translation team are based in Suzhou, China, and we intend to conduct clinical development and product commercialization in the US. So under the leadership of Dr. Yuanyuan Xu, who is our chairman and CEO, so the company raised $85 million in the last several years. And our major investors include Qingming Venture Capital and Advantech Capital. Our management team consists of highly skilled industrial veterans with extensive experience in cell therapy development and commercialization. So collectively, we advanced 14 cell therapy products into the clinical stage with 20 successful IND filings and 1 BLA approval in the US. And as a team, we are committed to navigate the company to overcome the technical, clinical, regulatory, and financial hurdles and bring our CAR-NKT therapy to the market. CAR-T cell therapy has revolutionized blood cancer treatment in the recent years. However, little success has been achieved against the solid tumor so far. So there are three major challenges faced by the industry. First is heterogeneous tumor antigen, and the second is suppressive tumor microenvironment, which inhibit CAR-T functions, and it reduces the CAR-T cells' persistence in the tumor. So what's different about our CAR-NKT approach? So AIM's CAR-NKT is powered by both adaptive and innate immunity, and it has multiple tumor killing mechanisms. On the top panel, it shows CD1D through the NKT T cell mediated pathway and the innate mediated pathway. So as you can see, CAR-NKT has the robust killing against either CD1D positive or CD1D negative, and also the target negative, but with some of the NKG2D ligand positive cells. And the T cells in both cases does not have such functions. On the bottom left panel, we co-culture with our CAR-NKT cells or NKT cells with the undifferentiated macrophage. So after 10 days of the co-culture, you can see NKT as well as the CAR-NKT cells promote macrophage differentiation into the pro-inflammatory M1 phenotype. The green bar is the T cells. So our NKT, which is red bar, and the purple bar for the CAR-NKTs, both are significantly higher than the T cells. And more interestingly, our CAR-NKT exhibited the resistance to the TGF-beta inhibition. As you can see, with addition of the TGF-beta, for 24 hours, our CAR-NKT cells cytotoxicity against the tumor cells remains the very same. However, cytotoxicity of CAR-T cells significantly reduced within the 24 hours. So we have three products in our CAR-NKT pipeline. Our lead product is CGC729, which is an autologous anti-CD70 CAR-NKT, and is also armored with IL-15. So we have two ongoing investigator-initiated trials in renal cell carcinoma as well as non-small cell lung cancer. The second product is CGC766, which is also an autologous CAR-NKT, but is armed with the dual CAR targeting. And we are very excited about its potential to treat a broad range of the solid tumor indications. CGC801 is our allogeneic product. and it's also under the preclinical development. In terms of the IP, the company has 1 granted and 3 pending patent applications. So for the relapse and the refractory metastatic renal cell carcinoma is a disease with high prevalence and dismal prognosis. There's a large patient population, both globally as well as in the US alone. So in the last decade, actually, the US patient population actually just doubled. So the 5-year survival rate is only about 12%. The market size reached 8.1 billion last year and is projected to rise to 12.7 billion in 2032. There's really limited treatment options post the second line of therapy. So tifusimab is the only FDA-approved treatment post the second line of the treatment for the relapse and refractory metastatic RCC. However, its overall response rate is just about 18%, and the median PFS is only about 5.6 months. And there's no benefit in terms of the overall survival. CD70 expressed in about 85% of the RCC, which make it a promising target. There are several anti-CD70 therapies under clinical development, including ADCs, as well as the CAR T therapy. So last year, Allogene updated their phase 1 study. They have the 16.7% ORR against all patients, including both CD70 negative and positive, and they have the 30% ORR in CD70 positive patient population. However, one thing I would like to point out is that all 3 responded patients are CD70 high expression. There's no response has been observed yet in the CD70 low or intermediate expression. So in our CGC729 phase 1 study, we have designed with the 3 different dose level, starting from 5 million per square meter, equivalent about 8 million per patient, which is a quite low dose compared to the conventional CAR T cell therapy. So enrollment started, the first patient was in last July, and as the data cutoff of April 11, we have 5 patients enrolled, 3 in dose level 1 and 2 in dose level 2. So in terms of the baseline characteristics, there's 3 patients are CD70 positive, and all 3 patients are either CD70 low or intermediate expression. All patients had at least 2 metastatic sites, and all patients had at least 2 prior lines of therapy. So here is a case study report from the 1 patient from the dose level 1, a 70-year-old male with metastatic RCC to pleural. Disease has been progressed following the administration of the 2 TKI treatment regimen. So if you look at this chart, the partial response was first observed in week 4, and this patient continuously responding to the treatment, and by week 36, the tumor reduction reached 65%. So the detailed interim findings about these 5 patients will be already presented in the American Society of Gene and Cell Therapy late-breaking session on May 10th, basically 2 weeks away. And the key takeaway from this interim data is CGC729 has manageable safety profile, no DLT, no greater than grade 2 CRS or ICANS. It has deep anti-tumor activity with 50% of the ORR in all patients and 66.7% in CD70 positive patients. And the longest of DOR is still ongoing after 9 months of treatment. We saw all the patients has robust expansion in terms of CAR-NKT, and in the blood, CAR-NKT can persist up to week 20. So we have the robust CMC manufacture process, and so far our manufacture process is 100% up to 8 patients. So we have dosed 6 patients, and the 2 will be dosed in 6 months. So here is the timeline for our key asset development. CGC 729 IND enabling study will be initiated in Q4 this year, and to support the IND filing both in US and China by the end of 2025. So our strategy is trying to do either do the co-development with big pharma, starting from the US phase 1, or we do the out-license after the completion of the IET studies. So this year we are aiming to raise more than $40 million US dollars to support both CGC 729 and the CGC 766 either IET studies or IND enabling studies.
Thank you, Sean. Yeah, I think it's about time now.
Sure. Go ahead. Yeah. Thank you.
Yeah. So now we'll open the floor to the judges. Questions?
Yeah, this is Michael. I just wanted to understand this more a little bit because it's a current NKT, right?
Yes.
Targeting out 70. So this is CD56 positive, CD8 positive, and the CD70 positive?
No, CD56 actually is one of the exhaustion markers, so we have very low single-digit expression of the CD56.
But CD8 is positive?
Yes, so in terms of CD4, CD8, I mean, depends on the patient, right? So actually, the majority of the patient is CD4 positive is more. And then, so actually, CAR-NKT, actually, one thing is very different from the T cells. Actually, I mean, some of patient has a very high percentage of the CD4 negative and the CD8 negative, double negative CAR-NKT cell, NKT cells. Yeah.
But 70 has to be positive.
CD70?
Yeah.
CD70 actually, so during the manufacturing, we are at knockout the CD70.
Knockout, yeah.
Yes.
Any other questions from the judges?
If you are planning studies in both the US and China, right?
Yes.
So the clinical material manufacturing site, where are they?
So right now it's in China. So as I said, so our business plan is trying to get a partner before the starting of the US phase 1. So once we have that, finalized, and so we are going to seeking for the CDMO here in US.
Any more questions from the judges? Charles, online?
Yeah, for the CMC and protocol, right now, when you commercialization in China, so will you build up your lab or you'll be outsourced to CDMOs?
So yes, that's a great question. So bottom line is, the company trying to keep the size as small and nimble. So right now the company has 70 employees. So we have 2 lines of the GMP production labs, and for the later phase, so our plan is trying to reach out to the CDMO in China as well. Yeah.
Does that answer your question, Charles?
Yeah, because now we know that the NKT processing is quite challenging, for large scale. So I don't know which CDMO can do that in China.
Yes, I think it's actually the Q Genetics actually is the second company has the clinical data. Let's say it's published. Let's say because we are going to present the ASGCT right 2 weeks later. So the first one published clinical data is Baylor, right? So we are the second one. So it's quite of the pioneer work. So I echo with you. Charles. So we have to train and collaborate with the CDMOs. Yeah.
Okay, thank
you.
you.
Have questions from the floor? Yes.
Hi, my name is Jeff Letter from Luminary Therapeutics. In engineering your CD70 cars, do you have any fracture side issues? And if so, what do you do to try and overcome them?
Yes, that's good question. So that's why, actually NK cells, especially the CAR-NKT cells, right? During the stimulation expansion, CD70 actually expression is very high. So our approach for now is a knockout CD70. Yeah. Actually, for some of the detail I cannot disclose today, but the CD70 knockout actually has additional benefit to the NKT cells. Yeah.
Do you use IL-7 or IL-15 to armor the cars at all?
Product itself is armored with IL-15. So yeah.
Any other questions?
Are the cells frozen when you do?
Yeah, frozen and yeah.
And what's the
Frozen and
recovery?
side. I'm sorry.
Recovery.
So recovery is above 80%.
So the cell therapy it become increasingly competitive. So I think one of the key driver to be differentiated is the low cost for patient. So what's your strategy on that?
So yeah, I mean, so direct answer to your question is, probably is our pipeline, the arrow is probably is, the best answer, to your question. So in terms of cost, but in terms of differentiation, our CAR-NKT actually has a very good differentiation, mechanism of action compared to the conventional CAR-T or CAR-NK cells. Because so far, it has really limited promising clinical data against the solid tumor. So now our data is preliminary, but very encouraging in the early phase.
So what do you think about recently there's a company, they talk about use invaluable CAR-T, and the CEO, Laura Sauer, made a statement, if their technology works in 10 years, there's no need for CAR-T therapy.
So, so in vivo CAR-T actually, in terms of cost, right? So in terms of the cost, yes, I agree with that. But for example, for the in vivo CAR-T treats the solid tumors, it still faces very similar questions. Also, in vivo CAR-T is trying to using the patient's own immune cells, right, inside the body. So as you know, so a lot of patients after all kinds of different treatment, so their immune cells may not respond that well in vivo. So that's also another challenge we have to overcome.
Can you tell us an example of another company that has done something what you're doing to get CAR-NK or CAR-T in China and get a CDMO in US and has shown some success?
Oh, you mean in China?
Or in US?
Yeah, really, the CDMO business, is really, let's say, have the very successful in US. So, for example, right, so Gracell, which just purchased by AstraZeneca. So, they are using the same approach here. So, to run the US phase 1 study.
Thank you. So if there are no further questions, it's almost 12, which means that it's lunchtime. And very happy to say that we have kept time very well. And we'll come back after 1 o'clock for the rest of the day. Thank you very much.
Thank you.
Yes.
Oh, hi.
Great. Yes, you're all the way from
Yeah, from the very beginning.
So it's like a
Yeah, no, I think some of this is quite interesting. Most of them are oncology.
Hello, yeah, this is
I am double from the city. We're saying very nice to meet you.
Yeah, nice to meet you. Yeah, so originally from China and yes,
Okay, yeah.
Working on this startup.
Okay, yeah, I just
Yeah.
So when guys come
Oh, nice to meet you.
nice to meet
the weather more sunshine.
that would be in Singapore.
Welcome anytime.
She's actually our investor. Now Boston.
Yeah, they run a pitch competition at their high school. Not as high okay. So you know, I want to do a pro startup. Like yeah, some of how I kind of just yeah. The high school senior like 19.
这个转过那个 CBA 的大会，每年一次这样的一次，就是在加州那边的高。
Nice to meet you. Oh, you are going to be last year.
Witness last year.
Oh yeah, this is Annie.
非常那个，非常中华那个第三学的。
对，主要可以选
然后跟崔律师。那我们是前爬的人，
人家说清楚，我操，你得说清楚。温城孩，
好朋友，好朋友。
Yes.
Michael 这边非常的像。
所以这个分儿。就双十啊，那也是。
挺卖的。
可以啊。当然。
然后他们会
大家对。Oh, sure. Excellent.
Oh, okay. We'll take a week.
Yeah, that's Amber.
Albert Wall.
Yeah, I've been to the
对，
你认为我以为你们早就有太多共同朋友了，真的是。
然后我想签了这个上大戏了。So currently， 所以我们香港的还有香港的。我这边大家 save the time， 到最后就发现2个 S 应该都是差不多。然后后来跟着我现在。Very small world， 做的还是。I think 所以我们想知道。
这个如果。
对，我们现在就想做那个diagnostic，
I mean, the more funding the government
So anyway, I also published maybe 7 papers on COVID.
Right.
We just moved
Our manufacturing.
projects.
Like your
Always around like
It's going to build it.
As a founder, I'm not an employee.
With
One
the
employee.
IDMC has the space. They're going to build a huge just so they all their used to be done by now it's
And our animals. Yes, that's the hardest part. Shows that you've been involved. Because what it ends up is that, well, we have 6 or 7 hours.
之前应该是在买超那个工作室，在买超那做 strategy， 然后现在是但是我帮国内生物在这边做举手，碰到这个叫生物细项。
What drugs might always eventually
we don't have anybody for them changes like that.
Right now, stage when I finish,
when you're all together to say, well, sometimes you get
there's nothing.
I have a hard stop at 8:30. We're going to go.
Yeah, so yes, well, multiomics is a skill tree. We actually have combined
combining. No one talks. Not much. So.
还是在中国，还是在中国。
BCIC.
I also
We also
NPU 西海岸。
哥伦比亚。
那他们有好的渠道吗？还是说直接就跟教授那个？
你们的是在创新。
是这样的。
就是外面你可以拿一个拉丝。
我还以为是带您去会议，
因为我们下午1点钟就是另外剩下的下午的时间还挺紧。拿个老师。
好，你好，base 在上海吗？
对，但是我们也经常哪儿都出差。
我们的 headquarter， 我们的主要的 office 也是在上海。
啊，我知道。
你知道，你跟
前一段时间你们有个
什么时候有机会看还跟打飞机？好啊，你那个是个飞鱼。
我真的是，我对他也挺
是。
他有一次，我有个校友也是自己有个基金，他有一次也要招人家过来做什么。那之前的是 investment director， 我说人家是 founding partner。
对，反正我觉得，但是你们投的一些早期项目还不错。
好，
因为您贝斯在美国，
我贝斯在美国，但是我基本上就是中国、美国时间一半一半。
那肯定的。
我6月份要回上海，在那边待到9月份的。
对，中国的项目还是多一些嘛。
我们可能以后主要的策略也是两边看那家伙，因为这个技术就是国内的整个的原创型的这个还是得从美国这边。
国内现在大家也用的就两极分化，
美国还是离谱。
对。因为大家也发现了特别新的国内就是靠国内就是号称可以做拜森。
我是那个 work very close with 易主席。
啊，易总。
我在 BCIC 是听到这个问题，咱们这个活动就是去年也是挺丰富的。我们没有牌，你们没，我们是 volunteer。
对，他跟我介绍挺好看，
健康待遇。
对，你是个 volunteer，但你平时是个
对，我主持是这个。
啊，在 LP，OK，
对，科望。
谁？我跟
Judy，对，那个谁 ，Darren 也是从Roche。
我知道，
对，
那个 Darren 就不用说了，就更熟。Darren 红涛啊，
对，
双博士加一个微信。
对，你们在 Judy team。
对，基本上我们就做
找我可以吗？你看你效率高。对。
到时候到上海的时候，您有没有跟我们这边的，就是合伙人像黄鑫往里面？
简单聊过几次，但是后来聊的少。
哇，
后面我们就是很多嘛。后来就是大家就
应该是比较
没有，我觉得就是后来就是很多时候各种各样的 VC 都会找过来，我们也会找他们。然后估计你们的黄总，因为利明本身他应该是个 professor， 利明
其实他
已经
完全
已经挂了，
他就是为了维持个 professor 身份才到这种深圳来的感觉。现在他其实基本上，我说80%都是在开发。
因为利明大家都喜欢看他的 我看不是他的那个大王雄心
啊，对
对吧？
他天天写那些玩意儿。
那个之前大家看到这个，大家估计好像不怎么写。
就最近我们这边比较各种事情忙，
他那个忙啥呢？
不会啊，我靠，
他还要忙投后啊？
他是我们生物组的组长。
但是你想想，他作为一个相对来说偏 science 的，你让他来做投后，他也没这个。
但是我们那边的这个 biotech 的人背景都很偏 science。所以他们都不是特别的，就在 bio sourcing 这块不是特别的。
bio sourcing 就要找我们找 BCIC， 因为我是去年的话，像那个海峰为什么就很有缘分？去年那个品牌王健，因为我之前做投资，做投资的时候看到他项目，然后这边办会，结果没想到我跟他也这么讲，没想到就是拿第1名，因为确实项目很好，对，然后今年我们是继续办，今年也是项目，对，去年我们是146个，今年我们明显是感觉到就是这个环境不如去年
是，
但对这样情况下，我们还是有100多个项目，然后那个包括我的话，还有几个其他的那个企业的负责人初赛的这个 resourcing 跟初赛的这个，然后后来
一项目都是很好，
就
因为很多项目都是其实你看都是国内的。
对，一个是就是说我们因为 BCIC 它以前呢，它有30多年历史，以前是好像是金融系的，后来转到这块儿，因为在 Boston 呢就做生物力，但很多人回去了，很多人回去了，可能他是。
from GB20 Therapeutics, Dr. Shirley Chu.
Yeah.
Right. Can you hear me okay? Maybe I need to unmute myself.
No, don't. Yeah, don't unmute. Yeah.
Okay.
The audio here is very good.
Okay, great.
There'll be a timer, so I think.
Right.
Right. Yes.
All right. It's my great pleasure to introduce GB20 Therapeutics, where we integrate AI, genomics, and cancer biology to create revolutionary antibody therapeutics. So let me start with a question. Do you know the top-selling cancer drug last year?
Here.
The anti-PD-1 and the related anti-PD-L1 antibodies together sold for $40 billion together. Do you know the conventional approach to discover an antibody drug like anti-PD-1 is from the target to the function to the drug? So scientists first need to know there's a gene called PD-1, and its interaction with PD-L1 makes the T cell dysfunctional and our immune system weaker. Then you can develop an antibody to block this interaction, to boost the T cells for cancer killing. This whole process is long and expensive. You know that cancer patients' own immune system already make anti-tumor antibodies such as anti-PD-1, PD-L1. So when we study a tumor, we don't just study the cancerous cells. The immune cells are also in the tumor, and many of them are making anti-tumor antibodies. But in order to win the war, you need an army of good soldiers, and sometimes they only have a few good soldiers, so they don't really get to fully cure the tumor. And so if that is the case, we wonder, can we learn from patient tumors on how to really fight cancers, And so at GB20, we have tens of thousands of tumors as our experimental system. So using existing tumor profiles, we can computationally assemble, by this time, over 100 million antibodies directly from tumors. And we can use AI to predict these antibody targets. And we are specifically interested in those targets that are associated with repeated antibody expansion across many, many patients, because that indicates that these antibodies might have therapeutic potential. And we recently joined the NVIDIA Inception program to gain more AI support. So the GB20 actually able to use this approach to find the novel target and antibodies in patient tumors. Yes, we did. From the AI, we found that many patient tumors make a small amount of antibody against a target called IGSF8. And also from CRISPR screens, this is another technology that we were the pioneer for. And using CRISPR screens, we found that this IGSF8 gene, it normally does not influence cancer cell growth unless the cancer cell is under immune pressure. For example, on the left is when we co-culture a cancer cell with NK cells. On the right is when we implant the cancer cell into mice with full immune system. That's when this gene is important. So without understanding at all what this target was doing, we decided to use AI to predict the antibodies directly from patient tumors and tried it on animal tumors. So this is just one example on the B60 melanoma. We let the tumor initially grow out a little bit, and each arrow indicates a dose of the drug. Blue is the control, red is the anti-PD-1, green is the anti-IGSF8, and purple is the combo where we have the dose of each. And you can see that anti-IGSF8 alone or in combination with anti-PD-1 have really good tumor inhibition effect. In fact, we have repeated this experiment in non-small cell lung cancer, colon cancer, 2 different triple-negative breast cancer, and prostate cancer models, and we always see these type of phenomena is unprecedented preclinical efficacy. And so here, then we say, okay, how does this work? This really work? So we are taking an approach opposite from traditional drug discovery approach. Here, we already saw a drug working. before we start working on the function and the mechanism of the drug. And so we found that IGSF-8 is kind of like a PD-L1, but for the innate immunity. We know that PD-L1 is like for adaptive immunity, but in this case, it's a immune checkpoint for the innate immunity. So IGSF-8 is highest expressed on malignant cells, and it has a receptor on NK cells and a receptor on dendritic cells. And the antibody that we predicted directly from patient tumors can block both interaction. And this is especially important in about 2/3 of the solid tumors that are immune cold because they lost the antigen presentation. So basically, once the cancer cell lose antigen presentation, they become invisible to the T cells. And theoretically, the natural killer cells should come and kill the cancer cells. But because of IGSF-8, these cells get suppressed. And when we have the antibody, the number of NK cells increase, it will start killing, the antigen presentation also increase, and the dendritic cell population increase, and that in turn turn on the T cells. So it is really a very novel mechanism and also very, very complementary to the current PD-1, PD-L1 antibody mechanism. So does this antibody drug work in cancer patients? We started the phase 1 clinical trial a year ago, and by this time, we dosed more than 30 patients. We are activating even more sites. The drug is well tolerated in patients and showing early clinical benefits. And we are about to start the dose expansion cohort. And also last week, we signed a contract with Merck for a collaboration. So they provide us with a free anti-PD-1 to start a combo safety study later this year. So why are GV20 and our drug GV20-0251 unique? So GV20-0251, this is the antibody against IGSF-8. It's a novel innate immune checkpoint, evolutionarily conserved between human and mouse. I gave an oral presentation about this earlier this month at AACR. And this week, we had our paper describing the function of IGSF-8 just published on Cell. And this is the first AI-designed antibody against an AI-predicted target in the clinic ever. And we have a first-in-class antibody and at least 2 years ahead of competitors. So we're very, very unique here. Do we have the right team to succeed? So initially, our core team started with academic pioneers in genomics and computational biology. And then we recruited more industry veterans. Xingfeng is in biology and translational work. Huawei is a veteran for antibody discovery protein engineering. And Ying had business development and strategy expertise. Jie has CMC and quality expertise. And Connie runs our clinical operation. And for our advisors, initially we had like world-renowned scientists, cancer genomics, adaptive immunity, innate immunity, and AI expert. But we also have industry veterans for research, early development, and the 3 really good clinical development experts. And we, in addition to co-founders and investors, we have Chris Mirabelli on our board, who has been he's the founding CEO of Tim Springer's very first company, and has been an investor and lead CEO for many companies. So our funding history, we have raised $74 million so far. We have about a year runway, and we'll continue our clinical trial. We have additional program going to be ready for IND enabling studies, and we're raising up to $80 million to finish, to get to the clinical proof of concept and have additional program ready. So I really have faith in the target and in this drug and in this company. That's why I left my 20 years of Harvard professorship to join the company full-time 2 years ago. If you want to make a difference in the world, willing to take a risk, please join us to find a cure for cancer. So thank you very much. There's a little bit more information about the company. Thank you. Thank you.
Thank you, Shirley. You finished with 1 minute to spare, so very good. So now we open the floor to the judges for questions. Shuso?
Yeah, Shirley. First, congrats on the Cell paper. Apologies, I have 3 questions. So one is that if you look at the
the past life of GB20, I think there are some some antibody programs validate targets, and also you have also some ADC targets for CRC, or also some programs on KRAS. Maybe you can highlight, okay, what are the status of these programs in addition to the ITSFA? This is question one. Question two is you mentioned about the competition, and just want to understand a bit more about the antibody of lacutamab from Pharma. So because that is targeting KIR3DL2, so we want to understand what do you believe is the benefit of targeting a IGSF8 versus that one, because that one, I guess that is ligand, but IGSF has several targets.
Yeah.
The third question is you mentioned you saw some efficacy data also in other models, not just the melanoma, but also I think lung cancer, breast, colorectal. Do you have like dose-dependent tumor killing across these different panels?
Yeah, maybe I answered the backward. So yeah, we do see dose-dependent killing, although at certain level it does plateau, right? Like we get a target saturation. In terms of how are we different in terms of lacutamab? The interesting thing is lacutamab does not actually block IGSF interaction with this KIR3DL2.
Okay.
And also IGSF8 has more than one receptor. I think this dendritic cell receptor that we is going to be also very, very important, right? Like so also for KIRs and also the dendritic cell receptor, I can tell you it's a different gene between human and mouse. It's and also polymorphic in human. So it's fairly hard to do preclinical studies, whereas IGSF8 is evolutionarily conserved and our antibody cross-react to human, mouse, monkey, rat target at the same time. What was your first question again?
The other programs.
The other programs, yes. So initially, we really focused on IO, but as you can see here, we get the drug working, and it took us another 4 or 5 years to work on the mechanism, especially if it's IO. It's just very complicated. And even with this, people still say, oh, we don't believe this, right? That's why we feel like now that a lot of the resources are being sent to the clinical development, we have to kind of conserve our resources for research. Therefore, we now focus mostly on ADC, because there, we can predict the novel targets, we can get very good antibodies, and then we can collaborate with pharma, with with a proprietary linker and payload. Then the biology is much easier.
Yeah, so for the ADC, you are not developing linker and payloads in-house, right? You are working with.
We are generating pilot molecules in-house, right?
Okay.
Because those you can work with like WuXi.
I see.
Yeah.
Thank you. About your animal models, have you done PDX studies? Do you have data from either PDX or CDX?
So for IO, you use syngeneic tumor models, not PDX, because we need a full immune system. But from the paper, we showed that basically with this antibody, if you deplete CD8 T cell, it has small effect. If you deplete CD4, it has stronger effect. But if you deplete NK cells, the effect completely goes away. So NK is really, really essential, but T cell may have some effector function still.
A question for the target of the IGSF8. So except the viability, it's also affect the movement of the cancer cells, right?
Potentially.
Yeah.
Yes.
So have you got any data from animal showing the movement?
We haven't specifically tested this, although we are collaborating with the university to check this. From the tumor RNA-seq with the monoclonal, this antibody treatment, we consistently see the EMT signal decrease, which suggests that this potentially can reduce metastasis.
Any questions from the data?
Can I ask a question? So just from the science-specific question, like IGSF8 is targeted innate immune cells. Did you find is there any relationship between this and the PD-1, PD-L1 signaling pathway?
There are actually some interesting observations. This gene is also a little bit expressed on immune cells. And when PD-1 is, T cells are activated, you see PD-1 go up, IGF-1 will also go up a little bit. Some of the behavior it has in terms of stimulation and also suppression of NK cells kind of are similar to the PD-L1 effect on PD-1 on T cells.
I think I have a question from Charles online. Charles, please go ahead.
Yeah, I saw your phase 1A almost finished, right? So is any clinical result you can share and any PR you see already?
We can't share the clinical results yet, but we are submitting an ESMO abstract next week, which I think in the fall we can disclose the data, but we do see some early clinical benefits.
Sure. I have a question from the anonymous attendee. So which MOA and safety biomarkers used for IGSF8 antibody in vivo models?
In vivo model, we can measure the tumor size and also the tumor RNA-seq, and also we look at the immune cell infiltration, basically in the tumors. NK cell population, dendritic cell population, T cell population all increase, and also we see increased NK killing, increased antigen presentation, and increased T cell signaling. So,
sure. I hope that answers your question, anonymous attendee. Right. Any other questions?
What is your current financing round? Are you raising another round?
Yeah, we are raising kind of a B+ round, but we know the market might be different, so we actually have different strategies depending on the amount of money we raise. Like different things, and also will last us to a different time.
Any other questions? I think no more questions. Thank you, Shirley, very much for your presentation.
Thank you.
And we're now changing gears to a medical device company. Fox, fine. Fox, fine, are you online? Ah. Sure. Moving along, Reperfusion Pharmaceuticals.
Good afternoon, everyone.
Good afternoon.
We don't answer. Hold on a second. So you can.
So just verbal, come on.
Sorry. You had a
update,
right?
Yeah, can you use that?
Yeah, of course. Right now.
Great. I'm really glad to be here. I'm Sean Hu, CEO of Reperfusion. So when the slides comes up, you'll see that I'll be talking about our non-degrading molecular glue platforms and our pipeline. So we are a chemistry innovation company. So if you go to the next slide. Quick intro on myself. I was majored in, I had my BS from Beijing University in organic chemistry, and my PhD in genomics from NYU in mid-90s. And then 99, I went to work for my MBA. So for the 25 years after that, mostly I focused on the commercial strategy part of our industry. Our company, you can, so my expertise in the past 3 decades mostly are in 2 areas. One is in drug development commercialization strategy. The other one is in building business. Just was mentioning that my major working is entrepreneurship, building company. So Reperfusion right now is at the tail end of an A extension round. So we initially had, basically we wanted to use it as a bridge until to the point we raised B round. So our initial quota was about 10 million, and as of last week, very delightfully, that we closed we had first closing for this round with 11 million. So we are oversubscribed. Given the strong momentum, if you can click again, so we decided to expand the quota to 15 to 17. So for anyone, any investor in the audience who would like to participate in this round, you're more than welcome to be part of the second closing. So if you think that the check size is too small for you, if you click one more time, we can start negotiation discussions for, if you can go back, yeah, for B round, right? So we're thinking about 50 to 80 million, right? That to close the first half of that next year. Historically, we've raised 30 million in the past. So if you go to the next one. So many of you may have heard molecular glues. So what is it? Now, if you just to share with you kind of two anecdotes so that we can see that molecular glues now has become a key focus of our industry, of the investor community. So one is that Dr. Jay Bratner, he is the new head of R&D at Amgen, right? So a couple months ago, right here at Boston, he gave a keynote speech, basically to advocate his view that molecular glue shall be the key focus for pharma industry and the investor community, and he concluded his presentation with those three words: just glue it. So even more recently, also at Boston, we had a molecular glue conference. So Genentech at the conference actually very encouragingly told people that the company has set non-degrading molecular glue as a high priority. So it's now putting the industry is putting resources behind it. So how does it work? What value does molecular glue bring to our industry? So the intention of molecular glue is arguably to address what we believe is the biggest challenge in our industry, which is how to drug the undruggable disease target, which accounts for the vast majority, at least 80–90% for all the disease targets we ever discovered. And most of them are protein-protein interaction targets inside cells. Then, as we know, antibodies powerful, but they're not useful for these kind of those kind of targets because in most cases they cannot go inside cells. Small molecules can, but they're too small to be effective for PPI. So how to solve the problem? Right. So you may have seen in the past years and decades that our industry is trying to come up with solutions, mostly towards the direction of how to overcome the small size of small molecule, kind of towards larger drug molecule design, more interactions, and more way to maneuver the interaction with PPIs so that can better inhibit modulate its function. So you may have seen something like microcycles to go from a small molecule to a little bigger, but they're just incremental changes from a small molecule. You may have seen recently, right, some major deals done surrounding cyclic peptides, but yet there's a lot of challenges with that approach, and also kind of they're yet to be proven. You may have heard the PROTACs, right? The protein degradation, and there's yet challenges that there's no product on the market yet based on these. There's for example the hook effect, deep challenge to find clinical dose, and so on. So all these challenges that just tells us we as an industry, we don't yet have a good solution towards the biggest challenge we have, right? So then what is a better solution? So a big problem requires a big solution. So this is where the non-degrading glue comes. So non-degrading glue basically is a way to generate very big protein-sized drug complex inside cells, right? So once it does that it can very effectively, because of its bigger size, because of the other superior drug properties, it can very effectively interfere with PPI and displace the natural kind of protein binder and modulate its function. So that's the kind of idea, right? The value. So now if you go to the next slide, I'll show you how it basically works. This is a typical structure of a non-degrading glue molecule. It's a cyclic molecule and has two functions. The yellow part is what you expect, which binds to specifically to a disease target. What's different is the blue part. So the blue part binds to a tool protein inside the cell. So once the molecular glue molecule goes inside cell, it binds to this very big blue, so-called typical FKBP, right? So it's a family of proteins. It binds to it. So then it transforms itself immediately into a protein-sized drug, still with the yellow part exposed. specifically binding to the disease target. And also as a result of this kind of gluing function to glue these two proteins together, it generates, it creates interactions, new protein-protein interactions between the blue protein and the target protein. So just going to enhance the interaction surface. And the result of that are the two critical advantages for this type of way of making drugs. One is, as you can see, right, much more, because of the much larger size, protein is very much easier to be inserted into two interacting proteins and interrupt so much more effective in PKI inhibition, but also much greater selectivity. Because that when you have bigger surface area, bigger interaction with the disease target, you can better discern the structural differences between similar proteins. So if you go to the next slide, I can give you one very tiny example of how much greater selectivity is usually accomplished as a hallmark for non-degrading glues. So here I'm showing you two very homologous protein, a hot target in the industry. CBP, a next generation immuno-oncology target, but it has a homologous protein called c-CBL. If you click, you can see that the binding site, unfortunately, for small molecules in those two homologous proteins, they're almost identical. As a result of that, using small molecule approach, it's almost impossible to have enough selectivity. Yet, if you look at the bigger areas surrounding it, the structural difference between the two proteins are very notable. So if you have had a glue molecule that brings that additional glue protein and binds to the bigger area, then you know right away that you must have much higher selectivity. Yeah, so that's kind of, you can see that structurally, that's the scientific foundation for much greater selectivity, usually 3 to 4 orders of magnitude higher than the usual approach. All right, so knowing that, so that's a non-degrading glue. That's a very attractive MOA. So what value do we as a company bring to our industry? So first was mentioning that molecular glue was first discovered in 1991 by these two, basically in this seminal paper, right, in Cell. The first author of the Cell, Professor Jun Liu, and the senior author was Dr. Schreiber at Harvard, right, so it's local. So Professor Jun Liu is our scientific founder, right? So he founded our company after he left Harvard and joined Johns Hopkins as a Kendall professor. So you can see, if we talk about molecular glue space, no, we come from the origin, right, of the scientific discovery. So go to the next one. So then, ever since the discovery of molecular glue back in 1991, our industry actually jumped on it right away. Right, because the advantage of this mechanism is it's not rocket science. Everyone can see it. So since that point, there's been about 8 drug products, blockbusters, on the market based on this MOA from various companies. But so far, the success has been limited to only 2 disease targets. And the industry, if you click, the industry has been wanting to expand this superior MOA to other disease targets. But it's the technical chemistry difficulty that's been preventing them. Those glues are very hard to design and very hard to synthesize. Then if you go to the next one, that's exactly where we come from. So under the leadership of Professor Jun Liu in the past 2 decades, that he envisioned a way to overcome that challenge of coming up with the glue starting point. So his approach and our approach.
This is the last slide because time is up. So you can wrap this slide, please. Yeah.
So can I rush through the rest? Yeah. So our approach is come up with the molecular glue libraries. If you can just click through. Yeah. That's a way to effectively discover molecular glue hits. And so our greatest contribution to the industry is that we have the first and largest proven molecular glue libraries in the industry. Okay, just very quickly go through this.
I think we need
to stop. So now we don't have time to go through our pipeline in detail, but I just want to highlight that. So we are preclinical stage that we expect to go into clinical trial stage for the program for treating acute kidney injury next year. So let's go through that. And I just touched upon CBP as next generation IO target. Now, because of the activity challenges that all other companies, including genetic face, we're in a position to be best in class. So that if we can just,
the last, just one last minute.
Yeah. So we have a strong team, experienced team, right? And if you can just go through, yeah, and we're supported by some of the most respected people in the industry, right? So former president of R&D at J&J and former chief medical officer at AstraZeneca, and we expect to, as we go through next round of financing, if you can just click through.
Yeah, I think thank you, Sean. I think we'll wrap up right
But
here.
go public. The goal is that 2026.
Okay.
Yeah, so that concludes it. Thank you.
Thank you. Yeah, and
Thank
we'll
you.
start the Q&A session.
Sean, couple questions on the MOA. So first, I think you have been, you are stick to the rapamycin or FKBP. What's the difference or the differentiation between your MOA versus the other non-degrading group, which is RevMed's cyclophilin A?
Yeah.
So that's also this is one question. The second is if it's a degrading glue, for example, there are drugs being utilized in cerebral, right? So is there a particular reason, for example, we decided, I mean, there are targets which there are value to degrade them, right? So why you just don't want to degrade them? So basically, I think I can start with these two questions.
Good questions. First one, you mentioned cyclophilin A.
Yes.
So I mentioned FKBP. So those two actually are the two tool proteins that's ever discovered under this mechanism. Cyclophilin A right now is showing has one marker, so there's major differences, right? Both are showing success. Cyclophilin A actually is being utilized by our reference company, benchmark company, Roche Medicines, which is a single target company, phase 1 product. Because it's showing success using non-degrading molecular glues, it's a market cap of $5 billion, right? Today, almost 100 times of ours. So we have a lot of room to grow. Now, difference is, both proteins are the two proteins are discovered by our founding scientists, right? So we understand the difference between the two very well. FKBP is superior in several aspects. For one, that is far more validated. There's 7 drugs on the market based on FKBP. There's 1 based on cyclophilin A. Secondly, there's also kind of major distribution of the two proteins in our body. FKBPs are exclusively inside cells. Cyclophilin A are both inside and outside cells, which means that for Roche Medicine drug, in order for it to go inside cell to reach the eventual target, a lot of it has they will bind to cyclophilin A outside of cells, right? So that in a way can decrease the efficiency of that. And lastly, FKBP actually enables us to build very large libraries, including DEL library. The different protein structure of cyclophilin A actually would disallow someone to actually build a DEL library. That's why. Because there's a spherical hindrance to attach it in the tail. That's why if you look at Roche Medicine, they have to use retinal design. We have a library for screening, which is very effective, and we can do retinal design. So that's why Roche Medicine, $5 billion company, it's very hard for it to expand beyond KRAS target they're working on. For us, we can easily apply our platform, this MOA, to many different disease targets. That's why I believe our potential is greater than Roche Medicine. That's why I believe our market cap in the future can go far above, far beyond $5 billion.
Okay. The second question.
Second question.
Cerebral.
Degrading glues, if you look at the 33-year history of molecular glue, but degrading molecular glues are relatively new. First discovered in 2014, right? The biggest is exciting, right? So as represented by some of the best-known products from Celgene, Revlimid, and so on. The biggest challenge with this degrading glue mechanism is that you do not know which disease target can be applied. That's why if you look at every single degrading glue companies, their primary focus is to identify which disease target can be applied. Can be applied with this MOA, right? And which then they determine which disease can be applicable. So it is so I call it as a reversed kind of approach, right? It's like first you find the suitable disease in the needle in the hay. Here it's very different. Non-degrading glue just can be applied to almost every types of diseases, right? Now with our libraries, we can readily do that. From our track record, if you give me any kind of disease target, we have a success rate of about 50 to 60%. When half of the time, we can give you a group hit.
Is there a particular group of targets that are more suitable for FKBP?
So we have been trying to answer that question. So far, we have not identified specific types of protein that's not suitable for this. Beyond that, we have found some sweet spots. Just more success come from. For example, we have had success with at least 3 membrane proteins, and we know membrane proteins is a top spot for degrading glues, right? So because like we have broader, but much more validated. So if you work with degrading glues, I think it will have its space in science in our industry, but we don't know where they are. Here we do know. It's ready to be applied for many diseases. So if you want to invest in this company, this is a sure bet.
Hi, a question for the library. You just mentioned the repa glue library. What is the criteria that you design library to cover the diversity of the proteins hard to drug? How you can make sure the diversity is covered?
Great question, right? So although I admit first I'm not the best person to answer that very insightful scientific question, but I can say that there's sufficient building blocks, the R blocks, that we can leverage in creating sufficient diversity, because we choose amino acids for the right half part of our microcycle. Now, but that's not the only key. Compared to any other libraries you have seen, for molecular glues, there's a very vital key that if any medicinal chemistry knows about it, they will jump up and love it, which is we call this 3-dimensional diversity. Because most of the potency and selectivity of the compound does not come from the direct binding of the glue molecule with the target protein. Rather, it comes from the binding between the 2 proteins, the blue, remember one, the blue FKBP and the disease target, which means that we can not only adjust the binding directly, the effector domain, the molecular structure, we can also change the linker between the 2 arts, alter the 3D orientation, so that the 2 proteins will come together at different angle, almost at the 360 degrees, and that will create tremendous diversity in terms of potency and selectivity. That's something that most other modalities couldn't accomplish.
Thank you.
Any other questions from the judges?
Hello, and what kind of animal study you have done, and how's the result?
Yeah, our lead program is for treating acute kidney injury. So we have demonstrated very significant efficacy in a standard rodent model. So that allows us to make a decision to launch that program into the clinic next year.
All right. Any further questions from the judges? Those will open the floor to everyone. Any questions? That's it, Sean. Thank you for presenting.
Of course, thank you very much.
Thank you. Next up will be D2M Therapeutics, Dr. Nanfing.
Yeah, so I will.
Yes. Nan will come on. Yeah, yes.
Hello, hi, nice to meet you. So this is Nan. I'm a CEO of D2M. I'm glad to have the opportunity to introduce our company to you. So, okay. So we are D2M. We call it data to medicine. So for innovative drug, we know that this is a high failure game, and most of drugs fail in clinical studies. So the reason is that the target usually selected by cell models or animal models, it does not translate into the efficacy in humans. So that's why most of the drugs failed in phase 2. And if we select the target using human genetics, that will increase the clinical success. There is a paper published in Nature a couple of weeks ago, and it mentioned that the target with human genetic evidence will increase the success rate for more than 2.6. Risk of probability. So our company is fully utilizing the genetic data to identify the targets that fully supported by human genetics, and we started our program from there. So that's what we do with E12. Here are our team. So I'm the CEO and the co-founder of this company. I was previously a senior director of human genetics in Pfizer, supporting immunology and information, target identification. So some of the examples including KL1A, which was successfully transferred to Roche for $7 billion. And Doc Zhang is my partner, and he was a director for immuno-oncology, developed the world's first anti-PD-1, PD-L1 antibody everyman. And also we have a team in business development, clinical, and operation in China. We have our advisors. Tony was SVP in AstraZeneca and R&D chair in CRISPR Therapeutics. Kai is a professor in Dana-Farber Cancer Institute. So we focus on autoimmune diseases and cancer. For autoimmune diseases, for almost all of the blockbusters, the target has human genetic evidence behind that. And for oncology, we think the germline genetics also affect the immune system about the risk of cancer, such as CTLA-4. What we are trying to do is that utilizing the same principle to identify a new target and build up our pipeline. And we have a platform fully utilize the human genetic data, and through the integration of those genetic information, we identify the disease causal genes, and we build up pipeline from there. We have pipelines on both oncology and autoimmune diseases. So today I will mostly focus the top two assets, one from oncology, the other from autoimmune diseases. And our first asset, through genetics, we found that the MICA and the MICB are highly associated with cervical cancer and endometrial cancer and beyond that. And then we learned that MICA and MICB are actually activating NKG2D on both NK and T cells. It has been shown that both NK cell and T cell are utilizing this mechanism to kill the cancer, which is MHC1 downregulated cancer. So those cancers are usually resistant to PD-1 treatment. So we are targeting a PD-1 resistant mechanism. So the cancer cell try to escape the attacking from NK and T by shedding MICA and B from the membrane. And our antibody is prevent the shedding of MICA and B from the cancer, so that the NK and T, we will restore the immune system attacking to the cancer. Actually, we are not the first company working on this. Cullinan showed their phase 1 dose escalation data last year in ASCO, and they found that their antibody particularly worked for endometrial and cervical cancer. That's the two cancer types. Using our human genetic data, we saw the same thing. So that's encouraging to us. That means what we predict through human genetics actually somehow validated by clinical data. And actually, we have a better antibody than Cullinan's in terms of affinity and preventing of shedding. And in the in vivo model, we have a better tumor inhibition than Cullinan's antibody. And in combination of PD-1, we see a single agent activity in syngeneic mouse model, and in combination for PD-1, we have a good synergetic effect. And in the SCID mice of a human tumor, and we see the antibody effect in multiple tumor types, including prevention of the metastasis of the cancer. And we are initiating our clinical studies. We think the clinical study can be driven by the biomarker. We can identify from human whether they have a soluble MICA and MICB elevation in the serum. And we have shown from the data, there are large proportion of cancer patients has elevated soluble MICA and MICB. And we also seen that NKG2D and PD-1, they are expressed in different clusters of T cells. So that's why our mechanism is complementary to anti-PD-1. And for tumors, we also see the PD-L1 expression is not related to MICA. That also provides the basis of why we are complementary. And we are initiating our class study. We already initiated. The first patient has been dosed. And we think that for monotherapy, we will concentrate on PD-1 resistant tumor. And then in combination with PD-1, we have a broader application. So that's our oncology program. Our autoimmune disease program concentrates on a set of diseases. Right now, the current therapy does not have an effect on those indications. That's including PBC, Sjogren syndrome, lupus, and systemic sclerosis. From genetic study, we found a set of genes within the same pathway associated with the same set of disease. Similarly, we have seen that for IL-23 pathway, for IBD psoriasis, and IL-4 pathway for asthma and atopic dermatitis, targeting those pathways leading to blockbusters. And we think we have the opportunity to develop the autoimmune blockbuster by targeting those pathways. We already have our antibody, extremely good binding affinity and good cell activity. And we also do FC half-life extension with very good half-life. I think in human it's more than 3 months half-life. And we also use the mouse surrogate antibody to demonstrate the in vivo efficacy in several autoimmune disease models. Right now, it's in skin inflammation model and the Sjogren syndrome model. We are doing lupus model right now. So we are targeting to 4 autoimmune diseases with very limited therapeutic option right now. So that's our potentially is a $12 billion market. And so that's where we are. So far we have raised $24 million. We are looking for a next round of financing to support the 2 programs I just mentioned. So we will try to complete the first one, the phase 1 study for our oncology program, and also the autoimmune disease program. And we also have additional programs, such as one program, COPD, I did not mention today, but we definitely have other programs in our early stage. So that's basically the background of our company. Thank you.
Thank you, Nicolas. Now we open the floor to questions from the judges. Charles, online, do you have any questions?
No, I don't have any questions.
I just want to ask a question about the validation. So for target validation, have you done anything about the proteomics or studying for the validation, or trying to screen off target as well?
You mean for specific target or in general?
Yeah, in general.
The validation?
Yeah.
So the validation, so we first identify the targets for human genetics.
Yeah.
So we want to make sure that there is a causal relationship between the target. The human modulation of this target has a disease impact. And from there, we will go back to the traditional drug discovery process, such as cellular validation, in vivo model validation. So for all of, at least for those 2 programs I mentioned today, we have gone through the whole process. So that means that for the target that has been fully validated from human to cell to animal, and then we come back to human in clinical stuff.
And also for the screening as well, right? For the off-target as
For
well.
off-target?
Yeah.
Yes, we also look at the off-target non-specific binding for antibody. So all of those programs, we are talking about antibody-based programs. Usually it's hard to find the target effect.
Yeah, a small question on 08008. So you mentioned this is first in class. Are there any other companies pursuing this target with similar MOA or with different modalities? So this is the first question. And the second question is through your platform, did you also find targets that are more responsive to RA or other kidney diseases? So these are 2 questions.
The first question, we are the only company working on that target. So under CDA, we can talk more. There could be some reasons that we think that this target has not been well noticed by others. And so that's the reason we think we have the blockbuster, because we are the first company working on this unmet disease area. So that's the potential we will have a blockbuster potential. And the second question on RA and kidney diseases, yes, we have another program that I did not mention today. That's also autoimmune disease target, and it's targeting Treg related mechanism. We think that mechanism is complementary to anti-TNF. And the Treg has been, people have been trying to identify an autoimmune disease-related Treg target for autoimmune diseases, and so far has not been successful. But that's very early. And potentially, we have seen genetics associated with RA and kidney. There are multiple autoimmune diseases. RA was there, kidney, lupus is there, and there are several indications. Potentially that may be related, but it's an early program.
Thank you.
We'll open the floor to the audience. Any questions?
And this is another question. This is all monoclonal antibody, right?
Right now, these three are all monoclonal.
Yeah. What is the CMC? So where get this down?
Yeah, we have an operation in China, and in China we mostly deal with CROs, CDMOs in China, and this CDMO is conducted in China.
So in the future, you guys are always chasing for the monoclonal?
Not necessarily. We for depending on the program, so we prefer to be monoclonal because as long as the target is correct and the antibody is there's no issues to generate a good antibody. Small molecule has advantages or adherence from the patient. Depending on specific target and depending on the mode of mechanism, we may consider small molecule as well.
Any questions from the floor?
So can DM919 be used as a first line of treatment option, or it has to be post PD-1 resistant?
So as a monotherapy, we will start from second line, third line as a PD-1 resistant patient, and actually our first patient has experienced the PD-1. And our competitor, most of the patients has experiencing PD-1, more than 60% of those patients. In the future, there could be a combination of PD-1 that could lead to the first line.
That's going to limit it on the market size, right?
So that's the reality. The reality is almost PD-1 will occupy all of the indications. And the unique thing that we are targeting PD-1 resistant, the mechanism is complementary to PD-1. So I think that's probably the broadest market size for oncology right now. And we are basically tumor agnostic. If we look at the soluble MICA, almost all of the cancer tumor patients has elevated soluble MICA. So potentially we may work for all indications. I think the market is not small.
So what's the investor excellence strategy?
As a small biotech, I think there are two strategies. The second is go IPO and very likely in Nasdaq. And the second definitely will seek the interest from multinational companies. Right now we are in talk with a few of them. Of course, they need to wait for more data.
So this DM919 would compete head-to-head with gamma delta?
DM919 for
DM919, which is targeting MIC-A, MIC-B as an NK strategy with tumor microenvironment, would it target head-to-head with a gamma delta therapy?
Gamma delta actually utilizes this mechanism to kill the tumor. So it's part of the mechanism. So
if you had a lava coming in with gamma delta binder, certain cancers that you are working on, would it be a head-to-head competition?
So yes, we can, but I think the gamma delta is just so small in terms of in vivo. There are so small proportion of gamma delta T cells, and gamma delta we utilize gamma delta T cell. Not only gamma delta, but also CD8 T cell, but also NK cell. It's utilized almost all of the immune killing mechanisms that are effective for tumor control. So this is very exciting. When we initiated the project, we were mostly thinking about NK. It turned out it's not only NK, it's also T cell. So that's very interesting.
I think there's a question from the end of the room.
I'm just wondering, is it an oral drug or?
It's intravenous.
Thanks.
What's the dosing frequency for 008?
008. we have not reached to the clinical. We have not completed the IND enabling stage, but the PK we use the FC modified FC to extend the half-life. We predict the human half-life is 3 months or more.
Questions? If not, we'll wrap up. Thank you very much, D2M. Thank you. Start point.
Great.
Okay.
Is your presentation on our computer?
Did you send through your presentation?
No, we didn't send the presentation.
Is that Sparks? I don't have the.
You need to get online if you are projecting from your computer.
Right.
Okay. Okay.
You need to join this.
How about we'll just let you set up with basically the next company first. You just set up, all right?
That's fine. Yep.
So maybe neuron off. Is neuron off here? I just want to use. Is it not here? F5 therapeutics. F5 therapeutics.
Yeah.
Azura Bio.
I guess the gentleman just left for bio break.
All right. Okay.
Thank you.
Leveraging, moving along very, very quickly. Leveraging. Right, maybe we'll take a 5-minute break to find the other contestants. If we take a 5-minute break, that's of course, it's not fine. Sure.
Yeah, be very quick.
Yeah.
Thank
Okay.
Ways to reach out to them.
So that's a bonus.
Yeah, they're probably just waiting.
Good to see you again. Oh, great. Yeah.
Where I can use the phone, right?这是1年之内。
如果去Austin Harbor.
That's meet you. Yeah, just send a LinkedIn connection. Yeah. Thank you. 我们看一下。
是。
But I'm here for the hands. Yeah, so you have time. Which is immediate, serious is the study. I did.
PKD.
I'll leave. So, colleague could here if you have time here tomorrow.
Email phone there.
Yeah, I send you an email.
Okay.
If you got a time.
Sure.
Thanks for your question.
We were just talking.
Thank you. Thank you so much.
Yeah, your should be.
No, I
online one is 10,000. So we're actually quite famous.
And then,
How many that's a good idea. Things like general, not just something called elevator, where it's whole building of 100 floors and stuff, and you have to pitch when you get around for the 100 floor. It's just elevator pitch. 1 minute.
1 minute. Correct. Wait, ladies and gentlemen, so we are ready to start off. All right, thank you.
Yeah. All right, thank you everyone for your patience. Apologize for the delay.
Just there's a 10-minute presentation, 9 minutes for Q&A. And there's a timer that will actually remind us.
Perfect. Thank you so much.
Okay, so whenever we are ready.
That's right.
Excuse me. I think we're ready to start.
Thank you, everyone. I apologize for the delay. My name is Luke Deal. This is my partner, Dr. Zeev Moses, and we're here representing Spark Spine. We're a medical device company that has come up with a first-of-its-kind biomaterial, and our first applications are within a $1 billion market segment of the orthopedic. Go to the next slide, please. Thank you very much. Oh, we can just go ahead to the next slide too. As I said, so we're going to be the first company to launch a piezoelectric biomaterial within orthopedics. And our products are going to facilitate faster, stronger, more reliable healing. And because we have experience in this particular $1 billion subsegment of the orthopedics market, we are planning on, we were able to get this to the FDA and through the FDA with a very short and efficient regulatory pathway. So I think that the pre-commercialization numbers here are going to be a lot lower than some of the other companies in this competition, but I hope that we have some similar upside. So here's our initial product design, and this is effectively our secret sauce. This material we call EMP. It's a biopolymer that we perform a special treatment to that gives it these piezoelectric properties. What that means is that when it's compressed or manipulated, it creates an electrical voltage that can be perceived by the cells around the implant. And effectively, we are mimicking the bioelectric signals that are produced by healthy bone cells under load that signal other bone cells to grow stronger and build the environment up to be more robust. So this is how it works. We perform a special treatment to this polymer that aligns the molecules, and then under pressure, the compression of the material basically realigns the molecules and creates a voltage across the material. Base material that we're using has been used in medical implants before, and the FDA was very familiar with it. Our treatment to that material is unique in medical devices, but because the FDA is familiar with this material from a toxicity and mechanical performance perspective, they've only required us to basically validate that our electrical properties are safe, which is the advantage that's given us this very efficient regulatory pathway. And again, this is basically just biomimicry. That's perfect. You can go to the next slide. So this is where our implant goes. This is a spinal fusion. I'll let Dr. Moses talk a little bit about this afterwards. But effectively, our product is a shim that goes in the anterior column of the spine. And it's all about growing bone. A successful surgery is when there's bridging bone that forms between this top vertebral body and the bottom vertebral body, locking that construct in place and providing room for these exiting nerve roots and the spinal canal to not be a pain generator. So this is some of the validation of our electrical responses. You can see here the response is extremely linear and predictable, and it's also very consistent. And through testing different sizes and different configurations of our implants, we now have the ability to design our implants to create a specific target voltage. Right now, there isn't a lot of data that suggests that there's a specific performance threshold that we need to hit. But once we discover that, we will be able to design implants specifically to hit that performance target. there's a tremendous amount of preclinical evidence on this, a lot of basic science. And effectively what we're doing is polarizing the ends of the ends of the implant. We're polarizing those metal end plates. And polarization, simple polarization, not dynamic polarization like we're creating. Simple polarization has already been shown to upregulate all of the bone formation processes local to that implant. Again, here, this is someone else's research on a very similar technology that never made it to market. But what we saw was that cells that are exposed to this fluctuating polarization form to be much more robust and much healthier and have much stronger adhesion than cells that were formed with just sort of a standard surface technology. This is a sheet model, and we're actually about to start a pilot study that's very similar to this. This is a different piezoelectric technology. This is barium titanate, which again is a piezoelectric material that this company was never able to get approved by the FDA. But you can see here the implants with polarized barium titanate with the same piezoelectric properties as ours performed much better in a sheet model than the regular titanium implants. Here's our team. So Dr. Moses is a neurosurgeon, assistant professor at Harvard, and operates at Beth Israel, right down the street. Yeah. He's the founder of the technology. Kevin and I ran a implant company in this same exact market segment and left that company 3 years ago to start a venture studio. But we were super excited to partner with Zeev because we think his technology is going to really revolutionize this space. And we felt like we knew it well enough to have a lot of quick success. Dr. Smith is an extremely busy private practice surgeon and one of our business partners. And then Dr. Bichara Ferrara and Dr. Ronsky, also a PhD. Together as a group, we've gotten over 150 medical devices cleared through this same regulatory pathway in the last decade. So we have a tremendous amount of experience, and we're locked in on our target. This is effectively what I just said, but some of the specifics, Kevin and I were able to take a business in the same market segment from no revenue up to $4 million a year as soon as the product launched. And we're going to follow that exact same playbook with this. So we're very confident that as soon as we achieve regulatory clearance, we'll have some significant commercial activity. This is some of the regulatory, this is actually an outdated slide. I'm sorry, I must have given you an old version. So we've completed several key sub-meetings with the FDA. Through this series of meetings, we've been able to agree on a regulatory path that both sides feel comfortable with and feel that it actively mitigates the risk around this device. So as I said before, basically they're requiring us to demonstrate that the electrical properties that we have that are unique do not pose a danger to the patient. We're not demonstrating that they have clinical benefits yet, although we do plan to at some point. Right now, we're just showing that they aren't dangerous, and that means that we only have to do a sheep study. We don't have to do any pre-commercial clinical work. So this is known as a special 510K pathway. This also might be updated. So this is called a special 510K pathway, which is basically substantial equivalency plus some supplemental animal research. These numbers are actually pretty accurate. So we're raising $1.5 million to cover the regulatory work. And we have outlined plans for a $1 million product launch. In total, we'd like to raise $3 million, and that'll take us all the way to profitability. So again, here are some of the specifics of our study. These have changed slightly since we built this. We were actually able to negotiate the FDA down from 3 months and 6 months down to 2 months and 4 months, which increased our costs slightly and allowed us to up the sample size, which is great. But again, we need $2.5 million. We're raising $3 million. We actually already have an LOI from a strategic partner that's offered to cover the cost of the product launch in exchange for some limited exclusive distribution rights. We're not sure whether we're going to take that or not. Really, we'd like to work with a private investor and maintain our flexibility because we do feel confident that we can grow and build this company here. We have 2 competitors. Both of them have much more complex implant designs than we do. And we see that as a massive advantage. They all use either complex circuitry, they use chargeable batteries, or they use lead-based piezoceramic wafers as their electrical generator. All of this put together means that they have a much different risk profile than we do because we're only using biopolymers that the FDA is familiar with. This is a carbon copy of the commercialization plan that we used last time we launched a product in this market segment. And it works really well. I've got a list of people that I'm excited to call who are excited to sell this product for me when it becomes available. Again, after running a business in this space, we know what the margins are like. It might not be pharmaceuticals, but it's still pretty healthy. So our average cost is going to be somewhere between $3,000 and $3,500 per implant. Our cost of goods is about $400. So there's plenty of margin in there. Typically, the sales channel takes around 40%, but there's still plenty of room for us to run a very profitable business. With our last company, we averaged between 20 and 25% net profit, even very early on in our timeline. We file patents all across orthopedics, and we really see this technology, or we see this material as a platform technology. And we're excited to grow it beyond the spine market as soon as possible.
The time is almost up, so you can finish off if you can.
Great. Our exit strategy, as we said, we've already got strategic interest in the form of an LOI. We would really like to run this company for at least long enough to validate our clinical performance and have a really attractive exit to a tier one acquirer like a Medtronic, Stryker, Globus Invasive. Something like that.
Okay, thank you very much.
Yeah, thank you.
Thank you. Now we move on to the Q&A session. So judges, any questions?
Your competitors, some of them in commercial stage, can you talk about how much revenue they have? And could you, maybe doctor, you can talk about the hospital needs. Is there is there clearly unmet needs when it comes to this?
Yeah, so I mean, as you all know, back pain is one of the most common visits to your doctor, right below a cough. And as with our aging population, a lot of people are having spine surgery done. It's one of the most common surgeries we do. And when we have to fuse the vertebrae together, there's still, a 5 to 10% chance that the bones don't heal well. And there are a lot of different products to try to address that unmet need. Some of them are things like BMP, bone morphogenic protein. That's very expensive, and it also has some side effects, and it's not on label for most spine surgeries. So, there's certainly a huge unmet need in this space. And, where we feel we have really an advantage is it's overall a pretty simple technology. From a preclinical standpoint, it's very safe. It's a thermoplastic that's used in other parts of the body. We're just treating it in a way so it has this piezoelectric effect, which mimics the normal healing of bone in general. So we see it as very synergistic with what the body's already trying to do. And then, sorry, what was your other?
Direct competitors.
Yeah, direct competitors. So it's a huge market where we're going to be competing with implants that already exist. All these predicate devices. The two competitors here, they're both still in the R&D phase. They're both going towards a PMA route because of part of it's their labeling claims. They're labeling their device as stimulating bone growth. And when you make those sorts of claims for marketing, you have to actually prove it in humans. We're sort of positioning ourselves as we're a surface technology mimic or analog, and the FDA is very familiar with surface technologies to help with osseointegration. Our plan is once we're FDA approved, we want to gather the clinical data to go back and make a stronger claim, but we would rather be more efficient in going to market and becoming profitable, rather than taking, an extra 5 years to gather all the data, to make those claims. The other thing is the other two competitors, ours is very straightforward. It looks like the stuff we already use in surgery. For one of theirs, they have to wear a brace to kind of power the implant. So there's a patient compliance issue with that. And then the other one uses some materials that contain lead, so it has to be hermetically sealed. So I think it's more, when you're talking with your patient and they look it up, there may be some questions about safety and if they feel comfortable with this device in them, and also whether it's MRI compatible. Those are things they're still working out. But neither of them are in patients yet. So I think from an FDA clearance timeline perspective, I think we'll be first by the time they're still, collecting their IDE data, investigational device, exemption, all of that stuff.
Okay.
So,
We probably have about a 4-year commercialization head start on our competitors.
Any questions from our judge online? Charles, please go ahead.
Yes, and this technology can only be used to spine or can be applied to other situations? That's first question. Second question is, you said you have 3 months and cash flow, and after FDA clearance, that will be insurance coverage or it will be self-paid?
Yeah. So because our device is being cleared through an existing regulatory pathway, it's all going to be reimbursed like other devices in the space, which is how we can predict the margin so accurately. Sorry.
Yeah, and in terms of spine versus other orthopedics, our expertise is more in spine, so we want to kind of build that blueprint to get this one approved. We do feel that this biomaterial has, relevance in, generally in orthopedics, areas where you need osseointegration, and there's a risk of, structural failure if the biology doesn't work out. And then we're also looking at areas like wound healing and tissue defects, those types of areas where this material and materials like it have been shown to improve growth of tissues.
Yeah. We just know the spine market better than the other markets. That's why we picked to do a spine device first.
Yeah. Okay. Thank you. So will you need a clinical trial for insurance coverage?
Nope, no clinical trials needed. We'll
Okay,
have
even for
we plan-
insurance?
Even for insurance, there are additional reimbursement codes that we plan to go after maybe 5 or 7 years from now, that would get us a $10,000 to $15,000 premium over our competitive products. And those will require clinical research and negotiations with insurance providers.
Yeah, when we schedule a surgery, when we get approved for the surgery, they approve us using, a spacer between the bones. But then the hospital and the surgeon can choose which spacer to place. So we don't have to get special for that.
Michael's question.
Yeah, a question, a follow-up on Charles' question. So, of course, it's on spine, but have you thought about using that merging with the titanium materials for the bone and also the other materials? And also, have you thought about using that on plastic surgery? Thank you.
Yeah, definitely. So, as Luke showed, we kind of went through it quickly, but we're actually using the latest titanium, 3D printed titanium, which is kind of state of the art in spine and inner body, but we're adding this as well. So, the titanium is what's actually touching the bone, and then this material sits in the middle to give it that polarization.
Yeah, and when Zeev mentions wound care, that would include some plastic surgery applications as well.
All right, thank you.
I want to circle back to the question concerning about predicates, because it sounds like you're very confident that you will win through the 510K regulatory pathway, which means you found the predicates already. So that predicates company, how much revenue they've made so far? I'm just wondering.
Yeah, so this product segment is a $1 billion market segment, and all of those products are cleared under similar 510Ks. So we'll just pick the, for example, Medtronic's product. Medtronic they're the market leader in this space, and they sell probably $250 million a year in this segment.
Okay, so you benchmark that market size with them.
Yeah, the $1 billion market size is from market reports. Yeah.
Okay, thanks.
I have a question. In terms of the application of it, does it has to be after the surgery and then the patient say there are a problem, or can you do it right there when you do the spinal fusion, prophylactically?
So I think that's what makes our biomaterial so powerful, is that it can be seamlessly integrated into an existing implant design. So the patient's receiving the benefits of this, but the doctor doesn't have to change their technique or do anything differently. So fit and form is exactly the same for our product versus all the other products that we'll compete with. We just add this additional electrically responsive property that's totally unprecedented.
Good question. So, do you foresee any challenges in FDA clearance, especially you don't have clinical study and with the piezoelectric implant, you are introducing electrical current into human body, right? So, do you foresee any challenges?
So we've had 3 very successful meetings with the FDA, and we've really agreed on all of the details of the regulatory clearance. So I don't foresee anything changing at this point, unless something unexpected happens with our sheep study, which is always a risk. But we're doing everything we can. We have ample sample size. We're doing a pilot program first. So at this point, we're really executing our business plan quickly, but also conservatively because we've been given this immense opportunity.
Any questions?
What do you expect out of the pilot program?
So the pilot study will be done by October of this year. And
what kind of results do you expect?
Just healthy sheep.
There's a lot of literature on fusion in ovine and the sheep model, and we're looking, to be quite similar to the already existing titanium products. And we're hoping to see an early signal that our interbody spacer with the material shows some earlier benefit than the existing control.
What kind of clinical benefits?
Earlier ingrowth of bone into the implant.
Yeah, faster healing and faster stability.
Okay.
Which for the patient would mean faster return to normal activity and a lower reoperation rate.
If you mention the faster, it's gonna stimulate the growth. They definitely need human clinical trials. I'm so confused why only need animal study to support the 510K. Because I was working at FDA. So that's why when you say that 510K pathway, I'm so curious why you're so confident we can get approval within 1 year.
Yeah, it's because we've agreed with the FDA that we're not going to market these bone growth stimulation properties.
Okay.
When
so
we do our
your use is
marketing,
different.
yeah, so we're substantially equivalent. Now we can describe the electrical properties. We can show lots of research and published research that demonstrates that these properties do have some stimulative properties also.
Okay.
But we can't say our implant stimulates bone growth.
Yeah.
Yet.
Yes. Just please correct that.
Of course.
Okay, I think on that note, we can wrap up because we have reached the end of the time for your company. So thank you very much for making it.
Thank you.
Company up is supposed to be Neuron off, but I don't see them online. So I could see F5. So F5 Therapeutics, are you ready?
Yes, I am. Apologies. I had this scheduled for this afternoon, but I'm available now. I did have team members who were supposed to join, but I'll take care of this myself. I'm ready to go.
Sure. Would you share your screen?
The other participants still sharing?
Yes. We'll unshare the earlier one.
Perfect.
Timer screen will come out and you can start when the timer screen is on. And the presentation is for 10 minutes and then 9 minutes Q&A.
Yep. Thank you. Hi, my name is Gary Choi. I'm a co-founder and CEO of F5 Therapeutics. We're a targeted protein degradation company based in San Diego, California. Thank you for everything to get us to the finals. We really appreciate it. So our mission statement really is to establish F5 as the discovery engine for big pharma, and it's really by expanding the scope of degradable targets that cause human disease. We could do this via an intimate knowledge of molecular glue chemistry and landscape. I have 2 ex-Celgene scientists that ran the chemistry and biology department at Celgene during the time of Revlimid and Pomalyst, 2 blockbuster drugs. One of the main reasons that BMS had taken out Celgene in an acquisition. We utilize a phenotypic screen. In essence, we're target agnostic. We create cell models that mimic human disease. We screen our library looking for hits, differentiated biology. When we get those, we go back and do proteomics to find out how we worked. And in that way, we've discovered 11 novel oncology targets that have great literature behind them. And under diligence, we could share those targets with you. But nobody's going out today and discovering novel oncology targets. And we have 11 of them with what we've accomplished so far. We have established proof of concept across 5 therapeutic areas in about 10 months with about 2.3 million. We're quite efficient with our money, with VC money especially. And we went for a platform play when we had our seed round, but now we are going into specifically degrader antibody conjugates. So talking about the DAC space, you could see from 2022 to 2023, just the increase in a very cold macro environment. Went from 23 billion in total deal value up to 86 billion in total deal value. And this is in the ADC space. I want to focus on degrader antibody conjugates over the last 7 months. And what you'll notice here is it's not only major companies working with targeted protein degradation companies, including this company that came out of stealth, but everything is very early. We are not late to the party in bringing degraders as payloads in ADCs. So you could see definitely good deal value. And again, very early. BMS took out a GSPT1 degrader from Aurum for about 100 million. They barely have phase 1 data. You could see the excitement around DACs. So why invest now? ADCs are one of the hottest spaces, as I just showed you. And why F5 Therapeutics? We have excellent glue expertise. We have this novel platform, a pipeline of payloads with novel degradation targets. Our lead asset is FFT135. It's a GSPT1/2 HBS1L degrader. It's beaten standard of care in 2 cell viability models, being HepG2, and an in vivo HepG2 mouse model. I'll share that in one slide. The timing of hitting our milestone 1 year from funding, we are asking for a $2 million C+ round. It will yield in vivo proof of concept, which is a key milestone not only for partnering, but also for further funding looking at Series A. Here's a look at our portfolio. You could see here, heavily focused on oncology. We do have degraders that can knock down pro-inflammatory cytokines with an oral molecule. And NASH is heating up again, or I should say MASH is heating up again with Madrigal's approval. We do stop type 1 collagen from a primary source, being human hepatic stellate cells. So again, these are at the lead ID stage. But today I want to focus on our payload library. These 4 molecules here, we do own everything. We don't have any licensing agreements with any academic institutions. This is all from our own library. So let's talk about ADCs. Excitement's quite high. I showed you some of the deal values. You start with a monoclonal antibody, has a high affinity for tumor antigens. So that way you're not affecting normal cells. And here you're looking to link a cytotoxic payload. And that generally is a topoisomerase inhibitor, a tubulin inhibitor. You're just taking basic chemotherapy. And as opposed to giving it systemically, you're trying to target it to the tumor directly. But there are issues, especially with safety. So what we're finding now is that the first ADC was approved in 2000, Mylotarg. And over the last 24 years, there's only been 15 approved ADCs. So while there's a lot of people focusing on this, and a lot of the investments going into bispecific antigens or looking at how potent you want your antibody to be, they're looking at novel linker technology to make sure that the payload doesn't cleave off prematurely and get on normal cells. So the safety issue is one thing they need to overcome. From an efficacy perspective, you need to beat chemotherapy. As we know, it's standard of care still in a number of solid tumors. And the risk benefit has not been there for the FDA. As I mentioned, again, only 15 approved over the last 20 years. So being a degrader company, we are heavily focused on replacing the cytotoxic payload with a glue. From an MOA perspective, we're not doing what chemo is, and a lot of time you'll have combinations. So we expect probability of increased complete response because there is a lot of tumor heterogeneity. We're going to widen that therapeutic index. Our molecules are targeted therapies. They are not cytotoxic. I'll show you evidence of that. So we want to reduce the myelosuppressive adverse events that we see, neutropenia, thrombocytopenia, the other penias that are involved with using ADCs. We'll have less off-target toxicity if the linker cleaves or does break off. In essence, we, again, are not cytotoxic. So that would lower the off-target toxicity. And we also target non-proliferating cells. So in essence, chemotherapy targets all cells, but they're going to basically go to tumor cells because they are replicating more rapidly. We expect less resistance, again, because we're not using what standard of care is right now. And we're not a PGP substrate, which is what chemotherapy is, and actually prevents the pooling of chemotherapy in tumor cells. And from a patient management perspective, when we do go to market, we do expect less training required, less ancillary medications, again, for these myelosuppressive adverse events. A look at our payload library going across. We'll get into FFT135 a little bit more, but 087 actually activates TME cells in the presence of TGF-beta, and we have single targets. We're only degrading one target here, and we're working to disrupt ER cell protein. Very similar mechanisms of action here. We'll stimulate TNF alpha locally in tumor cells, akin to a STING agonist, but doing it with a degrader. So the markets we're going after first are HCC and colorectal cancer. There are huge unmet needs here as we look at new cases and 5-year survival rates. If we look at specifically the target market that we're going after, we're going to use GPC3 and Claudin-6 in unresectable sorafenib pretreated patients, about 71,000 for HCC. In colorectal cancer, we're going to target the antigen CEACAM5 and 6, stage 3 and 4, second, third, and fourth line eligible. So in the G7, we're looking at around 110,000 total addressable market for patients. So again, this is a very significant market with blockbuster potential. Quick look at the data on 135. Again, this is the GSPT1/2 HBS1L degrader. We did a cell viability model. These are human liver cancer cells. You see here the standard of care FFT135, a nice sigmoidal curve, but it does kill all the cells. And you can see here a much more potent 135, a nice sigmoidal curve that does not kill all the cells. So you can see here from a 72-hour EC50, we have much higher fold potency than Nexivar. And then we took this into an in vivo model. And here over 17 days, we grew HepG2 cells on the back of mice, hit them with a low and high dose orally with a 135. And you can see here that we return the tumor back to normal. Nexivar does have some tumor suppression, and vehicle and another IMiD comparator had no effect on solid tumors, which is what you'd expect. So we've beaten standard of care here in 2 different viability, cell viability, as well as in vivo. And how we're able to do this, we have a very, very well-seasoned team. I mentioned my name's Gary Choi. I've been in industry 30 years. Spent about half my career, 8 years at Roche, 8 years at Janssen, but had 4 years at AbbVie. Worked for some small biotechs up in Cambridge, Idenix, you may have heard of them. Came out to San Diego, was a head commercial person at Samumed and the chief commercial at Kalidi, which is an oncolytic virus company. Number of brands represented here. Really, John Boylan and Neil Rahaja, their work at Celgene on Rev and Palm is critical here, as these are the most clinically and commercially validated glues that are on the market today. We've won a number of awards. This is the 3rd time I'm presenting this group. We did join JLabs as a virtual residency. I want to show you what's really important. I didn't show this last time, is 2 paths forward for DAC development. We can get an R&D collaboration with an antibody company. We're talking with 5 right now that are very interested in our payloads, and 2, we're under diligence with 2 of them right now. Or we can get C+ funding, which we're looking for as well. These are parallel paths where we could use a commercially available antibody via the CDMO that we're going to utilize. From there, they can make a licensing decision. With the antibody company can do that after we've gotten in vivo POC, and then expand that out for additional payloads. Or we can get POC on our own and then go back and look again to partner with an antibody company, which leads into our Series A with a number of different data points. I know I'm going quite quickly here. I'm willing to share this if you reach out to me. So I apologize for going so quickly. In the 1 year, we're going to scale up compounds, make sure that the degrader and linker are still functional, that we still have activity, and then go right into a tumor cell panel. So by Q3, we'll be scaling up 2 antibodies, and by Q4, we'll have an antibody degrader with in vivo efficacy. In the following 6 months, again, with C+ money, we'll have, with and without standard of care, we'll have tumor PD, and we'll have our proteomics and cell panel screen. And this is a huge inflection point for us for Series A, as well as for partnering efforts.
Thank you, Gary. I think it's time up now, so I think.
Oh, thank you.
Okay?
Thank you.
Right. So now we
Yes,
move on
absolutely.
to our presentation. Right.
Yeah, I'll leave this slide up, but I'll start taking questions.
Yeah, Gary, 2 questions here. One is on your slide 10. You mentioned your FFT135, the efficacy curve. So just a bit curious about why It is seems that it is not dose dependent. If you look at the 25 and 75.
We're overdosing. I think the doses are too high right now. What's fun about the ADC is we can go with very small doses, which will again lower the systemic adverse events. So we're quite excited about 135, the potency of it. So yeah, we only had 2 million from our seed round from Viva Bioinnovators in China, and that only got us to one in vivo study. So we crammed a bunch of molecules in there. We obviously want to repeat that at lower doses.
Okay. And you mentioned that the drug is orally taken? That's
Correct.
Oral? Okay.
We are
What's the
molecular
molecule?
glues. Yes, sir.
What's the molecular weight?
Molecular weight, we're all rule of 5 compliant. So from a C log P polar surface area, we are small molecule for sure. We probably will cross the blood-brain barrier. We want to look at glioblastoma down the road as we grow. I could give you specifics on 135 under diligence. I'll give you the exact solubility and everything. But again, they're all rule of 5 compliant, all of our molecules in the library.
Sure.
Thank you.
Thank you.
Yeah, Gary, a question for the DAC. I think it's not very close to ADC. It's probably closer to molecular glue or PROTAC. For your product FFT 135, have you done anything with the molecular glue comparison for your target GSPT1, if I remember correctly? Have you tried to make the comparison? Thank you.
Yeah, so GSPT1 is actually a novel target. There's a number of molecules from BMS. I mentioned Aurum. There's a few people working in it. And what they found is it's has very good anti-tumor properties, but it is also toxic. So they're finding some adverse events with GSPT1. We see here, Aurum was very smart going the DAC route, but we are a GSPT1/2 and HBS1L degrader. So again, as I go back to the phenotypic screen, they've targeted one neosubstrate being GSPT1. What we did in our cell model on the phenotypic screen, it may not be just GSPT1. It may require Icaros, Iolos, other degrader targets to maximize. And again, putting it in a DAC will reduce some of the safety concerns because you're delivering it directly to the tumor. So we haven't done any head-to-head comparisons because we're not just a GSPT1 inhibitor. We actually have multiple substrates degraded with 135. I hope that answered your question.
Thank you.
Any other questions from the judges?
So how is your off-target?
Excuse me? Off-target.
How are they off-target?
Oh, yeah. We have to explore off-target effects. We did proteomics at 8 hours, which is direct degradation. We want to do repeat at 24 and 72 hours. We are also upregulating some substrates as well, some proteins, which could be used as biomarkers. So we do have a full proteomics. And again, in 10 minutes, I couldn't have shown you that. But right now, direct degradation, we're very certain at 8 hours when you do, it's about 8,000 proteins in our screen, in our panel. So we haven't seen any other off targets from what we know.
All right. Thank you.
Thank you.
Any other questions from our judges? If not, I open to all the audience questions. If there's none online.
Can you talk to us about the Capital Therapeutics GST1, GST2, Next 7, and how it compares to your GSPT1 degrader?
Yeah, we haven't actually done any head-to-head. Again, with the 2 million that we had, we went platform first, but that would be something that we want to look at down the road, but we haven't done those experiments. I think from a novelty perspective, a lot of the GSPT1 companies outside of Aurum are pursuing them as single agent molecules. We feel in a DAC, not only, we could explore it in certain tumor types, we can expand out depending on the antigen that we're targeting. So a lot of the antibody companies that we're speaking to today have different antibodies that target different tumor types. And 135, we've tested it in Hep G2 as well as HT55. So that's why we have the colorectal cancer up there as well as a second indication that we'll be pursuing.
I have a question. So you're seeking $2 million seed. That will last 18 months?
Yeah, it'll get us through the experiments that you see. Obviously, we've been in business since 2020. The founders, we haven't paid ourselves anything. We put in all of our money into the science. So here for a Seed Plus, the feedback that we received from VCs as well as partners, it would be great if we could show that we could put a handle on our glues and not affect cerebellum binding and not affect what we're degrading. And then we can attach it to an antibody to get in vivo POC. So very confident in that 1-year timeline in getting the in vivo POC. The next 6 months would just be a continuation, adding it to standard of care and getting the tumor PD and proteomics. So that would last us through the 18 months.
So I think time is money, right?
Correct.
From what I know, talking to many investors, so you need addressing currently if they give you money, then their dilution, so many factors to consider. Then take it almost, let's say if anything delay, will take 2 years to reach to next level, Series A. You're looking for 40 million. I like your technology. I think it's very innovative. Maybe rethinking about the size of the seed, what another inflection point to reach, I think.
Yeah, if you recall our portfolio, we have FFT012, which is a Wnt pathway, which again has not been cracked in cancer. I know I have 2 minutes here, but specifically with some of the upfront money we're going to develop that candidate as well. So yes, DACs are very critical for us. And I think once we partner with a company, we could then expand the partnership out. We didn't focus on DACs when we founded the company. The molecules that we have in our portfolio today are all applicable as payloads, and it's very easy to expand that library out. And then again, through our phenotypic screening, we could screen specific cancers and targets of our partner. So not only do we have this Wnt pathway, which targets TCF4 and the nucleus, but we have this other ongoing expansion of the library as well, which will be part of that Series A. So now we're not just working on these 4, that's what we have today, but a lot of interest in 135 from the companies I've spoken to. But we have the ability to expand out our library and portfolio quite quickly. So even within that 2 million, while it's going to be focused on moving 1 DAC forward, and again, non-dilutive money, but yes, that asset will be, we'll say the value of that asset will be reduced, but the number of assets we have in the pipeline would clearly make up for the value as we go for evaluation for a Series A. So we're not concerned about that at all.
Thank you. Right. And
Thank
last
you.
question, quick one before we wrap up.
Yeah, so for the link for DAC, the linker you develop by yourself or you purchase from somewhere else? For $2
Yeah, the
million
linker technology will come from the CDMO or CRO. They have the great part about antibody development today or ADC development today is they are armed with a number. It's almost like a menu. Any antigen I choose or any tumor type, they'll have an antibody available that's commercially available for testing, as well as linker technology. So yes, that will be provided. We are only providing the payload. And the AB companies we're talking to today also have the linker technology for combination.
Thank you, Gary. I think we'll wrap up here. So, yep.
Thank you all. Appreciate your time.
And next up is Azura. So we have David, who is here with us, as well as John, who's online.
Yes, thank you very much. I'm David Staska. I'm Chief Medical Officer and founder of the Azura Bio. Azura, we're developing a unique, absorbable, soft tissue reconstructive solution. And this is in response to problems that you may be aware of with polypropylene and permanent mesh materials. Our lead indicator is stress urinary incontinence. I'll show you our leadership and our board, and you'll understand why. Additional potential indications include hernia, orthopedic and breast reconstruction, but we're not going to mix that all up with what our primary laser focus is, which is bringing this product to market. We have 2 years of highly positive animal and human clinical data
demonstrating efficacy without the problems, meaning we've already run patients with this, okay, and shown that a long-term absorbable material works. We have switched out the materials because during the process, we found what we thought was a better material that we can actually use in combination. And I'll show you our intellectual property. So we're going to redo what we have already done so that we have the tissue labs, we have the animal facilities, we have the regulatory people, we have everything keyed up, and again, we have experience with it. We're seeking $5 million in a Series A investment to complete our preclinical studies. And I will show you that will get us to the point where we can start our clinical studies, and that will bring in additional money. Next. This is the group leadership team. It's hard to see from the seats here. John Nealon, who would have liked to have been here today, but had back surgery, but he is online. He's the CEO. Myself, Sue Watte, who ran clinical regulatory for American Medical Systems and since has taken a lot of products through the FDA. I can tell you she is my hero when we're on the phone with the FDA. John Paul Ruvers, who's the Chief Scientific Officer, known as Europe's absorbable materials in female pelvic surgery, both for his surgical abilities and also for the research he's done with Celia Guler, who's here, and Kevin Arnell, who was the vice president of R&D for, again, American Medical Systems, and has taken these products through. Our advisors, the top advisors in the United States in urogynecology, Pam Mawali, who's known for tissue research. We're very fortunate to get her on board. Peter Rosenblatt, who's local here, but who's nationally and internationally famous. Avi Winkler, who again has worked with absorbable materials. And Catherine Matthews. So we've really put together a great team, both for advising. These are the KOLs, and so we're prepared when we finish our clinical study to be able to utilize these people to support the product. Next. Huge addressable market, 30% of women cough, laugh, sneeze, leak. That's stress incontinence. Okay, and it's from anatomic support. It's in the diagram on the right, the bladder's in right. The sling is placed in order to re-support the anatomy on the mid-urethra and the upper part of the vagina. It's put in through 2 small incisions on the abdomen and a small incision in the vagina. Needles are passed and the sling is pulled into place. I patented this procedure from above in 2001. Again, we are extremely familiar with the surgical procedure. Only 4% of women get slings. In 2011, there was an FDA warning about mesh for prolapse, not for slings, but it dropped the market for women having the surgery by 40%, both because the women were concerned and fearful, and the doctors really didn't want to spend the time to start implanting and trying to convince somebody that they have a surgery, even though it's still considered the gold standard operation for fixing these patients. Next. So long-term absorbable resorbs over time, so we have the efficacy without the complications. PBS, polybutyl succinate, is what our core base material is. It has an existing 510K, so the FDA was very happy to see it again, and no problems with that. They do want a study because it's going to be used in the vagina. We welcome that study. We want the clinical data to be able to launch the practice. And we'd like to see other companies that want to go ahead and follow us, because there is no one developing an absorbable material like this, have to go through the regulatory pathway. So we're pleased to see that. We want to integrate PLGA, which is a rapidly absorbed protomer polymer within that, in the zones. So there's intellectual property there. It's embroidered in, it dissolves in a few weeks, it brings in the macrophages that see the foreign material, it then brings in fibroblasts, and it recreates functional tissue, which re-supports the woman's anatomy and stops the leakage. Next. This is the functional tissue ingrowth thing. That little square there, this is the urethra head-on and the sling that's been put into place, and the zones that are part of the patent are here, just so that you understand what we've patented. Next. Patient reaction and surgeon reaction, incredibly positive. 97% of the doctors who were polled in an independent research poll, not by the company, said this would change the way that I practice. And in fact, the permanent mesh sling, they say there would be about a 35% initial acceptance when they present it now in their office. They anticipated an 83% acceptance of an absorbable material. These doctors also said that they believe within 1 year of the product launch, 50% of the surgeries they do would involve this material based on the clinical study and the early clinical experience. So physicians who do this type of surgery are ready to switch. Next. This is just some quick numbers showing the market size. The real numbers here really are, if you look at the US market, 250,000 surgeries a year, we believe that will double. The price right now for putting in one of these slings for the actual kit with the needles and everything else is probably around $1,000. We'll get a premium price. There's no question it could be higher than this. Ex-US, and I heard a wonderful talk about ex-US this morning, and the ex-US market is absolutely incredible. And development further of this type of technology for hernia brings those numbers even higher. So you can see that. And we feel very, very confident that we will double the market, and I'll show you some of those numbers. Next. We have 2-year data, again, on a sister material. This lasts longer than that sister material. This new PBS shows better fibroblast ingrowth than the sister material. The human pilot confirmed safety and efficacy in the sister material. So we'll need to redo some of that. Again, we have our animal facility in South Africa where that was done. We have the 20 patients who we have already done, which showed incredibly promising clinical data. And people have asked questions today about 510K pathway. The FDA is very happy to see polypropylene go away because these were approved products that caused a major problem in pelvic mesh, not in slings. They only asked for a 120 patient clinical study. We were so happy, no control. Okay, so they were very, very happy to see that if we follow these patients for 1 year, that's approval. Okay, now that is a 50% improvement in at least 50% of the patients who are operated on. Our original study showed 70% cured with a long-term absorbable. So our hurdle is 50% of people, 50% approved. We already know that 70% of our patients were 100% improved. Had no leakage with activity using, again, the sister material. We believe that we have something superior to that. Next. This is the timeline. Of course, this will move back or it won't move forward. They always move back a little. But with funding, we're talking about $5 million for preclinical. We're talking about $10 million for the clinical study, which we already understand and are able to price based on current research costs for this. At the preclinical close, we have multiple companies that are interested in this. None of the strategics that are in this space are developing an absorbable material. They have all said to us, show us that the FDA will approve a clinical study. We funded too many companies that told us that it would only take 6 months to get there. So you guys go ahead and get there, and then we will support you for the clinical study. And then again, exit would be sometime in 2027. Next. This is the projections that we have for sales. The interesting thing is that this was made incredibly conservatively by us on purpose. It shows a 5% of the market in the first year of launch, a 10% of the market in the second year of launch, and 25% in the third year, where doctors said 50% of their surgeries, okay, would be done with this method. So I consider this extremely conservative. It does show $100 million worth of sales after the first 3 years. Next. Again, there are other areas. We are able, based on funding and what our focus is to determine whether we're going to pursue any of these other indications
The time
prior
is
to
up, so I think
finishing.
it's time to wrap up on this slide.
Okay.
Sure.
Next.
Right. If that's okay, we'll.
Yes, this is it. Exit strategy. Strategics interested. Those strategics again are listed on the right side. We've discussed this with, or we can go to an IPO path at launch. Thank you.
Thank you, David.
Yes.
So now we'll proceed to Q&A. Questions from the judges.
Yeah, a question. So the sling is supposed to be permanently implanted in the body, or there's a time you have to change it after a while?
No, once the surgery is done, the tissue grows in. There are excellent results on permanent slings. But what we suspect is that with the tissue ingrowth here, that this will behave like a permanent sling.
Yeah. So in terms of the quality of life improvement, is there any quantitative way to do that? Because it's improvement for the life, for the behavior. Are there any quantitative ways to do that?
Well, I mean, there certainly is, but what it's really based on is a pretty objective measure. If somebody has stress incontinence, which means urinary loss with effort, they're dry, and they know it because they know that they were wet before. So the outcome of continence. Now, if you say 50% of people, 50% improved, as a surgeon, I would not even consider that a valid metric. I'm very happy that the FDA is happy enough to see that a device works and helps, but our goal is really continence.
Thank you.
Have you seen recently any non-surgical way of treating incontinence?
I mean, there have been ways of trying to treat the urethra in order to either bulk up tissue or modify tissue in some way in order to get better closure pressures. But in a disease that's really determined by anatomy, just bulking really doesn't have as good an outcome or as long an outcome.
Hey, David.
Yes.
Yeah, so the one thing I would add to that is that the non-surgical treatments generally don't involve the physician. They don't get good clinical data, and they generally don't have good outcomes, right? A lot of those things, there were some, through your experience, there's been a number of products that have come along that had external devices and just never really worked well. So I think that, right, so I think that whole issue of the non-surgical ones generally haven't worked, and that's why the sling has been a great procedure. It's just the material isn't the right material, and that's where our solution will help move forward and bring more patients into the treatment continuum.
Any other questions from the judges? If not, we'll open the floor. The audience, any questions? And online, any questions? You don't see any questions, I think, yeah, that concludes.
What's the estimated profit margin for this type of?
Well, John, do you want to answer that?
Yeah, I think we're gonna price the product at $1,600. We have good data from the sling market of how much can be handled. The hospital reimbursement is $4,600 for a sling. So we're going to charge $1,600, and our cost will be about $245. It'll be about $245. So the margins will be up in the high 80s and eventually get into the low 90s.
The other thing about reimbursement is that this is a procedure that doctors already do. So in the actual kit that includes our mesh sling from a sister company, all we will be doing is replacing that with an absorbable. So there's no training, no new training, no new CPT code. The doctors know how to do it. They're very, very comfortable with it. So it really doesn't have that idea of trying to convince anybody that there's something new other than what they're actually putting in.
And I think, David, just to add to that, that's why our physician research, that independent research we did, showed that those physicians said if you had a different material, we'd switch over right away. And the switch would be driven by patients, because as he said, the physician said the patients will accept an absorbable material over a permanent material. They just don't have one, and we're going to be the first one to develop it.
So are you currently talking to any strategy?
John, as far as the companies that we've talked to that we're showing there?
Yeah, I mean, you look at the whole list, it's Boston Scientific, it's Coloplast, J&J, Medtronic, Hologic, Laborie. They're all Organon, which is a company that was spun out of Merck to focus on women's health, Cooper Surgical, Teleflex. They're all interested in this technology as a way to build to the future, right? There's a lot of money being spent in treating women's health right now in overactive bladder, which is a sister, it's women have urinary incontinence. They either have the bladder issue where it's overactive, or they have the stress incontinence, which means they're leaking under activity. And so what's exciting is there's a lot of money being spent in this space, and companies like Boston just bought Axonics, right? So you saw they spent $3.4 billion to buy Axonics, which was over 100 million in revenue. We're projecting to be over 100 million revenue at year 3. So we have a huge opportunity here. We just need to get through this clinic, the preclinical stage. We need to get that $5 million, lock that down, run the $10 million study. And the study likely will be funded by a strategic with a likely exit built into that, which would likely have a predetermined price that'll be based on market numbers that we can all sit down and agree around the table on.
One question. Is your patent, your IP on the tissues, the polymer, and also technique?
The patent is on the ability to create zones where you have increased tissue ingrowth.
Oh, so the technique as well,
Yes.
not just the polymer. Okay.
Yes.
We have a question from the online judge. Charles, please go ahead.
Yes, and David, can you explain more about how the sling can help the growth of the scaffold?
Yeah. Do
you want to go to that slide? Do you want to go to the slide?
Can you go back to the?
No, David, go to the slide that we have in the appendix.
Oh, go ahead. Go forward real quickly then.
I think that'll answer Charles' question.
Next.
Keep going.
Keep going. Well, I mean, this is basically when you place the sling in the anatomic position that we've shown. Two things happen. The body recognizes it as foreign. Macrophages go in. Fibroblasts follow the macrophages and lay down tissue. So with the rapidly absorbing one, we're attracting this early into a mesh framework, which will stay there for about 9 months to a year. And this area on either side of the urethra to the back part of the bone will reestablish what are known as the pubourethral ligaments, which are the natural way to support things. So we know the surgery works. Okay, we know the surgery works if you make an abdominal incision and suture it up. We know the surgery works if you do it through the vagina, but we can place this very efficiently with something that will create an excellent tissue.
Okay.
And I think there's a lot of good science that we have this scientific references there that support this, and that research that was done at the Ruvers lab in Amsterdam.
Your current addressable market is what, 300, 400 million?
Current addressable market?
Yeah, so there's a slide in there. Go back a slide. The current addressable market, there's about, if you look at the sling market today, there's 300,000 sling surgeries today, right? And what we're saying is we're going to do two things that are going to happen. One is we're going to increase the conversion rate of patients that come in to get surgery, because they're going to find a surgery that they're going to like. When they're told polypropylene mesh, the patients, they've heard so much negativity, they just back off. And so if they hear, hey, you can get an absorbable material, it's not the same material, it's not polypropylene, it will resorb and go away. That's going to increase the physician conversion rate over the next 5 years after the launch. And then the other thing is we're going to be able to get, we're done now? Okay. Okay.
Yeah, thank you for your question.
So anyways, that's the market. Yeah. Thanks. Bye-bye.
Our last presenter today is Leveragent, and they'll be presenting in person.
Thank you, everyone. Thanks for BCIC for the invitation. Very honored to be presenting at last, company here. So the company is Leveraging. I'm the founder and CEO. We started a company about 6 years ago. We're based in Boston. So we are trying to address this very large therapeutic antibody market, which is expected to be $300 billion in 2025. And the current way to generate antibodies, the therapeutic antibodies, the main method is basically different type of genetic animals. So you can see several very successful companies, the early ones like Madarax and Apgenix. They both were acquired by larger companies later. And Regeneron, of course, generate their own tool, and then they become from a small biotech to a pharma giant today. So this progress you can see from Regeneron, they spend really 10 years time, like 9 rounds of modification of one allele and then 8 rounds of another allele. Cross together is 10 years of work. So from there, they really transform the company from a small entity to a large pharmaceutical company. So a more recent example is that Teneo Bio. The Teneo Bio has its 2 models. So one is UniRat, the other is OmniRat. So one is for the single domain antibody and the other for common light chain. So then they didn't license the model, they just do internal development. So they generate 10 different assets, mostly bispecifics and some of the CAR-Ts. So, and from there, they license different programs to larger companies. So one of the first on Teneo One licensed to AbbVie, that is upfront $90 million, and the complete purchase is over $400 million. And then, the whole set, except for 3 smaller companies. So basically, majority of those assets was acquired by Amgen in 2021 for $2.5 billion, including their platforms. So what we want to do is that we came in late, we hope to do it better. So we founded the company in late 2017, and we are working on genetic engineering, so basically generating different type models. So including the single domain antibody model and common light chain model, and also regular antibody models, just like Regeneron, but we hope to have a higher diversity. And this is the current team. So we have mostly antibody engineering team on the top, and then also genetic engineering team on the bottom. So these are our advisors. So there are famous scientists. I believe many people know Tom Maniatis, who's my mentor doing a postdoc later in his company. And he obviously is one of the pioneers of modern molecular biology. And currently he's leading the Columbia effort for precision medicine, and also founder CEO of New York Genome Center. And he co-founded many biotech companies, starting the very first one in Boston as a genetic institute, and the more recent Calliope, that was company I work with. And Charles Zucker is a famous neuroscientist studying pigs. So he also co-founder of many companies. And Zhijian Liu is a seasoned drug developer. More recently, he was a head of biologics in Novartis Shanghai. And today, I want mostly present this leading model we have now. That's called singularity sequence. So singularity, we named this way, just like the start of the universe, the Big Bang. That is a single domain, single chain, single class of antibodies. But from this very little thing, you can generate a universe of antibodies. So this is how it looked like. So a small antibody is like 15 kilodalton, comparing to the very giant, like conventional antibody, that's 10 times the size, like 150 kilodalton. But this small size is sufficient to deliver very similar affinity and also many properties. So I will elaborate later. So the advantage of single antibody over the conventional antibodies are several. One is that they are modular, just like Legos, right? They're small and modular, and they can be turned into many complicated antibodies like multispecific, multivalent, And also because it's such short sequence, easily engineered into CAR-T, ADC, and mRNA and AAVs. So engaging more of the modern therapies. And then also because they're very small, they can tackle the challenging targets like GPCRs. The deep grooves are very hard to get in with the regular antibodies. With antibodies, a lot easier. Then, for tissue penetration, like for like solid tumor, including brain. Brain is the blood-brain barrier, right? The tight junction very hard to go through. And some of the antibody can be engineered that way, some antibodies. And also, finally, this, intracellular targets, which is missed by the current immunotherapy because it's very hard to get into the cell. So you can engineer antibodies way so that they can have a cell penetrating peptide and already deliver siRNA. So the current predominant way to make single domain antibodies are still using there's a llama, alpaca, the camelid animals. They have a natural mutation, mutate out the CH1, which is a critical link to the light chain. So without that, you generate this heavy chain antibody in the animal. And then from the tip of that, that's this famous VHH antibody, single domain antibody. And so Amlinks is an absolute leader in this field. So they were acquired by Sanofi for $4.8 billion a couple years ago. So they use a traditional way, basically immunize llama, and you take the blood and generate B cell libraries, and you screen for your binder. So there are several issues here. So one is that it's a very long process. You immunize for several months, you compare to mice, and also you need to modify sequence later on. And second, the immune response is not only the heavy chain response, it also includes this regular antibody because they have all the traditional antibody, IgA, IgD, IgE, IgA, and all the IgGs. So actually, non-antibody part, basically the heavy chain part is only sometimes less than 10% of total serum, like IgGs. So that would be an issue, like you cannot gauge, because of computation of other antibodies. You may not derive very high affinity antibodies from there. And also, most important, they are not human. So you need to humanize it afterwards, so which will require additional engineering. So the approach we took is that we want to go to mice. It's a lot easier to engineer animals in mice, models in mice. So we started with wild type animal. So we want to first mimic the llama logic. So basically mutate out the CH1 domain so that now we can decouple the light chain, so generating heavy chain antibody. And two, we want to solve the llama problem, which is a co-production of all the regular IGs. So we eliminate everything, basically leaving only one heavy chain, that is IgG1. So that's in part we call it singularity, not a single class, a single chain, and then a single domain. So the next step is a theoretically easy but practically very difficult process. That's why Regeneron will take 10 years to do it. So it's to replace step by step the mouse sequence, the VGG sequence, with human sequences. So because the reason is that it's very long. This whole piece is 2.5 minute basis. There's not a single vector can include all that piece. And then also, we started by engineering constant region on the right side. You can see the big red dots are actually enhancers, very critical for this translation. And then we did a 3-step modification, removing everything else, only have the CH1 truncated, so CH1 truncated IgG1 exons in there. So then from there, we did a engineering process to get rid of all the VDJs. So generally that leaves called singularity hyperdoc. So from the doc, we have our patent technology to engineer the fragments step by step. Each step is like 200 kb. It's very, very difficult. So in the end, we reconstituted human VDJ region into mouse allele. So that is best, for the regulation. So here is evidence that we actually accomplished the whole thing. So this entire, reconstituted human genes, there's 48 different regions. There's the maximum number you can ever get. And so this evidence that actually works out. So you can see that on the top, you can see the wild type mouse has all different IGs, where mutant mouse only have IgG1 that is truncated. And then in the serum also, you can see there's no IgM and no the other IgG, and you only have a truncated IgG1. And this replacement process works out. And then also more important, you can look at the level, right? So in serum, in wild type, you can see there are other type of IGs. And the mutants are only one, IgG1, truncated IgG1 expressed in a very high level. So, and then we also carefully examined the B cell development process, and we found everything looked normal, very comparable to the wild type mouse. And then, most importantly, we found that their immune response is very robust, highly comparable to the wild type. So from there, we go for different ways to generate antibodies. Just one special way we can use, and the other.
About time to wrap up. About time. The time is up.
All right.
So you can finish your last slide.
Okay. All right. So yeah. All right. So let me quickly go through. So basically, we're using very fast, like sequence-driven process, to and then we further characterize the antibodies. So the antibodies, they are soluble, stable, and they're all different affinity. Other testing are all great compared to the green dot that's a control antibody. And then we can also generate the bispecific multispecifics. And then basically we have a model that's the current leading model for single domain antibodies, and with different features. And then we are currently collaborating with multiple companies, including Moderna, Invaluable, the first Invaluable, and also other like the reagent company, and also like AI companies.
I think we'll do a hard stop now if it's okay.
Okay.
Yeah.
Yeah.
Right.
Right.
So we'll proceed with the Q&A. Any questions from the judges?
The program is really promising, just a lot of pipelines. So have you thought about using some of the collaborations, license out some programs, some other companies to share the single domain?
Yes. So sorry, I didn't get a chance to go through. So basically, we are collaborating with multiple companies. So one way to do is to do technology licensing and also co-development. So they're all very recent because we just got to the market since end of last year. So the last 6 months of work, we already collaborated with mRNA companies and bispecific companies, and also CAR-T companies. So that's the main use of it, and also including other parts. Also, we could, since we have this one sequence, one antibody feature, so we're working with AI companies like Ginkgo Biowork, for this ability to generate better antibodies from our.
So you said you started the process late last year, and you are in the process to be
Yeah,
licensed?
they already signed.
They already signed.
So we're working with them. So it basically took 4 years to make mice, and then we moved to market since last year. So one way is to license, and the other is that we will have an internal development, and we have several programs going. One is a bispecific antibody for T cell engager, NK cell engager, and also for targeted delivery using RNPs, it's protein antibody. And more recently, we started a program that we want to target the brain. So that's one of the main features of the nanobody. It can go into deep tissue, including blood-brain barrier. So that's where we are working on.
Can you talk about current financing?
Current financing.
Financing round.
So we are still seed stage. So we received $1 million investment for developing the model. So right now we are generating revenue from collaboration, but we hope to raise funding for internal programs. Some of those are like, we don't want to use hard make money to drive these programs, right? So like I say, those three are innovative programs and hope to engage some investors on that.
Okay. You mentioned that you are already generating revenue from your collaborations.
Yes.
So do you have any more sort of partners in the pipeline that you potentially can increase?
Yeah, since we just started, I think there many are in the process. So as a regular licensing deal, they have upfront payment, like platform access, and then we have this revenue, but the party have milestones, so the royalties.
How big is your team right now?
The team, there are 12 people.
And you're based in?
Based in Woodland, not too far away from here. Boston. We're here.
Okay. Right.
Hong Kong.
Yeah, so. All right. Okay. Yes, we've been to Woburn. Yes, sir. We were there yesterday for the record. Any more questions? Or Leveragent?
So can you talk more about the blood-brain barrier part? What can you talk about which targets you're using to generate antibodies? These are traditional transferrin, CD98, or do you have something else?
Yeah, thank you for asking the question. I didn't have time to elaborate. So this collaboration actually, so I have a master's by training. My postdoc was on neuroscience. So the lab studied neural wiring and also ARS. So of course, we also affiliated with other labs working on like Alzheimer and Parkinson's. Actually, as of now, we're working with Brigham Hospital, both Parkinson and AD models. So my personal reason is that my mother's Alzheimer patient. So I'm seeing the whole process. We want to do something. And now nanobody is perceived as small. There's some nanobody can be engineered a way to go into the brain. But it's still, for most others, the blood-brain barrier is hygienic. It's very hard to go through. Some of the nanobody with a high PI, you can go in, but many still don't. So the way to do that is using the brain transporter, like you mentioned, the CD98. The more popular now is TFR1. So TFR1 been tried so many years, but now recently they have some success. Right? So from Roche, there's a A-beta drug itself doesn't work, but with their transporter, they can go into the brain and quite effectively clear up the A-beta plaques. So we want to develop a nanobody version of that. So that was a regular antibody. So a nanobody version can easily engage with other antibodies. Right? So whatever antibody you want to link to, such short piece, a lot easier to do. So that's going to be one of the first we want to do on that part. And then others, of course, we want to try every single thing to be tested. Insulin receptor, CD98. So basically a pipeline of those. We want to test out. They're very good in vitro models. And also we have mouse model for that too.
Can you speak about the challenges in dealing with this nanobody? Maybe half-life or engineering?
So we haven't, half-life is their solution for that. So one is that they hook up with FC. It's linked to FC. They turn into very similar to regular antibody. So that will sterilize it. Second is that you can link that with this, basically, albumin, like serum albumin, that will stay in the blood much longer. So, that part has been worked out with other companies. So engineering per se, we don't foresee much problem. One of the big advantages is that we have so many sequence servers. And because it's just one sequence, one nanobody, then you can actually use a sequence-driven way. I didn't get a chance to go through this, but very efficient. We can just go through the, immunized mice, take all this immune repertoire that includes all the antibody sequences. And then we'll go for just basically immunized mice and doing NGS bulk sequencing, because one sequence is one antibody, right? So then we get an entire repertoire. We can go from there to predict which one, might be better. And also we can eliminate certain things that having liability for development. And then more importantly, after you find binders, the efficiency like average 30%. Now we're working with Ginkgo Bioworks to utilize AI program, to further improve, selection on affinity and also the solubility and other developability assays. So then hopefully we can further increase the throughput. I think that's one major advantage. And after you find the hit, you can always go back to your library to find the entire family, right, and test it out. So then, in a way, you don't really need to engineer because you have so many candidates already evolved in the mouse, in vivo. I think that's the best way, to predict anybody's functionality. So that's I think the one additional advantage, other than being small, modular, is also very easy to use a sequence-driven way to for the discovery. So.
Any other questions?
Yes. So fantastic, promising technology. I think based on what I know, if you have an elephant slide and show the potential, right, and how much money you seek and how you create value for the investor,
right. Yeah.
Then right now, then time, right? How much money you need, how long going to take, and
right.
how much value created.
for each stage.
Actually, I do have slide I took out because my financial advisor asked me to take out. I guess a different use, right? So I'm looking for around like $5 to $10 million for this round for like pre-A, because we want to incubate some programs. And then from there, we hope to raise large sum funding, because a lot of investors they want to see the evidence, right? Not just the theory. So now we have a model, we haven't got the molecule yet. So basically this round I'm looking for about like $5 million.
Good time. So I think that'll conclude our presentation.
All right.
I think because Neuron Off sends apologies, there's been some family emergency, so they can't be with us today. So I think that's the end of the pitching.
Thank you.
Thank you very much. Now it's time for the judges to do some work. So I think Min, you already gave some instructions to the judges on the scoring.
Yeah, so all the judges just provided the rankings for these 4 teams. And so we can collect numbers and aggregate all of them. So we're going to have a retreat. And so 20 minutes retreat, and feel free to do the networking and also take some pictures over there. After 20 minutes, there are 2 more road shows. And 2 more great teams, they're going to do a road show. Each team have just 10 minutes without a QA, 10 minutes presentation. So. Yeah.
We have our last roadshow here by Sorana Therapeutics by Dr. Dan. And the stage is yours. We have 10 minutes. Thank you.
Thank you. Welcome.
Thank you, everyone. I really, really appreciate this opportunity. So it has been a long day for everyone. So now we are not competing, try to relax. And so it's my great honor to introduce what we are doing. So for Sirona Therapeutics, we are trying to target mRNA and it's modifying an enzyme for improving cancer treatment. The major challenge in cancer treatment is actually solid tumor. We know T cell immunotherapy so only show limited efficacy and effects for improving cancer patient survival. And targeted therapy has been considered really so good, but there are lack of targets in promoting a lot more targeted therapy. So we try to develop new drug targets. We try to develop small molecules, protect for all of these. New drug targets. So for targeting mRNA and mRNA biogenesis, mRNA modifying enzyme. So we believe we are quite unique because there are not a lot of companies show you. So what other companies are doing for targeting this. And we're trying to target billions of market for solid tumor treatment to avoid these problems brought by other targeted therapies. Toxicities and resistance. What we try to do is based on the founders of Sirona Therapeutics. They're a team, so long-term collaborators, originally from Harvard Medical School. I was a professor at Harvard Medical School before I moved to Duke, and so our founders shared the expertise of oncology, cancer drug development, and we have a medicinal chemist working with us to try to target the targets that we are trying to develop. So we have been contributing to a lot of cancer therapies. So by discovering new drug targets, and we try to bring these new drug targets and collaborate with many farmers and provide the approach for overcoming the resistance and look for more indications. But what we are trying to do right now is to bring our original target to the market by ourselves. So this is the pathway that we discovered. So with years of study, we actually proved how transcription and mRNA play important role to promote cancer cell survival. This is based on the high volume of transcription and a lot of problems cancer cells are dealing with. So in this process, cancer cells really rely on messenger RNA for cell survival, especially during this process, there are RNA modifying enzymes. The first pipeline that we are trying to work on is the enzyme called TRDMT1. So this is a RNA methyltransferase to modify mRNA and stabilize transcription machinery to promote cancer cell survival. So with years of study, so we've been publishing many papers in Nature review articles and how we can target this pathway in cancer treatment. And we also spend a lot of time and efforts to incorporate AI-based drugs design and build our in-house compound library and use them for compound screening. So we now own a lot of patents. For small molecules and products targeting this mRNA modifying enzyme and RNA methyltransferase and telomeres using RNA as a template. So we have the patents for the composition matters, indications, and also the biomarkers that we developed to guide the treatment of cancer. So we developed a cellular platform that enable us to launch new molecules continuously. So we have a lot of experience to detect the fit of mRNA, even it is unstable, even it's highly dynamic. So with years of study, we have a lot established in vitro and vivo assays to perform drug screening and further validate the effects of our compounds using vivo models. So I'll just show you one example, our first pipeline and our first lead compound. This is YW. So YW is a TRDMT1 inhibitor. We've done a cancer cell line screening in broad and showed what type of cancers, especially cancers that are hard to treat, ovarian cancer, glioblastoma, and there's a guidance of existing biomarkers either being used in the clinic or the biomarkers that we developed. So it is very effective to suppress tumor growth as a first-line therapeutic approach as a monotherapy drug. In addition, we know it actually synergizes with some of the existing therapies. For example, it synergizes with targeted therapy PARP inhibitor and synergizes with anti-PD-1 based therapy. This is one data I want to show you. So with low dose of the TRDMT1 inhibitor, we don't see much tumor suppression effects. And with low dose of anti-PD-1, we also don't see these tumor suppression effects. So high dose TRDMT inhibitor is 20 milligram per ml. So with oral GABA treatment, it significantly suppressed tumor growth. But when we use very low dose of TRDMT1 inhibitor and anti-PD-1, so tumor just stop growing. So we know the mechanism by changing the tumor microenvironment to activate esteemed pathway. So for ovarian cancer model, we also know it can synergize with the existing platinum chemotherapy. So the current achievement, because we are a university-based startup, so the current achievements with we've won several translational awards to support us. So for this translational purpose of study, so we have the in-kind support with the great chemist team for optimization of compounds. We now have backup candidates as the second, third generation of the compound. So the PK and the PD are very good about availability. So they are either suitable for oral gavage or IV treatment of patients. So we want to further develop our pipelines, and in the company, we build up a strong management team. So right now we have Duke and BA. So I recently moved to Duke and I built up the team there to help us for the management, and we both have strong background in pharmaceutical companies and experience in commercialization of drug products. And we have because we are now still in the university, so we really want to raise angel fund. So we have the support from Duke Translational Department. So they are supporting us to file the IND of our compounds, and we have the clinical team. We help us to start with phase 1 clinical trial once we pass the IND. Meanwhile, we want to get ready to launch out everything into the company. So we are now trying to raise angel fund, help us to to manage company, get the patent transfers from the university to our company, and setting up the R&D in the company. And we also see multiple access possibilities. So because we are already in this process, hopefully in next 1.5 years, so we are able to license out the existing compound and develop new compounds for future revenues. The last slide, I want to show you how unique we are. So spinning from mRNA vaccines for preventing cancer. So there's a long way to go. There are only 2 existing companies we try to target RNA modifying enzyme. One is in United States and the other is in Europe. But the pipelines that they are having and the targets that they are trying to work on are different type of modifications that are enriched during development. So a lot of them are relevant to neurodegeneration disease. So we are very unique to target them in cancer. We target unique type of modification, and we target unique enzymes. So we are still in the first class, and we have a set of pipelines. So I I don't have time for question and answers, but I'm here all the time. So please stop me and also reach out at Victor, so who is also here today, for any of your questions. Yeah, I'm done.
Thank you.
Thank you, Serrano Therapeutics. I know it's a very tense day, and we are ready to. Okay, so therapeutic. Oh, I'm sorry. Yeah, it's my bad. So it's your time.
Okay, so yeah, can you hear me?
Yes.
Okay, so let me share the slides. Okay, yeah, first of all, thank you for the organizer for this opportunity to present our company. So our company called Acon Pharmaceuticals. We are located in New Jersey. We also have a subsidiary company in Hong Kong and also in Shanghai. So we focus on the first-in-class siRNA inhalation drug delivery, as well as GalNAc siRNA. We're also developing the complex drug delivery system, including the LNP and the microspheres. So our advantage, we are a leading company in the niche area of lung targeting, and we can also target to the liver, and we're also developing a long-acting microsphere delivery systems. So we have our own IP for the GalNAc siRNA, as well as the lipids and the LNP formulation. We also have a patented long-acting microsphere injectables. This is our team. So myself is the founder and CEO of Acon Pharma. I work in different pharma companies before, including GSK and Johnson & Johnson. So our CSO is Dr. Li. She's a biologist, and she also worked for Vertex and Scripps before. And other team members, including Joseph in chemistry and Dr. Yang in pharmacology, and etc. And this is our product pipeline. So currently we have 3 areas. One is primary disease. So we are using the siRNA inhalation for the treatment of primary fibrosis and also lung cancer. And we are also using the GalNAc siRNA for the treatment of hemophilia and also high blood pressure. And in the CNS, we're currently developing a long-acting microsphere injection for the treatment of schizophrenia. So the first platform we have is the RNA-NP inhalation platform. So basically, we use the AI to assist to design the siRNA and mRNA, and we do the chemical modification. Then we use the AI to assist the drug delivery system optimization. Then we use the ANP inhalation device to deliver the siRNA. And we have done some animal studies to evaluate our siRNA efficacy inhalation technology. So from the animal study, for the RPF animal, the animal normally cannot survive more than 2 weeks. Basically, after 2 weeks, all the animal would die. But for the animal treated with siRNA inhalation, 80% animal will survive. And even after 3 weeks, 70% animal can survive. And we are also doing some breakthrough long-targeting siRNA and mRNA-NP by the IV injection. So from our study, so we found our LNP, we can achieve like close to 89% long targeting in 10 minutes after injection. And we also found our mRNA-NP can achieve about 92% long targeting in about 10 minutes. Our second platform is GalNAc siRNA platform. So currently we are using this platform to develop a product to treat hemophilia. So hemophilia affect about 1 million patients with about 15.8 billion market size. And currently we are developing the siRNA and targeting to antithrombin. The benefits for our treatment is first is a long term, so we can develop 1 month and 2 months injection instead of a current injection like every 3 days, which is very troublesome for the patients. And currently, our candidates can lower the antithrombin and promotes thrombin generation to restore hemostasis and prevent bleeding. Our third platform is long-acting microsphere platforms, and currently we are using this for the treatment of schizophrenia. So for this product, the currently the drug molecule is already available as oral dosage form, but is the patient need to take it every day, so it doesn't have good patient compliance. By using our technology, we encapsulate the drug into the microspheres to provide long-acting drug release over 1 to 3 months. So in this way, we can solve the patient compliance problem. And the patents are already granted in both China and US. Currently, it's pending in Europe and Japan. And currently, we are doing the CMC and also the PK studies for this product. And this is our scientific advisors. Most of them are college professors and also the doctors from the hospitals. And this is our prior experience, to develop siRNA drug delivery. So we publish several papers and also patents. And this is our business model. So we can either co-develop the product with our partners, or we can also license our product to other companies. So these some our competitors of our products. One company called Recode in California. So they are developing mRNA inhalation for cystic fibrosis, and they just get to phase 1 clinical studies. So their current valuation about 576 million. However, their AMP has some cytotoxicity issues. Ours AMP so far does not show any toxicity. And of course, for the siRNA, Alnylam is also developing a GalNAc siRNA for the treatment of hemophilia, currently in the phase 3. But our candidates has a better efficacy comparing to the Alnylam candidates. And the J&J has a long-acting Invega Sustenna injection for the treatment of schizophrenia. But our product is a third generation product with a better efficacy and a lower side effects compared to Johnson's second generation drug. And also we are using the AI to assist for the formulation development as well as the siRNA sequence design. So in this round, we plan to raise $5 million to give out 20% share to finish to R&D and submit R&D to the FDA and CDE. And in next round, we are going to plan to raise 20 million and to conduct phase 1 clinical studies. And in the long run, in 2028, we plan to go to IPO. Yeah, so this is my contact information. So if you are interested to invest or want to discuss with us on those products, please contact with me with the email, or you can also scan the WeChat, and we can connect through the WeChat. Or you can also just feel free to call me. Yeah, so that's it. Yeah, thank you very much.
Thank you.
All right. Hi guys. Thank you, Atrium Therapeutics, for your presentation. And I know it's an intense day, and after like 10 hours of competing, so I have the final winners from on my computer. And before that, once again, I'd like to thank our organizations, the Hong Kong Science and Technology Parks Corporation and Boston Chinese Investment Club, for organizing this great platform for this innovation. And right now, it's my absolute pleasure to announce the winners of the third Biomedical Pitch Competition. And our first place winner, whose groundbreaking ideas and exceptional presentations have found a new target for cancer immunotherapy, GV20 Therapeutics.
Thank you.
you. Congratulations.
All right, so if you want, I'll just take a picture with you.
Thank you.
Thank you.
Is it like Oscar? We don't have
Yeah, you should try.
You also have the music. Thank you. Maybe we stay.
All right, yes, yeah. So our second case for their visionary solutions in ADCs and TME platforms, Affinity Biopharma and Synesis Therapeutics. Congratulations.
Two teams.
Yeah, they are tied two teams. for the
Synesis?
second phase.
Yeah. Just one by one. Just one by one is better.
Yeah.
So
No,
Affinity
is
Affinity Biopharma for their visionary solutions in TME platform.
I think Diana, who's the representative from Synesis?
Then if Dana is not here, so
Okay, you
yeah,
can
so I will
represent.
be. I think it's here.
Yeah, congratulations.
Yeah, and also a tied second winner is Asus.
Is Synesis?
Yeah, because Dana, I think Affinity is online.
It's online? Are you?
No.
So,
She
okay.
presented.
So we finished the first, second places. Now goes to the third places. The 4 teams. The first team.
Right. Okay.
First, third place.
The first, third place. Okay, I got it. In Vera Group. Congratulations.
Probably online.
Yeah, probably online.
Yeah. Okay. So there's not, the result we can announce the post on the website as well. So you can make it. Let's go. Maybe we can go next one.
Okay, so our second, third place. Congratulations, Rapid Fusion Therapeutics.
Oh,
Thank you.
congratulations.
Thank you.
Thank you. Thank you. Thank you.
You're gonna hold it.
Yes, prototype. Congratulations. Thank you. Thank you. Thank you.
Oh, thank you.
Thank you.
So the next third place winner is Pure Genetics. Congratulations. Our last third place winner, also famous for the data-driven analysis, D2M.
Thank you.
All right, for the rest of the team, we also thank you for all your incredible presentations, and we'd like to award you the Innovation Award to all of you. Thank you so much for today's presentation.
We should call it.
Oh, okay, we should call it. So leveraging the first innovation award. Leveraging.
Congratulations.
Thanks.
Congratulations.
Our next innovation award is for I5 Therapeutics Online. I5 Therapeutics. I5. F5.
Five is fine.
Okay. Function 5.
And then
Our next innovation award is for Agilus Robotics Limited.
Angela is also sleeping.
Oh, yeah,
They're in Asia.
right. Our next award is for Spark Spine.
They-
Where is that? So they're probably online.
Oh, probably.
Congratulations,
Congratulations. Our next innovation award is for Azri Bell.
Zero
David?
by, I think it's on.
Oh, yeah, yeah.
Azri
Yeah.
Bell. Congratulations.
Thank you. Yeah.
So our last innovation award is another for a team that is leading, I guess. AI power painting blood test devices. Yeah, they're in Hong Kong.
Not sure.
Not yet.
They're in Asia. So,
They're not theranos.
Okay.
yeah. Okay, so
So maybe we should take a group picture together. So for the award winner who's holding the award, can we just sit on front? Can we just stand in front?
It's too small here.
It's too small. Maybe just go to the banner.
Maybe outside.
Yeah, we can go to the banner.
We have a banner in the front.
Banner.
okay, let's proceed to.
We're gonna.
Photo session.
Yeah.
Okay. Oh,